this time as compared to last night? Yes? Alright. So let us start off with where we left last night. We had finished with what are the factors affecting velocity of conduction, and we had discussed that diameter and myelin, right? Diameter and myelin, which is responsible, the factors which are responsible for conduction is axoplasmic diameter and the presence of myelin. We also discussed that large diameter fibers have a low axoplasmic resistance. And myelin has got, myelinated nerve fibers have got a high membrane resistance and a low capacitance. If you remember what we discussed yesterday, we said that the myelin increases the resistance but decreases the capacitance, isn't it? We said that cell membranes have a high capacitance, but the addition of myelin lowers the membrane capacitance. So a large diameter nerve fiber has low axoplasmic resistance, high membrane resistance, low capacitance. Then we discussed which are the nerve fibers which show the maximum velocity of conduction. We said nerve fibers with low axoplasmic resistance, high membrane resistance, low capacitance. Last, I discussed with you which are the electrical properties which favor conduction is. We discussed it is a low membrane resistance and a high capacitance. So this is how we had made a distinction, and hopefully you would have gone through the notes, yesterday's class notes, right? And so let's start. Today's a long day, so hopefully you'll be a little more patient than you were last night. Yes? Okay. So let's continue. The next thing which we're going to do with you is, which I'm going to do with you is nerve fiber classification. Nerve fiber classification. Now, nerve fiber classification is a fairly important topic as far as your MCQs go, because a lot of questions have come from here. Now, what is this nerve fiber classification there are two types of classification. There is Erlanger and Gasser classification. Erlanger and Gasser classification, right? This is a classification which is meant for all types of neurons, for sensory, for motor, as well as for autonomic neurons. Erlanger and Gasser classification. And the second classification is what is known as Lloyd and Hunt's. Lloyd and Hunt's numerical classification. Numerical classification. Now, this is only meant for sensory neurons, right? Sensory neurons. So you've got Lloyd and Hunt's numerical classification, Erlanger and Gasser classification, and Lloyd and Hunt. Double L, this is double L, OYD, and Hunt's, HUNTS, Hunt's numerical classification, right? This is the Erlanger and Gasser, GASSER, right? This is for sensory, motor, and autonomic. Right? Now let's see, nerve fibers have been divided into type A as far as the Erlanger and Gasser classification is concerned. It's type A, type B, and type C. Now as you go from A to C, the diameter decreases, and so also the velocity of conduction. That means, simple question, which is the largest diameter fiber? A. Smallest diameter fiber? C. Fastest conducting fiber is A, slowest conducting is C, right? So this is your, as you go from A to C, the diameter decreases and velocity decreases, right? Largest diameter, fastest conducting, smallest diameter, slowest conducting. And when you look at the myelination, type A and B fibers are myelinated fibers, and C is an unmyelinated fiber. C is an unmyelinated fiber. So you've got myelinated and unmyelinated fibers, right? Now A is further subdivided into A alpha, A beta, A gamma, A delta. A alpha, A beta, A gamma, A delta, right? Now, as far as the A alpha neurons are concerned, A alpha neurons, I have already discussed with you a little bit yesterday. A alpha neurons are your motor. They are motor as well as sensory. There are some motor neurons, A alpha. There are also some sensory neurons, which are A alpha. So, motor neurons are, A alpha, like I said, is somatic motor. Somatic motor, right? And A alpha also sensory. And they are also meant for proprioception. Somatic motor and for proprioception. The fastest conducted sensation is proprioception. Fastest conducted sensation is proprioception, right? Now, somatic motor, somatic motor, it has no number in the numerical classification. Isn't it? A alpha, which is a motor fiber, will have no number. Makes sense now, because your Lloyd and Hunt's numerical classification is only for sensory. But A alpha, which is meant for proprioception, is also known as type 1 fiber. Type 1 fiber. And this type 1 fiber is further subdivided into 1A and 1B. 1A is the sensory fiber from the muscle spindle. 1B is the sensory fiber from the Golgi tendon organ. Muscle spindle, please remember, is a receptor for muscle length. Golgi tendon organ is a receptor for muscle tension. These are both going to tell us what is they are both the receptors which are responsible for proprioception, right? Position sense. What is the length of a particular muscle, what is the tension in that particular muscle will be given by the muscle spindles, and the muscle spindle will give us information about muscle length. Golgi tendon organ will tell us information about the muscle tension. Afferent from what does it say? Afferent from the muscle spindle is type 1A. Afferent from the Golgi tendon organ is type 1B, right? This is the sensory fiber from these are the sensory fibers from these two important receptors. Afferent or the sensory fiber from the muscle spindle is 1A. Afferent from the Golgi tendon organ is 1B, right? Now, A beta, now this is your numerical classification on the right side, right? Now, let's go on to back to the Erlanger and Glasser classification. Now you have A beta. Dibrugarh, not to worry. Manipal, sorry, not to worry. I've just started. I've just told you the nerve fiber classification. There are two types of nerve fiber classification, Erlanger and Gasser, and Lloyd and Hunt's numerical classification. Erlanger and Gasser classification is for sensory, motor and autonomic fibers, where the fibers are divided into type A, type B and type C. Type A, type B and type C. A and B are myelinated nerve fibers. C is unmyelinated nerve fiber. A is divided into A alpha, A beta, A gamma, A delta. A alpha is somatic motor. A alpha fibers are also sensory. They are meant for proprioception. Now, the fibers which are meant for somatic motor, they will have no number in the numerical classification, right? Because why? The second classification I told is Lloyd and Hunt's numerical, which is only for sensory. So, A alpha, which are motor fibers, have no number in the numerical classification, but A alpha, which are for proprioception, are called type 1 fibers. So, type 1 fibers, fastest conducting fibers, largest diameter fibers, these are fibers which are further subdivided into type 1A and type 1B. 1A is the afferent from the muscle spindle, 1B is the afferent or the sensory fiber, afferent fiber from the Golgi tendon organ. So, that's all that I've covered right now. A beta is a pure sensory fiber. A beta is pure sensory. And what is A beta meant for? A beta is meant for fine touch. It is meant for vibration. it is meant for proprioception. But please understand now, proprioception only from muscle spindle. This is my type 2 fiber. Type 2 fiber. Now remember, I said numerical classification is meant only for the sensory fibers. Numerical classification is only for sensory fibers, and the A beta, which is a pure sensory fiber, this is fine touch, vibration and proprioception. Fine touch, vibration and proprioception, right? So this becomes my type 2 fiber. Type 2 fiber. A gamma is a motor fiber. In fact, there are only two motor fibers. One is A alpha, which is the motor fiber to the muscles, and A gamma is another motor fiber. This is motor to the muscle spindle. It is motor. When I do muscle spindle with you in CNS, we will again come back to this and we will discuss where does A gamma fiber go, right? So it is motor to the muscle spindle. Suffice to know at this stage is that A gamma is a smaller motor nerve fiber and it is motor to the muscle spindle. Now A delta is again a pure sensory fiber. Pure sensory fiber. This is meant for fast pain. This is meant for temperature. Temperature it is meant for cold sensation. So obviously this becomes now my type 3 fiber. A delta is meant for fast pain, temperature it is meant for cold sensation, and this now becomes my type 3 fiber, right? Then you come to B. B is preganglionic autonomic. Preganglionic autonomic. Now this preganglionic autonomic fiber will have no number. Obviously, because it's an autonomic nerve fiber. So this is preganglionic autonomic. C fibers are both sensory as well as autonomic. C fibers are postganglionic sympathetic. Again, no number in numerical classification. Numerical classification is only for sensory. And C fibers are meant for crude touch, pressure, slow pain, and temperature. Temperature it is meant for cold and warmth. This becomes my type 4 fiber. Type 4 fiber, right? So if you can just see this. Let's go over the entire thing once again. A alpha, somatic motor proprioception. The one which is for proprioception will have a number, that is 1. This is divided into 1A and 1B. 1A is the sensory fiber from the muscle spindle, 1B from the Golgi tendon organ. A beta is fine touch, vibration and proprioception. This is again proprioception but only from the muscle spindle now. And this is your type 2 fiber. A gamma is motor to muscle spindle. A delta is fast pain and temperature, like I said, this is type 3 fiber. And your B is preganglionic autonomic, right? This is it has no number, and whereas C is postganglionic sympathetic, and C fibers are also for crude touch, pressure, slow pain and temperature. This is for cold and warmth temperature. So this is your type 4 fiber, right? So this is your detailed Erlanger and Gasser classification as well as the numerical classification. Erlanger and Gasser only is for sensory, autonomic and motor fibers, whereas the Lloyd and Hunt's numerical classification is only for sensory. So now let's see what are the types of questions which have come from here. Let's try and solve these questions. Now my first question is, of course we've already done this, largest diameter type A, smallest diameter type C, fastest conduction type A, slowest conduction type C. Next question, which is unmyelinated? Which is unmyelinated? Is it type 1, type 2, type 3 or type 4? Right? Unmyelinated is type 4. 4 is the C type of fiber. 4 is C type of fiber, so this is unmyelinated. Next question. Which is the fiber which is meant for vibration? Fiber meant for vibration, A alpha, A beta, A gamma, A delta. So this becomes, for vibration is A beta, right? Next question. Which is the afferent or the sensory fiber from the muscle spindle? Sensory fiber from the muscle spindle. Is it only 1A? Is it only 1B? Is it 1A and 2 or is it 1B and 2? What is the answer? 1A and 2. Okay? Again, next question. Afferent from Golgi tendon organ. Same options as in the question above. Only 1A, only 1B, 1A and 2, 1B and 2. So afferent from the Golgi tendon organ is only 1B. Only 1B. Next, which is the fast pain fiber? The fiber for fast pain. This is A delta. Slow pain. This is C. Right? Then another set of questions. Which is the fiber for cold sensation? Only A delta or A delta and C? Only A delta or A delta and C? Yes, A delta and C. But for warm sensation, it is only type C. For warm sensation is only type C. For pressure, is it A, B or C for pressure? A, B or C? And answer becomes C. Right? So these are the kind of questions which have been asked from here. So that is why it becomes extremely important to know the nerve fiber classification. Another one, which is most numerous? Which is most numerous? Is it A? Is it B? Is it C? And your answer becomes C. C is the most numerous. The number of fibers, not A, Karamsad. I'll tell you why not A. Because please remember, A fibers are very large diameter fibers. And whenever, see, if A was most numerous, if A was most numerous, your nerves would have been as thick as ropes. Right? So A fiber cannot be the most numerous. It's the C which is most numerous. Mumbai, itch sensation, these are all a form of crude touch and pain. So this will be carried by C fibers. We'll be doing this when we come to CNS. We'll be doing this in detail. We'll be doing fast pain, slow pain, itch, tickle, all that. Right? So not to worry. It is the C type of fiber which is going to carry your itch sensation. Yes? So this is as far as your nerve fiber classification and questions which have come from here. Next, let's see susceptibility. Susceptibility. Now, as far as susceptibility is concerned, susceptibility to let's say pressure, hypoxia, and local anesthetics. Right? Now susceptibility. For pressure, that means which nerve fiber will be most affected by pressure? Most Aligarh, don't worry about muscle spindle, Golgi tendon organ. We still have CNS to do. All this will come in CNS, right? What if you've not understood something from what I've covered? Tell me that. Not to worry, I'll be doing muscle spindle and Golgi tendon organ, right? Now, as far as susceptibility to pressure is concerned, that means which are the fibers which are going to be most affected by pressure. Now, this is in pressure, it is type A fibers are most susceptible, then B, then C. You've heard of Saturday night palsy, isn't it? Saturday night palsy, that is when this person comes home drunk on a Saturday night and he sleeps with his arm around the chair like that, and there is now pressure on the brachial plexus, and in the morning he gets up with paralysis, motor paralysis, isn't it, of that particular arm. There is a palsy of that is called Saturday night palsy. So most susceptible to pressure are your A fibers, and if you remember, motor fibers are type A. Right? So, Saturday night palsy. So, this is pressure. To hypoxia, susceptibility to hypoxia, it is B is more susceptible than A and more than C. B is most susceptible. Now, local anesthesia. Now, local anesthesia is a little controversial in the sense that physiology insists that it is, physiology insists that this is C more than B more than A, right? C fibers are the most susceptible, but I think anesthesia insists that it is B fibers which are most susceptible. So as far as we are concerned, we have a reference in Genong. 24th edition also says the same thing. It says that C fibers are more susceptible to most susceptible to local anesthetic, right? So this is as far as your susceptibility is concerned. Now, in the C fibers, you know, are both sensory as well as autonomic. So remember this, that C fibers, like I said, for slow pain, which to follow. Alabad, I wish I had an answer for that. But as far as I'm concerned, physiology, there is a solid reference for it, and that is Genong, 24th edition in our first chapter. So as far as I'm concerned, C more than B more than A, right? This is, again, there was a question which was asked last time, and that said, which sensation is blocked first? Which sensation is blocked first when a local anesthetic is given? Is it pain? Is it temperature? Is it touch? Or is it pressure? Right? Which sensation is blocked first? Which of the following is blocked first by a local anesthetic? Now again, this is a direct lift from Genong. 24th edition of Genong has very specifically, there is a line which says, it is the pain sensation which is blocked first, then temperature, then touch, then pressure. It is very clearly mentioned in this one line in our first chapter itself. It's in a clinical box, and you could probably have a look at it if not convinced, right? So this is pain which is blocked first, then temperature, then touch, then pressure, right? And it also says that the same line goes on to say that motor fibers are most resistant to the action of local anesthetics, right? So this is a direct lift from a line given in our textbook. So this is as far as susceptibility goes. After this, after we've done the nerve fiber classification and the susceptibility, let's go on to the last part in neuron, and that is axoplasmic transport. Axoplasmic transport. Axoplasmic transport. Now neuron, as far as neuron is concerned, axoplasmic transport. Now a neuron, what is unique about the neuron is what is unique about the neuron is that the cell body and the end of the neuron are very far away from each other. So they need a system of transport. And there is what is known as axoplasmic transport, which will help to transport substances from the cell body towards the end of the neuron, right? Now, as far as Allahabad, I think you've asked me before I go on to this, just one small request to all of you, kindly just make a note of your questions, type it out in the break, and let me sort it out in the 15 minutes after that, right? But nevertheless, whatever 2 questions that you've asked me, let me try and answer it right now. Ludhiana has said, is it sensory more than autonomic, more than motor for local anesthesia? No, it's autonomic more than sensory, more than motor for local anesthesia. So, and you know, C fibers are both autonomic as well as sensory. So it's autonomic more than sensory, more than motor. Right? Allahabad has asked me a question which says that temperature warm or cold? No, it will be both warm as well as cold. Okay? So just a small request that kindly make a note of your questions, and I will try and answer as many as I can in the moment we come back after the break. Okay? So this is now axoplasmic transport. Now axoplasmic transport is this is due to the presence. Now, what does axoplasmic transport require? It requires the presence of tubes which are called microtubules. These are called microtubules, right? These are sort of hollow tubes which are present within the axon, right? They're hollow tubes. So, axoplasmic transport, what do I need? I need calcium, I need ATP, I need microtubules. These microtubules, like I said, are hollow tubes. How are these microtubules formed? Now, microtubules are formed of, there are proteins called alpha tubulin, beta tubulin, alpha tubulin, beta tubulin, alpha, beta, alpha, beta, alpha. These are proteins called alpha tubulin, beta tubulin. Alpha and beta tubulin. These sort of alternate with each other to form a hollow ring. There are 14 of these which form a hollow ring, right? They form a hollow ring, alpha, beta, alpha, beta, alpha, beta, alternate and form a hollow ring. Right? Then what happens is these rings get stacked one on top of the other. Right? These rings get stacked one on top of the other, and so it forms a hollow tube. This is your alpha and beta tubulin. Right? Now, the microtubules, therefore, they have what is known as a plus end and a minus end. And what is the plus end? Plus end is called the assembly end, and minus end is called the disassembly end. Assembly end mane where these rings are getting stacked one on top of the other is called the assembly end, and where they are being removed is called the disassembly end. So you've got a plus end and you've got a minus end. Microtubules, if you remember, are also required for mitosis, spindle formation. And you have antimitotic drugs which interfere with the assembly of these microtubules. So microtubules are also antimitotic drugs like paclitaxel. It interferes with the assembly of the microtubules. Okay? So microtubules in axoplasmic transport, what do? They serve as tracks. Tracks mane sort of a railway ki patri, right? And what moves on these tracks are chota gadis which are called molecular motors. molecular motors, right? And these molecular motors are kinesin, dynein, and myosin 5. Kinesin, dynein, and myosin 5. So this is what I require for axoplasmic transport. I need calcium, I need ATP, I need microtubules, I need molecular motors, right? So this is what I require. Now let's see what are the types of axoplasmic transport. What are the types of axoplasmic transport? So let's see this. Types of axoplasmic transport are, there is a fast axoplasmic transport, and there is a slow axoplasmic transport. Fast and slow. And fast can be fast anterograde, and fast can also be fast retrograde. Whereas slow is always anterograde. So this is fast anterograde and fast retrograde, whereas slow is always anterograde. Fast anterograde is 400 millimeters per day, the rate of transport. Fast retrograde is 200 millimeters per day, and slow, so the difference is in the rate of transport. 400 millimeters per day, this is 200 millimeters per day, whereas slow, which is always anterograde, is 0.5 to 10 millimeters per day. Now what is anterograde and retrograde? Yes, so let's see this. What is anterograde and retrograde? See, whenever the substance moves away from the cell body, towards the end of the axon, this is called the anterograde. And when it moves in a reverse direction, this becomes retrograde. So you've got fast anterograde and fast retrograde. Anterograde means away from the cell body, retrograde towards the cell body, right? So this is your fast anterograde, fast retrograde. So let's go back to what we were trying to do. What is the difference between fast anterograde, fast retrograde? Now fast anterograde, the molecular motor which is required is kinesin. And for fast retrograde, the molecular motor which is required is dynein. Fast anterograde is towards the plus end, where I have already described it to you. What is the plus end? The assembly end of the microtubule. So this is towards the plus end of the microtubule, whereas the fast retrograde is towards the minus end of the microtubule. One is towards the plus end and the other one is towards the minus end. Now the next point is, what travels in an anterograde fashion? What all travels in an anterograde fashion? Anterograde fashion mein, what goes in an anterograde fashion is whatever is produced by the cell body. Whatever is produced by the cell body. For example, neuropeptides. These are manufactured by the cell body, so they have to be transported to the end of the axon. So neuropeptides. Then vesicles, neuropeptides, vesicles, right? Number 3, mitochondria. Mitochondria move from the cell body towards the end of the axon. Reticulum, they also go from the cell body towards the end of the axon. So neuropeptides, vesicles, mitochondria, reticulum. Retrograde, what will go in a retrograde fashion is neurotropic viruses. Neurotropic viruses, right? Neurotropic viruses mane, for example, neurotropic viruses mane जैसे rabies virus, the bite is on a peripheral part. Now the virus will travel in a retrograde fashion towards the CNS. So neurotropic viruses, that is rabies. Clear? Then tetanus toxin. Tetanus toxin will also travel in a retrograde fashion towards the CNS. So tetanus toxin. Next, let's see. Nerve growth factor. Nerve growth factor. This will also travel in a retrograde fashion towards the cell body. So neurotropic viruses, tetanus toxin, nerve growth factor. Clear? So this is as far as your susceptibility is concerned. Okay? Sorry, this is as far as your fast and slow axoplasmic transport is concerned, right? So here you've got your fast anterograde, 400 millimeters per day, requires kinesin towards the plus end. What is going to be transported in a fast anterograde fashion is neuropeptides, vesicles, mitochondria, reticulum. Retrograde is 200 millimeters per day, right? Requires dynin towards the minus end, and what is transported in a retrograde fashion. So this is as far as your. Now there was an interesting AIMS question which was asked, and let us try and solve that. The AIMS question said that axoplasmic transport, all of the following are required for axoplasmic transport except A, is it microtubules? B, is it microfilaments? C, is it neurofilaments? And D, is it kinesin, dynin? Now we already know we need microtubules, we need kinesin, dynin. So now let us see which are the ones which are not required. Which are the ones which are not required for axoplasmic transport, and your answer becomes neurofilaments. Now what are these microfilaments? Microfilaments, like microtubules, are hollow structures. Microfilaments are solid tubes, and they are sometimes used as tracts. Not all the times, but they are sometimes used as tracts. Right? These are your microfilaments. But neurofilaments are never used in axoplasmic transport. What are these neurofilaments? These are proteins which help to maintain the structure. So microfilaments can be used for axoplasmic transport, but neurofilaments are never used. They are basically proteins which help to maintain. They help maintain the diameter of the axon. They help maintain the diameter of the axon. Right? These are your neurofilaments. Neurofilaments are never used. They help to maintain the diameter of the axon. So this is your all except, right? This is your axoplasmic transport. So once we are done with axoplasmic transport, let's see this. Let's go on to our. So this brings us to the end of our chapter on neuron, and let's go on to our next chapter, and that is skeletal muscle. Skeletal muscle, right? Please understand examples of slow transport. Please understand there is no difference in the fast and the slow axoplasmic transport. The same Jabalpur, yes, I'm answering that. The same substances will be transported. The ones which go by fast axoplasmic transport will also be transported, can be transported in a slow fashion. But please understand the difference is only in the rates. That's about it. Fast anterograde is 400 millimeters per day. Slow anterograde is 0.5 to 10 millimeters per day. Right? So that is why, so there is no difference in the examples. It's just the difference is in the rate. rate of the movement in slow anterograde. Slow anterograde is 0. So same substances, mitochondria, reticulum, if they are traveling in a slow fashion, slower speed of transport, then that becomes a slow exoplasmic transport. That's about it. Now, as far as skeletal muscle is concerned, let's take on the first thing, and that is what you know. Skeletal muscle is a striated muscle. It is a voluntary muscle. This is a striated muscle. This is a voluntary muscle. Yes, there are different types of proteins which are present. Let's discuss the different types of proteins. The proteins which are present in skeletal muscle. Number one are the contractile proteins. Contractile proteins. And the first contractile protein, of course, is actin. Actin. Now, there are two Mysore endocytosis? I don't know. A contractile protein is actin. Actin is, there are two strands of actin. There are two strands of actin in a double helix. There are two strands in a double helix. Just like a DNA molecule, you've got two strands in a double helix, right? Now, each of these strands, this is a polymer of F-actin. It is a polymer of F-actin. Each of these strands is a polymer of F-actin molecules. Two strands of actin are present in a double helix. Now, the second contractile protein is, of course, myosin. And the myosin which is present in the skeletal muscle is myosin 2. Myosin 2, why do I call it myosin 2? Myosin has got a tail. It has got a tail. It has got an arm. And it has got heads. And these heads are 2 in number. This is your myosin molecule. Tail, arm and heads, and the heads have got ATPase activity. ATPase activity, right? This is as far as your myosin is concerned. It has got ATPase activity. Now, please remember, at any given point of time, only one myosin head combines with actin. At any given point of time, only one myosin head combines with actin, right? So this is your, like I said, your myosin molecule, tail, arm and 2 heads, and these have got an ATPase activity, right? Next, let's see the second type of protein which are present in the skeletal muscle. These are your regulating proteins. Regulating proteins. Now, regulating proteins are tropomyosin and troponin. Now, tropomyosin, these are the 2 strands of actin, and tropomyosin now covers the active sites on actin. It moves along with actin, the one you can see in green. And what does this tropomyosin do? It covers the active sites on actin. See, myosin head will combine with the active sites on actin. So here, one molecule of tropomyosin covers 7 active sites on actin. It covers 7 active sites on actin. One molecule of tropomyosin covers 7 active sites on actin. The second regulating protein is troponin. And troponin is a globular protein. Troponin is a globular protein. It has got 3 components. there is troponin T, there is troponin I, there is troponin C, and now calcium attaches to troponin C. Calcium attaches to troponin C. And where is this troponin attached? Troponin is attached over here. Troponin is attached to tropomyosin. Right? It is attached to tropomyosin. Thank you. Tropomyosin. बेटा एक काम कर, थोड़ा सा चीनी है? हाँ? एक glass. So you've got troponin is attached to tropomyosin. Yes. So like I said, there are three components of troponin, that is troponin T, troponin I, and troponin C. Calcium attaches to troponin C. So let's look at the sequence here. What happens now? The moment calcium attaches to troponin C, there is a conformational change in troponin. Conformational change in troponin, right? And when there is a conformational change in troponin, it causes the tropomyosin to slide. Tropomyosin slides, and कोल्हापुर पढ़ो ना, ये क्या लिखा है? Covers 7 active sites on actin. Right? Now see what happens now. Calcium attaches to troponin C. This causes a conformational change in the troponin, and this causes the tropomyosin to slide. Tropomyosin slides and it goes deeper into the groove between the two strands of actin. Now the active sites on actin are exposed, and myosin head will now combine with actin. Right? It will combine with actin. So this is the sequence of your muscle contraction. Have you got it? So we can write it down so that it becomes clearer, and that is, let's see this. What happens now? Here now calcium attaches to troponin C. Calcium attaches to troponin C. Now there is a conformational change. Conformational change. Now this conformational change, this causes calcium attaches to troponin C, there is a conformational change, and this conformational change causes tropomyosin slides and exposes the active sites on actin. It exposes the active sites on actin, and now what do I get? I get the actin myosin cross bridge formation. Actin myosin cross bridge formation means actin myosin cross bridge formation and cross bridge cycling will now cause the muscle to contract. Have you understood? So let's see the sequence again. Calcium attaches to troponin C, conformational change, tropomyosin slides and exposes the active sites on actin. Actin myosin cross bridge formation, and now starts the muscle contraction. So this is your this is the sequence. This results in a muscle contraction. So here are your regulating proteins. Regulating proteins. So we've done contractile proteins. We've done regulating proteins. Let's have a look at structural proteins. Structural proteins. And the structural proteins are number 1, actinin. Actinin binds the actin to Z line. Number 2 is titin. Titin binds the Z line to M line. And M line, see what is the Z line? Between the two Z lines you have the sarcomere. Sarcomere, and we know that sarcomere is the structural and the functional unit of the skeletal muscle. So between two Z lines you have the sarcomere. And what is M line? M line is present in the middle of the sarcomere, right? M stands for middle. So it's present in the middle of the sarcomere, right? This is titin. Now titin becomes important for us because there's a question which has been asked, and that is, this titin is responsible for the elasticity of the skeletal muscle, skeletal as well as cardiac muscle. And this has been asked as a question, which is the protein responsible for elasticity of the skeletal and the cardiac muscle? So the protein responsible for elasticity is titin, right? And like I said, this has been asked as a question, so we should have no problem in answering that. The last one is desmin. Desmin. And what is desmin? Desmin binds the Z line to the plasma membrane. So these are your three structural proteins, actinin, titin and desmin, right? These bind the Z line to the plasma membrane. So we've done three sets of proteins. There was contractile proteins, actin, myosin. Then we did regulating proteins, which is tropomyosin and troponin. And I said that one molecule of tropomyosin covers 7 active sites on actin. Troponin, we said globular protein. And if you remember, I discussed with you in detail what is the function of troponin. Calcium binds to troponin C, and because of that there is a conformational change. And if you remember, I told you, because of the conformational change of troponin, tropomyosin slides and exposes the active sites on actin, which causes the actin-myosin cross-bridge formation, right? And the muscle contraction starts. So that was the regulating proteins. Next, I did with you structural proteins, actinin, titin, desmin. Yes. Now, let's have a look at bands. Which are the bands in a skeletal muscle, right? For example, this is the muscle fiber. Yes. Ludhiana, just be careful. Just listen to what I have to say. These are the two Z lines. And between the two Z lines is the sarcomere. Sarcomere. And what is a sarcomere? It is the structural and the functional unit of the skeletal muscle. This is the sarcomere between the two Z lines, right? Now, attached to the Z lines are actin. Actin is also known as the thin filament, right? This is your actin which is attached over here. Okay? Right? These are your two Z lines. This is another Z line. Attached to the Z line is the actin. And remember I said which is the protein which connects the actin to the Z line, that is actinin. Now present in the middle are the thick filaments, and the thick filaments are myosin. This is the thick filament or the myosin which is present. Clear? Thick filaments of the myosin is present over here. Let's have a look at the bands in a skeletal muscle. Bands in a skeletal muscle. This is known as the dark band or the A band. A stands for anisotropic to light. Anisotropic to light. Okay. All right. So like I said, the between the two Z lines is the sarcomere, and within the sarcomere are the A and the I band. So first we were going to talk about the A band. And what is the A band? Like I said, A band. Now, what does it consist of? It consists of only actin, only myosin, or myosin plus overlapped actin. And the answer is this is myosin. A band is myosin plus the overlapped actin. Myosin plus the overlapped actin. Myosin plus the overlapped actin. This is your A band, right? This is myosin plus the overlapped actin together form the A band or the dark band, right? Next is the I band. Let's see the I band. Now, I band is your lighter band. And this I band is over here. This is the I band, right? I band is isotropic to light, and I band consists of only actin. Plus, if you can realize this, that half of I band lies in one sarcomere and half lies in a neighboring sarcomere. Half lies in one sarcomere, half lies in a neighboring sarcomere. So this is your only actin, only actin. Half lies in one sarcomere, half lies in a neighboring sarcomere, right? Now, that means one sarcomere consists of half I band plus the A band plus half I band. This is one sarcomere between two Z lines. I've got half of I band plus the A band plus half of I band, right? Now, present within the A band, so you see this is half of I band plus A band plus half of I band, right? So this is between the two Z lines. One sarcomere consists of half I band, A band and half I band. Now, within the A band is a slightly lighter area. Within the A band, there is a slightly lighter area. And let's see this slightly lighter area, this one here. This is the H zone. This is the H zone. And what does the H zone consist of? H zone consists of only myosin. This is only myosin, right? H zone consists of only myosin, right? Now, what happens during a muscle contraction? When a muscle contracts, the two Z lines, they come closer to each other. When they come closer to each other, if this is the A band here, this is the I band moves into the A band. This is my right hand, what you're seeing over here, this is my I band, this is A band. A band, now I band moves into the A band, right? So during a muscle contraction, what is going to happen? What happens during a muscle contraction? What happens to the I band? I band decreases. I band will decrease. Yes, during a muscle contraction, I band will decrease. I band decreases. H zone decreases or sometimes even disappears. Isn't it? Because the I band is moving into the A band. So I band, H band will either decrease or even disappear. And what will happen to the A band? A band remains the same during a muscle contraction. A band remains the same during a muscle contraction. This has been asked as a question. Which band is unchanged during a muscle contraction? And your answer becomes the A band. A band is unchanged during a muscle contraction, right? So this is as far as your bands are concerned. These are your bands which are present. Let's go on to the next one. Next, let's have a look at what are sarcolemmal proteins. What are sarcolemmal proteins? Sarcolemmal proteins. Sarcolemmal proteins, also called the dystroglycan, sarcoglycan complex. Dystroglycan, sarcoglycan complex. Right? Dystroglycan, sarcoglycan complex. This is your sarcolemmal proteins. Sarcolemmal proteins. Yes? Dystroglycan, sarcoglycan complex. Yes? Now let's see this. Sarcolemmal proteins. Please understand that the muscle fiber is equal to the muscle cell. Muscle fiber is equal to the muscle cell. Right? Now muscle fiber, the cell membrane of the muscle fiber is therefore called the sarcolemma. Sarcolemma. Jammu, anisotropic means anisotropic to light. It is your dark band, right? Dark band, right? So muscle fiber is equal to the muscle cell, which is sarcolemma. Okay. Now let us see the membrane of the muscle fiber is the sarcolemma. So if you can just see over here, this is the sarcolemma here, and let's discuss what are the sarcolemmal proteins. Sarcolemmal proteins. Now, the first sarcolemmal protein that I'm going to talk about is this one. This is a beta dystroglycan. Beta dystroglycan. This is attached to the alpha dystroglycan. These are called your dystroglycans. Okay? This is ECF here, this is ICF, and this is of course your sarcolemma. Right? I've already discussed it with you that muscle fiber is equal to the muscle cell. Right? Now present here is उनका connections का देखो ना problem, वो एक सब sentence बोलते जा रहे हैं. हाँ? This is actin here. The two strands of actin in a double helix. Two strands of actin in a double helix. Linking the actin to the beta dystroglycan is a rod-like protein, and this rod-like protein is called what is called the dystrophin. This is dystrophin. And we know that absence of dystrophin produces a dystrophy known as Duchenne's muscular dystrophy, right? So this is your actin, linking the actin to the beta dystroglycan is dystrophin, and also associated with dystrophin is a small globular protein which is called syntropin, and a third protein over here which is called the sarcospan. This is what is known as sarcospan, right? So these are your important, you should know the names of these proteins, because what are the types of questions which come from here? Which of the following is not a sarcolemmal protein? Yes, so your sarcolemmal proteins are dystrophin, syntropin, sarcospan, dystroglycan. Also associated with the also associated with the dystroglycan is another set of proteins. Those are known as sarcoglycans. So let's see this. There is alpha, there is beta, there is gamma, and there is delta. Alpha, beta, gamma, delta, sarcoglycan. These are also your sarcolemmal proteins. Dystroglycan and sarcoglycan. These are your sarcolemmal proteins. Now this alpha dystroglycan is linked with a protein in the extracellular matrix which is called laminin. Laminin is in the extracellular matrix. Right? Laminin is in the extracellular matrix. So these are your sarcolemmal proteins. Dystrophin, syntropin, right? Dystrophin, syntropin, sarcospan, dystroglycan, sarcoglycan. Okay? These are your sarcolemmal proteins. Now see what happens in the case of Duchenne's. What happens in Duchenne's? Alapad, what is the problem? Syntropin is just it is linked with the dystrophin. It is a sarcolemmal protein. Right? This is the small globular protein which is called syntropin, and this is sarcospan. Right? Now we know that. Now see, these are your sarcolemmal proteins, and these are you know that whenever if there are mutations of these proteins, it gives rise to muscle weakness. Different types of dystrophies. There are almost 40 different types of dystrophies which are known, and the more important ones, when dystrophin is completely absent, this is known as Duchenne's muscular dystrophy. You know this, isn't it? The child suffers from proximal muscle weakness, and when he's trying to get up, he climbs onto himself to get up. So that is Duchenne's muscular dystrophy, when dystrophin is completely absent. If dystrophin is present but reduced, decreased amount of dystrophin, the kind of muscular dystrophy it produces is known as Becker's muscular dystrophy. Becker's muscular dystrophy. But if there is a mutation of the sarcoglycan complex, this gives rise to what is known as the limb girdle dystrophies. Limb girdle dystrophies. Yes, so these are three important dystrophies that we must know. Dystrophin is absent, that is Duchenne's muscular dystrophy. Dystrophin present. Dystrophin is present. This is called the Becker's muscular dystrophy, and limb girdle dystrophies in mutations of sarcoglycan, limb girdle dystrophy. Mysore, M-line, pseudo H zone, I'll tell you a little later. Just concentrate on these sarcolemmal proteins. I'll tell you after the break. I'll take these questions after the break. Please concentrate on what I'm trying to tell you right now. This is what are the different types of dystrophies. Now the question which has been asked from here is, it has come in AIMS, and that is, which are the proteins, which are, all of the following are sarcolemmal proteins except dystroglycan, sarcoglycan, dystrophin, and perlecan. So obviously your answer becomes perlecan. Perlecan is in the extracellular matrix. Extracellular matrix, right? So these are important. So you must know which are the sarcolemmal proteins. Yes, and again to repeat it, dystrophin, syntropin, sarcospan, dystroglycan, beta, alpha and beta dystroglycan, alpha, beta, gamma, delta, sarcoglycan. Please understand, dystrophin is attached to the beta dystroglycan, right? So these are your sarcolemmal proteins. Next, let's go on to neuromuscular junction. Okay, now let me, your questions are sort of, let me just finish your questions here, whatever you have asked, whatever doubts you have, Mysore and I think somebody else was also asking me pseudo H zone. So just see this once again and see what I was trying to tell you is, if this is the muscle fiber, remember I said muscle fiber is equal to the muscle cell, right? Now these are your two Z lines, and what are present between the two Z lines is the sarcomere, which is your structural and functional unit, right? Now here you have the actin molecule. Actin is attached to the Z line, and in between you've got the myosin, right? This is your myosin which is present here, right? Now, please remember, I told you there are tails, there is an arm, and there are heads of myosin. See, how is this myosin molecule arranged? Myosin molecule, these are the heads. These myosin molecules are arranged like this. And these heads go in all the 360 degrees. You see, the heads are arranged like that. So, if you can just appreciate it over here, the heads are attached in this way, right? And heads, they're like, they're like a group of golf sticks. So the heads are attached like this. Yes, I don't know if you can appreciate it here. Now, I told you what is the H zone. H zone, I said, is where there is only H zone is where there is only myosin. And what is pseudo H zone? Pseudo H zone is this one where there is actually no head. There is no myosin head which is present. Because please remember, the direction of the myosin head reverses in the middle. Heads this side and heads this side, right? It's something like this. I can just make a larger diagram here. The heads here will now be in this direction, right? So what's happened now? This part which has got no head forms the pseudo H zone. H zone is where there is only myosin, there is no actin, right? A band is myosin plus overlapped actin. H zone is where there is only myosin. Pseudo H zone is where there is actually no head of myosin. Now what is the M line? M line is formed by this reversal of polarity of the myosin heads, and this is found in the center of the sarcomere. This becomes your M line in the middle of the sarcomere, right? This is your M line, which is formed due to reversal of polarity of the myosin heads. The myosin heads have changed direction, so that's called the reversal of polarity. So that's how I've tried to simplify pseudo H zone and M line for you, right? So now let me go back to what I was trying to discuss with you, and that is your next topic, which is neuromuscular junction. Now, as far as the neuromuscular junction is concerned, let's see the neuromuscular junction. Now, neuromuscular junction is, there is an alpha motor neuron, and this is the muscle fiber, right? This is the A alpha motor neuron, and this is the muscle fiber here, and the junction between the two is called the neuromuscular junction. Okay. Now what happens over here is, you know that the alpha motor neuron is got a Aligarh, laminin is not a sarcolemmal protein. I told you it is in the extracellular matrix. It is not a sarcolemmal protein. I wrote in brackets over there that it is in the extracellular matrix. Ye dekho. It is in the extracellular matrix, right? This is your laminin, right? Let's have a look at neuromuscular junction. Now, neuromuscular junction, alpha motor neuron. This has got the vesicles which contain acetylcholine, right? Now, when the action potential reaches the end of the muscle fiber, this has got voltage-gated calcium channels. It has the presence of voltage-gated calcium channels. Voltage-gated calcium channels, right? So as when the action potential reaches these voltage-gated calcium channels, now there's going to be influx of calcium. Calcium enters into the alpha motor neuron, and this causes the remember I told you yesterday, for exocytosis I need calcium, I need ATP, I need docking proteins. So calcium has now entered into the alpha motor neuron, and now this is going to help us in exocytosis of acetylcholine. So there is exocytosis of acetylcholine. Yes, acetylcholine is released. This acetylcholine diffuses across the synaptic cleft. It now binds with the acetylcholine receptors. It binds with the acetylcholine receptors. And these acetylcholine receptors, what are these acetylcholine receptors basically? These acetylcholine receptors are nothing but ligand-gated sodium channels. Ligand-gated sodium channels. Acetylcholine binds with its receptors, with the acetylcholine receptors. These are your acetylcholine or the ligand-gated, these are the ligand-gated sodium channels. The moment acetylcholine binds with the receptors, these sodium channels open up, and when the sodium channels open up, there is influx of sodium, and now an action potential will be generated in the muscle fiber membrane. So let's see this again, transmission across the neuromuscular junction. First, the action potential reaches the end of the alpha motor neuron, causes the opening of the voltage-gated calcium channels, influx of calcium, exocytosis of acetylcholine. Remember, I told you for exocytosis, I need calcium, I need ATP, I need docking proteins. ATP and docking proteins are already present. Calcium has entered into the muscle fiber membrane. There is release of acetylcholine. Acetylcholine binds with its receptors on the motor end plate. And what are these acetylcholine receptors? Nothing but ligand-gated sodium channels. The moment the acetylcholine binds with its receptors, these ligand-gated sodium channels open up, there is influx of sodium, and an action potential is generated in the muscle fiber membrane. This is your transmission across the neuromuscular junction, right? Now, docking proteins, syntaxin, synaptobrevin are present over here in the alpha motor neuron itself, right? I told you yesterday that when botulinism, for instance, botulinism inactivates all the three docking proteins, syntaxin, synaptobrevin, SNAP25. So there is no exocytosis of acetylcholine which takes place, and that results in a flaccid paralysis. This is botulinum toxin. It results in a flaccid paralysis, right? Now, which is a disease of the presynaptic membrane? Presynaptic membrane, where antibodies are against the voltage-gated calcium channels. Disease of the presynaptic membrane. This is Lambert-Eaton syndrome. disease of the presynaptic membrane is the Lambert-Eaton syndrome, where antibodies are against the voltage-gated calcium channels. Ligand-gated sodium channels, antibodies against the acetylcholine receptors, disease of the postsynaptic membrane. This is the myasthenia gravis. Myasthenia gravis. So Lambert-Eaton syndrome and myasthenia gravis. So I've discussed these three basic points with you. Botulinum toxin inactivates the docking proteins synaptobrevin, syntaxin, SNAP25, produces flaccid paralysis. Lambert-Eaton syndrome, antibodies against voltage-gated calcium channels. Myasthenia gravis, antibodies against the acetylcholine receptors. This is your neuromuscular junction. Next, please see. Next, let's see what is denervation hypersensitivity. What is denervation? What is denervation hypersensitivity? Now, denervation, I've just discussed with you, this is the alpha motor neuron, this is the muscle fiber, right? And neuromuscular junction. Now, supposing there has been denervation. Denervation means this alpha motor neuron has got cut. Yes, maybe it's a surgical cut, it's a nerve injury, whatever. There is a section of this alpha motor neuron, right? Denervation. So obviously now the action potential will not be able to reach the end of the alpha motor neuron, and obviously there will be no exocytosis of acetylcholine. Right? No exocytosis of acetylcholine because the action potential cannot reach the terminal of the alpha motor neuron. Okay? Now, I hope you're familiar with what is upregulation of receptors and downregulation of receptors. Now, what is upregulation of receptors? This is whenever there is a hyposecretion of a hormone, decrease in secretion of a hormone, like for example, hypothyroidism. Now, what happens is at the target organs, there is an upregulation of receptors so that the small quantities of thyroid hormones can now produce its action on the target organs. So there is an upregulation of receptors, right? Supposing there is hyperthyroidism, this causes a downregulation of receptors. Downregulation of receptors, right? Now, in this situation where there is a denervation, now this results in, denervation means there is no release of acetylcholine now. On the muscle fiber membrane, we get an upregulation. क्या हो गया बरेली? क्या दुखी क्यों हो गए? Upregulation of the acetylcholine receptors on the motor end plate. Right? What has happened now? See this? There has been a denervation. Okay? There is no release of acetylcholine. Possible नहीं है ना? Acetylcholine का release नहीं हो पाएगा. There is a denervation, there is no release of acetylcholine. Now, this causes upregulation of acetylcholine receptors on the motor end plate. Upregulation. Now, you have higher number of receptors on the motor end. Motor end plate means the part of the muscle fiber which forms a junction with the alpha motor neuron. So it causes upregulation of acetylcholine receptors on the motor end plate. Now, when there is an upregulation, normally there are very small quantities of acetylcholine in the circulation, right? Very minute quantities of acetylcholine in the circulation, and this minute quantity of acetylcholine normally cannot affect muscle contraction at all. ये acetylcholine कहाँ से आता है? It comes from, it leaks from these millions and trillions of neuromuscular junctions that we have. So small amounts of acetylcholine releases. Or it comes from the autonomic ganglia. Leakage from these ganglia. Okay? So this very minute quantities of circulating acetylcholine is normally present. Normally it does not affect muscle contraction. Lekin now, when there is increase in the number of acetylcholine receptors, now what happens? The minute quantity of circulating acetylcholine, now this causes contraction. लेकिन किसका contraction? Is it causing the contraction of the whole muscle or just this individual muscle fibers which shows an upregulation of acetylcholine receptors, right? So it will cause the contraction of this individual muscle fiber. This contraction of the individual, it causes contraction of the individual muscle fiber, right? And this individual muscle fiber, the contraction of this individual muscle fiber is known as fibrillation. This is known as fibrillation, right? Will this contraction of the individual muscle fiber be visible to the naked eye? No, you cannot see this. Right? So you've got, let's see this again, once again, for the sake of Mumbai. What is this? Denervation, no release of acetylcholine, upregulation of acetylcholine receptors on the motor end plate. Now the minute quantities of circulating acetylcholine. Now the receptors have increased, so the small quantity of circulating acetylcholine which is present, which is in the circulation, which normally has no effect on muscle contraction, now manages to produce a contraction of this particular muscle fiber. It produces contraction of this particular muscle fiber, and this contraction of the individual muscle fiber is known as fibrillation. Fibrillation. Is this visible to the naked eye? No. This is not visible to the naked eye. It is not visible to the naked eye, right? This is fibrillation, right? So a question, what is the reason for, please understand this is different from ventricular fibrillation. This is fibrillation of the skeletal muscle fiber, right? So reason for fibrillation in skeletal muscles is your denervation hypersensitivity. Denervation hypersensitivity is responsible for skeletal muscle fibrillation. No Alabad, phantom limb, I will do it with you in CNS. This is not because of this. So this is denervation hypersensitivity. This is very, very typically seen, like I said, in the muscle fibers after denervation. So denervation hypersensitivity. आगे देखें, let's go on to the next one, and the next one is your sarcotubular system. Sarcotubular system. Now, what is the sarcotubular system? We've just discussed this, that there is the alpha motor neuron, there is the muscle fiber membrane, transmission across neuromuscular junction. I have already discussed it with you. Yes, we discussed that there was release of acetylcholine, there was acetylcholine binds with its receptors, and now there causes a generation of an action potential on the muscle fiber membrane, and this action potential travels all over the muscle fiber membrane, right? Now this muscle fiber membrane, it dips into the muscle fiber, and it forms what is known as the T-tubule. It dips into the muscle fiber and forms the T-tubule, right? It dips into the muscle fiber, it invaginates and forms the T-tubule, Remember, I've always said muscle fiber is equal to muscle cell, and so this is of course your sarcolemma. And this is ECF and this is ICF. ठीक है ना? यहाँ तक हम लोग कर चुके हैं, right? Now on either side of the T tubule is the L tubule. Either side of the T tubule is the L tubule, the longitudinal tubule, right? This L tubule is nothing but a storehouse of calcium. It is a storehouse of calcium. Now let's see what is the sequence of muscle contraction, or what happens, what is excitation, contraction, coupling. See this action potential, it travels all over the muscle fiber membrane. It now reaches the T-tubule membrane. On the T-tubule, you have a receptor which is known as dihydropyridine receptor. Dihydropyridine receptor. Now, what is this dihydropyridine receptor? This dihydropyridine receptor is a voltage sensor. Voltage sensor. That means it detects a change in voltage. Present on the L-tubule membrane is another receptor called the ryanodine receptor. And what is this ryanodine receptor? You know this, if there is a mutation of the ryanodine receptor, it produces a condition known as malignant hyperthermia. And what is this ryanodine receptor? This is nothing but a ligand-gated calcium channel. Ligand-gated calcium channel. So this is a dihydropyridine receptor, which is a voltage sensor, ryanodine receptor, which is a ligand-gated calcium channel. Right? अब होता क्या है? Now the action potential reaches the dihydropyridine receptor. Dihydropyridine receptor is a voltage sensor. The moment the action potential reaches over here, there is a conformational change in dihydropyridine receptor. And this conformational change, now between the dihydropyridine receptor and the ryanodine receptor is a small molecular connection. So the moment the action potential reaches the dihydropyridine receptor, there is a conformational change of dihydropyridine receptor. This causes the ryanodine receptor to open. It causes the ryanodine receptor to open, and now this calcium which is present in the sarcoplasmic reticulum, this calcium now comes into the sarcoplasm, right? फिर से action potential reaches the dihydropyridine receptor. Between the dihydropyridine receptor and the ryanodine receptor is a small molecular connection. This causes, whenever the action potential reaches the dihydropyridine receptor, conformational change of dihydropyridine, causes the ryanodine receptor to open. This releases the calcium. Now this calcium will go and bind with the troponin C, जिसका एक conformational change होगा, जिसकी वजह से tropomyosin slide करेगा. Active sites on actin are exposed and the actin-myosin interaction starts and the muscle contraction starts. Yes, so this is what is known as your excitation, contraction, coupling. This is excitation, contraction, coupling. Mane, now excitation, which is the electrical activity, has got converted into a mechanical activity. The electrical activity has got converted into a mechanical activity. So this is your excitation contraction coupling. And which is the excitation contraction coupling agent? Agent is calcium, right? Electrical activity has got converted into contractile activity. So that is what is known as excitation contraction coupling, right? And the excitation contraction coupling agent is calcium. Clear? Now this is, so that means if I ask you this question, what is the trigger for muscle contraction? Trigger for muscle contraction is availability of calcium in the sarcoplasm. And what is the source of calcium over here? Sarcoplasmic reticulum, right? Now that means a muscle will remain in a state of contraction till. 2-3 questions which I've asked you here. Let me just write them down for you. So this is reinforced. First question was, what is a trigger for muscle contraction? Trigger for muscle contraction is the availability of calcium. Next question. Trigger for muscle contraction is availability of calcium. Next is, a muscle will remain in a contracted state till. Muscle contraction continues till. Till calcium is available, or till ATP is available. Please understand, muscle contraction will continue till calcium is available, right? Because please remember, till calcium is available, sarcoplasmic calcium is present, there will be active sites of actin are exposed, and if active sites of actin are exposed, the actin-myosin interaction takes place, and actin-myosin interaction ka matlab hai ki there will be sliding of actin and myosin, which produces muscle contraction. Right? So till calcium is available, muscle contraction will continue. That means if I want to produce a relaxation, then this calcium has to go back into the sarcoplasmic reticulum. And how does it go back into the sarcoplasmic reticulum? It goes back, see, when a muscle has to relax now, what will happen? When a muscle has to relax, sabse pehle to ye we want to terminate contraction and now start the relaxation. सबसे पहले तो ये action potentials आना बंद हो जाएगा. Action potentials आना बंद हो जाएगा, right? And if the action potentials have stopped, these action potentials have stopped, now what will happen? There is no release of calcium from the sarcoplasmic reticulum. There is no release of calcium from the sarcoplasmic reticulum, and whatever calcium is present in the sarcoplasm now goes back into the sarcoplasmic reticulum by a primary active transport, and this primary active transport is known as SARCA, sarcoplasmic endoplasmic reticulum calcium pump. This is a primary active transport. Remember, I told you yesterday, if I shade the carrier protein, that becomes a primary active transport. If I shade the carrier protein, here you have a protein which is shaded. So this is your primary active transport. Okay? This is your sarcoplasmic endoplasmic reticulum. So that means to produce a muscle relaxation, I must push the calcium back into the sarcoplasmic reticulum, so that now what will happen? The tropomyosin will again go back and cover the active site on actin, so that the actin-myosin interaction stops. This is SARCA, sarcoplasmic endoplasmic reticulum calcium pump. Mane, now for relaxation also, I need the presence of ATP. Why do I need ATP? For the activity of SARCA. SARCA is primary active transport, right? I need ATP for muscle contraction, for the sliding of actin and myosin, I need ATP. So ATP is required for muscle contraction. ATP is also required for muscle relaxation. In other words, if I ask you this question, relaxation is an active process. Relaxation is a passive process. or is it an active process? My answer becomes active. Why active? Because I need ATP to pump the calcium back, to send the calcium back into the sarcoplasmic reticulum. Is it clear? To send the calcium back into the sarcoplasmic reticulum, I need ATP. So this is SERCA, right? Now answer this one question and then I'll take a little break, and that is, this was asked last year in DNB. Let me see if you can answer this. Supposing I inhibit SERCA activity. Supposing by use of a certain drug, I inhibit SERCA activity. What will happen to the duration of contraction? Does it increase? Does it decrease? Great. You've all got it. बीकानेर decrease नहीं. सुनो. SERCA activity कम होने का मतलब क्या हुआ? कि now calcium remains in the sarcoplasm. Yes? और जब तक calcium is available, तब तक muscle एक contracted state में रहेगी. So if I decrease the SERCA activity, now you have removal of sarcoplasmic calcium decreases. That means my duration of contraction will increase. Duration of contraction will increase, right? So this is your decrease in SERCA activity. Clear? Chalo. I'll take a 10-minute break, right? And then I'll continue. Take it. And meanwhile, whatever questions you have, you can just type them down, right? Okay, let me take your questions one by one. Kolhapur says, trigger for muscle contraction in options both calcium and action potential. Then what is going to be the answer? Answer has to be calcium. Calcium is the one which is responsible for conversion of the electrical activity, which is the action potential, into a contractile activity. So Kolhapur, your answer will be calcium here. Now Karamsat says magnitude of RMP is inversely proportional to the excitability. Lower RMP, so why more excitable? Please understand that the RMP is a unique feature of every tissue. Have you understood? Now the unique feature in the sense that the RMP of different tissues is different. You've got a large skeletal muscle fiber. Now this RMP is dependent on, number one, the gradient of potassium, and number two, the cell membrane permeability for potassium. Yes, we've discussed this yesterday, right? So every tissue will have its unique RMP. RMP does not determine the excitability. When you compare it, it does not mean that a tissue with minus 90 millivolts is less excitable as compared to a tissue which is minus 50 millivolts, right? Excitability will change if the resting membrane potential of that tissue changes. If it becomes more negative from whatever it is it RMP, then the excitability decreases, right? Now when I say, smooth muscle has an RMP of minus 50 to minus 60, what is going to determine the excitability is the response. How quickly does the cation, in smooth muscle it is because of influx of calcium. So how quickly does the calcium come into the muscle fiber? So that will determine the excitability, right? And if the smooth muscle RMP now, instead of being minus 50, it becomes minus 70 millivolts, then of course its excitability will decrease. So please remember, excitability will increase or decrease depending upon the potential with respect to its RMP. You cannot compare it for different tissues because every tissue will have its own unique resting membrane potential. Okay, so what will be the limiting factor for muscle contraction? Calcium or ATP? Please understand that, Gorakhpur, it will be calcium, not ATP. You must understand that ATP, if we are alive, ATP is never exhausted. ATP cannot be exhausted. In normal physiological conditions, you will not get exhaustion of ATP. ATP is will be exhausted only post-mortem, after death, and that is what is responsible for the rigor mortis, right? Now this was function of troponin I. What is troponin I? I stands for inhibitory, right? So it's no function as such. You've got a calcium attaching to the troponin C. Troponin T is attached to tropomyosin. Troponin I is inhibitory. During whenever there is no calcium which is attached, it is basically does not allow the interaction between tropomyosin, between the actin and myosin to take place. Phospholamban, I will tell you in cardiac muscle, there is no phospholamban in the skeletal muscle. Yes. What is the difference between fibrillation and fasciculation? See, Ludhiana, what is fibrillation? Fibrillation is the contraction of an individual muscle fibers, or at the most a group of muscle fibers, which is not visible to the naked eye. And what is fasciculation? Fasciculation means now a fascicle. Fasciculation would mean that there is fasciculation is made, that means now it is a contraction of a bundle, of a fascicle, muscle fascicle, right? And this fasciculation will be visible to the naked eye. And fasciculation is because of an abnormal discharge of spinal motor neurons. Abnormal discharge of spinal motor neurons. Okay, neurofilaments and microfilaments are made of trigger for SERCA activity. Trigger for SERCA activity is, number one, the action potential has to stop coming. Only then SERCA will get activated. Have you understood? There has to be no further release of calcium. Then the SERCA gets activated. So that will be your SERCA activity. How is ryanodine receptor related to hyperthermia? I told you, if there is a mutation of if there is a mutation of the ryanodine receptor, this causes mutation of the ryanodine receptor. Now what happens? What is malignant hyperthermia? When this patient is exposed to inhalational anesthesia, halothane, ether, right? Now there is uncontrolled release of calcium, release of calcium, and that uncontrolled release of calcium is what causes the shivering, muscle contraction, shivering, and the temperature shoots up. It's an anesthetic emergency. A patient with mutation of the ryanodine receptor, will be able to mutation of the ryanodine receptor. There is malignant hyperthermia, uncontrolled release of. Can we see the fibrillation in the tongue? I'll have to check up, right? It is difficult to, fibrillation in the tongue muscles. Yes. Okay. Neurofilaments, what is this now? I think more or less we've answered most of the questions. Let's go on to the next one. And the next is, denervation is responsible for fibrillation in lower motor neuron disease. Absolutely right. What is the difference between T-tubules and I-tubules? Mangalore? T-tubule kya tha? T-tubule, Hyderabad, Emirin, I do not know what you're talking about. Orthodromic, Meeraj, orthodromic, antidromic is conduction of the action potential. Again, I will do it with you in CNS. Mangalore, what is the difference between T-tubule and L-tubule? T-tubule is the transverse tubule, which is formed by the invagination of the sarcolemma, and L-tubule is nothing but the sarcoplasmic reticulum. It is sarcoplasmic reticulum. Physiological tremors similar to fasciculations? No, physiological tremors are what you see whenever there is increase in sympathetic physiological tremors will be seen whenever there is increase in sympathetic, right? So this is not, that is not what is fasciculation. Fasciculation or fibrillation, it is because of fasciculation. I told you Aurangabad is because of an abnormal discharge of spinal motor neurons. Abnormal discharge of spinal motor neurons. Prashant. Okay. All right. So let's go ahead. Now the next thing which I want to do with you is what are the types of different muscle fibers. Types of muscle fibers. Now types of muscle fibers are, number one, there are two types of muscle fibers. There is a type 1 and a type 2. Type 1 and type 2. Now type 1 fiber is SOR. Type 1 fiber is a small diameter. It's a small diameter, it's a slow fiber. So this is, how do I remember this? Type 1 is a very sorry looking fiber. Sorry looking fiber mane? Now what is this SOR? S stands for, this is slow, this is small, and this is oxidative and red. Right? So this is slow, small, oxidative and red. This is your type 1 fiber. Type 2 fiber on the other hand, if this is type 1 is slow, this becomes a fast fiber. This is a large diameter fiber. This is a glycolytic metabolism. This has a glycolytic metabolism. Type 1 has oxidative, type 2 has got glycolytic. Type 1 is red, type 2 is white fiber. So these are your type 1 and type 2 fibers. Slow, small, oxidative red, whereas type 2 is fast, large, glycolytic, white. Next, let's look at, once you know the basic differences between type 1 and type 2, then it is not very difficult to answer questions. So my first question, which has got a larger diameter? Which has got a larger diameter? Type 1, type 2? Type 2, right? Next question, which one has got a faster myosin ATPase activity? Type 1, type 2. Faster myosin ATPase activity. Again, type 2. Right? Next question. Which one has a longer twitch duration? Twitch money? Twitch duration money? It is the duration of the muscle contraction. Or contraction. Twitch is the contraction of the individual muscle fiber. Longer twitch duration will be type 1. Right? Next question. Which has got more mitochondria? Type 1, type 2. More mitochondria. Again, type 1. Oxidative metabolism. It needs more mitochondria. Jabalpur, be careful. This is type 1 fiber will have more mitochondria. Oxidative metabolism. Right? Next, which one has more cytochrome enzymes? More cytochrome enzymes. Again, this is type 1. Right? It has more cytochrome enzymes. Next, which one has a higher capillary density? Which one will have a better blood supply? Higher capillary density will again be type 1. Isn't it? The one which needs more oxygen. Higher capillary density, the fiber which needs more oxygen. And which is a fiber which needs more oxygen? Which has got oxidative metabolism. Chennai, be careful. Oxidative metabolism is type 1. So it will need more oxygen. So better blood supply. So higher capillary density. Next question. Which one will have more which one will have more myoglobin? More myoglobin. More myoglobin. Again, what is myoglobin? Meerut. Myoglobin is a storehouse of oxygen. Which one needs more oxygen? Type 1. So myoglobin will be more in type 1. Have you got it? It will be more in type 1. That is why the muscle fibers appear red. Right? So please understand, please remember that type 1 fibers are meant for, these are slow, small, oxidative red. Type 2 is fast, large, glycolytic white. Right? Next question, fatigability. Which one will fatigue earlier? Fatigability is early. Fatigability sets in early in which type of muscle fiber? Type 1 or type 2? Your answer is type 2. This is a faster fiber. Faster, जो तेज भागता है वो जल्दी थकता भी है. So your faster fiber will tire easily. Fatigability. Right? Mysore, what is twitch duration? Twitch duration means for how long does it remain in a contracted state. That's called a twitch. Twitch duration. Twitch duration, you talk in terms of a single muscle fiber. Right? So this is your fatigability. Fatigability is early in type 2 fiber. Clear? Now, that means your type 1 fiber is made, type 1 fiber, this is responsible for slow sustained contractions. Slow sustained contractions is your type 1 fiber. Whereas type 2 fiber is meant for brief and powerful contractions. Brief and powerful contractions are your type 2 fiber. Brief and powerful contractions. Right? So slow sustained contractions type 1, brief powerful contractions type 2. See, a muscle will have both types of muscle fibers. A muscle is made of both types. Right? You cannot have only type 1 and only type 2. Have you understood? A muscle will have both. Now, but it depends upon the functioning of the muscles. Yes. For example, your muscles at the back, which are contracting throughout the day, I need more of type 1 fibers. Slow sustained contractions. Have you got it? So a muscle, wherever I need, whichever muscle is required for a brief, powerful contractions, you will have predominantly type 2 fibers, but slow sustained contractions will be type 1 fibers. So this is as far as your distinction between type 1 and type 2 fibers is concerned. Next comes what is known as the size principle. Let's see what is the size principle. What is the size principle? Size principle states that, see, like I told you, in a muscle, you'll have both types of muscle fibers. Size principle states that whenever a muscle starts contracting, type 1 fibers will be utilized first, and then, if need be, type 2 fibers will be recruited. Have you got my point? When a muscle starts contracting, only the type 1 fibers are utilized first, and then the type 2 fibers, right? In fact, if you ask me, the type 2 fibers are a sort of a reserve. They are a sort of a reserve in a muscle contraction. Type 1 fibers are utilized, and then type 2. Type 2 is sort of a reserve in a muscle contraction, right? If I need extra power, if I need extra strength, then the type 2 fibers will be utilized. Type 1 first, then followed by type 2. This is the size principle. Answer this question. Which of the following is true? A. Fast muscle fibers are recruited before slow muscle fibers. True, false? Bolo. False. Second option. अरे Mysore, थोड़ा सा तो रुक जा बेटा. Let me just finish this. I will answer your questions. I'm just coming to it. It's the next one. Which are the motor units recruited during walking? Now listen to this question first. Fast muscle fibers are recruited before slow muscle fibers. You said false. Next question. Large muscle fibers before small muscle fibers? False. Number 3. Fast muscle fibers before slow muscle fibers? Again, false. And the last one, well perfused muscle fibers before poorly perfused muscle fibers? That becomes true. Please remember, it is type 1 followed by type 2. So my first option I told you, fast before slow, false. Large before small, false. Stronger before weaker, false. But well perfused before poorly perfused, yes, because type 1 fibers are your well perfused fibers. So this is your size principle. Now the next thing over here is, what is a motor unit? What is a motor unit? Now when I look at a motor unit is a single A alpha motor neuron. This is a single A alpha motor neuron along with all the muscle fibers it supplies. A single A alpha motor neuron along with all the muscle fibers it supplies. This is known as a motor unit. A alpha motor neuron plus all the muscle fibers it supplies. This is one motor unit. Right? Now please remember, one motor unit will have only one type of muscle fibers. There is only one type of muscle fibers which is present in one motor unit. That means a motor unit will have either type 1 fiber or a type 2 fiber, but never a mixture. In one motor unit, you will have only one type of muscle fibers. So this is a motor unit. A alpha motor neuron plus all the muscle fibers it supplies forms one motor unit. Right? Now, how many fibers in one motor unit? This is my next point. How many muscle fibers in one motor unit? Now, this again depends from muscle to muscle, right? Whichever muscle requires a fine control, I will have smaller number of muscle fibers in that particular motor unit. For example, if you look at the extraocular muscles or the ocular muscles, now ocular muscles have got a very fine control. So in the ocular muscles, I have 3 to 5 muscle fibers per motor unit. 3 to 5 muscle fibers in one motor unit. But look at the muscles of the back. Muscles of the back where I do not need a fine control. Muscles of the back where I do not need a fine control. Here you have almost 600 muscle fibers. 600 muscle fibers in one motor unit. Per motor unit. Right? 600 muscle fibers in one motor unit. Right? So this is your, sorry, this has not come out as 600. 600 muscle fibers in one motor unit. So these are your ocular muscles, 3 to 5 muscle fibers in one motor unit, and muscles of the back where I do not need very good control. You have almost 600 muscle fibers in one motor unit. Right? This has been asked as a question in DNB this time. Ocular muscles have number of muscle fibers in one motor unit of ocular muscle. Is it less than 10, less than 100, less than 1000, less than 10,000? So your answer becomes less than 10. Right? So this is, because that means this is now a fine control. Because of smaller motor units, there is a very fine control which is present over here. Clear? Now this is as far as our motor unit is concerned. Now let's see a little bit about how nomenclature of these motor units. Nomenclature of these motor units. Now I already told you type 1 fiber is slow oxidative. Right? This is my muscle fiber. And the motor unit. Now the motor unit which has got, remember I said in one motor unit you can have only one type of muscle fiber. So a motor unit which contains only the type 1 fiber, the name of that motor unit will now be slow and resistant to fatigue. Slow and resistant to fatigue. Make sense now? Type 1 fiber has a delayed fatigability, right? So a motor unit which contains only type 1 fiber is slow and resistant to fatigue. Now just to make life difficult for you, the muscle fibers, the type 2 muscle fiber is further subdivided into type 2A and type 2B. We've done type 2. I told you type 2 is fast, glycolytic and white. Yes, fast, large, glycolytic, white. But further subdivided into type 2A and type 2B. So what is type 2A? Type 2A is fast oxidative glycolytic. Fast oxidative glycolytic. Whereas type 2B is only fast and glycolytic. माने क्या? Type 2A fiber में there is also some amount of oxidative metabolism. Type 2B is fast and glycolytic only. Now obviously the motor unit which has got type 2A fiber, now the fatigability will be less as compared or delayed or slower as compared to the type 2B fiber because it has got oxidative metabolism. So the motor unit which has type 2A fiber becomes fast and resistant to fatigue. Fast and resistant to fatigue. Type 2B fiber, this is now motor unit with type 2B fiber becomes fast and fatigable. Slow and resistant to fatigue is a motor unit which has only type 1 fiber. Fast and resistant to fatigue is a motor unit with type 2A fiber. Fast and fatigable is the motor unit with type 2B fiber. Right? Now comes my sword. Now pay attention to the question that you were trying to ask me. When we are in a standing position, during standing, your SR motor units will be utilized, right? While you're standing, SR motor units are being utilized. Now, if I start walking, then the FR motor units will be utilized. But if while walking I start running, yes, now the FF motor units will be utilized. So this is how it goes. In the standing position, SR, but आप खड़े हुए हैं and you start walking, now the FR motor units will be utilized, and while walking you start running, then the FR. This is your size principle, isn't it? I told you that when a muscle starts contracting, पहले जो छोटे fibers हैं, they are going to be used, then the larger ones. So पहले type 1, फिर type 2A, फिर type 2B. Right? And the motor units, slow and resistant first, then fast and resistant, and then fast and fatigable. Whenever we need extra power, extra strength, then fast and fatigable motor units. So this is as far as your motor units is concerned. Right? Next, let's go on to what are the types of muscle contraction? What are the types of, Mysore, you must understand that which muscles are predominantly, now see this, like I said, a muscle will have both types of muscle fibers. Number one, it depends upon the function of that particular muscle, right? And number two, there is also the variation also is determined by your genetic, right? It's also a genetic makeup, right? Like for instance, if you look at soleus is more responsible, what you're asking me, soleus is more responsible for slow sustained contractions. So we will have more of type 1. Whereas gastrocnemius, will have more of, but again, it depends upon also the genetic, not just the use of that particular muscle, it also depends upon the genetic composition, right? For example, if you see this, see what happens in China is, China, the children from a small age group are recruited into their athletic program. Yes. Now, how do they decide a 5-year-old child will be a better sprinter or a better marathon runner? What do? They analyze the muscles. Yes. If the gastrocnemius of this particular child has predominantly type 1 fibers, that means this child will be better suited for marathon running. But if you have a child which has predominantly type 2 fibers, that means this child will be better suited for sprint running. Sprint running, I need sprint running, it's brief and more powerful. So that this child will be better suited for sprint running. Have you got it? Like when they say that an athlete is born and not made. Yes, that means the genetic composition also determines what kind of an athlete you're going to be. Not all of us become athletes. Yes, not that all of us have tried, but then the point remains that there is there is a genetic element to it. What is the composition of the muscles? Yes. Now, this was your motor units. Next, let's see what are the types of muscle contraction. Types of muscle contraction. There is an isotonic muscle contraction and there is an isometric muscle contraction. Isotonic and isometric. Now what happens in isotonic is, here the tone remains the same, but the length decreases. Whereas in isometric, the length remains the same, and the tone increases. Tone remains the same and the length decreases. Isometric, length remains the same and the tone increases. This is isometric. Now isotonic, external work is done. External work is done. Isometric, there is no external work is done. No external work is done. See, it's like very simple. Supposing, like for instance, if I do a flexion at the elbow, this is an isotonic contraction. Yes. There is a decrease in the length of the biceps and this is an isotonic contraction. But if you stand in front of the mirror and you start seeing your muscles like that, what are you trying to do? You're doing an isometric contraction. There is an increase in the tone of the muscles without any change in length. So isotonic contraction, external work is being done, but in isometric, no external work is being done. Next, the heat released is more in isotonic. Heat released is more in isotonic, less in isometric, right? The examples of isotonic muscle contractions are like you have walking, running, jogging. These are whatever is the tone. Tone remains the same during that particular activity, right? Walking, running, jogging, these become examples of isotonic. Isometric is if you place your hand on a wall and you push against it, that becomes an isometric. Pushing against a wall. If you push against a wall, this becomes an isometric muscle contraction. So isotonic and isometric. Tone remains the same, length decreases, length remains the same, tone increases. External work is done. Heat released is more in isotonic muscle contractions, less in isometric. Now let's discuss this heat released is more. Let's see what are the different types of heat released during a muscle contraction. What are the types of heat released? Let's see the types of heat released during a muscle contraction. Number one is what is known as the resting heat. Now, resting heat mane, this is the heat released at rest. That means the basal metabolic processes will be Jammu, see, I just said when you are doing a normal flexion, this is isotonic. But once this is flexed, and now when you stand, I cannot show you my muscles, but when you stand in front of the mirror and then you try and see your muscles and try and see the tone of the muscles, that means now you're doing an isometric contraction. So I hope, Jammu, that is clear, right? Now, what is resting heat? This is the heat which is released when a muscle is at rest, right? Now, this will be seen in both isotonic as well as isometric muscle contractions. Both isometric and isotonic will show you the resting heat. This is the heat released when a muscle is at rest. The basal metabolic processes in a muscle will release isotonic as well as in resting heat will be released in both isotonic as well as isometric, right? Next is what is known as activation activation heat. What is activation heat? That means when I stimulate a muscle, the heat released is known as the activation heat. This is initial heat. See, this is what happens when I get distracted. Just 1 second. I'll just take this heat released once again. Please just pay a little attention. I'm going to ignore what is written on the chat box. Yes, and concentrate on my work. Let's see the types of heat released. Number 1, I said resting heat. Resting heat, we said, is going to be released in both the isotonic as well as the isometric muscle contraction. The second type of heat released, this is the initial heat. Initial heat will also be released in both types. Initial heat in turn consists of what is known as activation heat and shortening heat, seen in both isotonic as well as isometric muscle contraction. And number 3 is what is known as recovery heat. Recovery heat is present in, again, both types of muscle contractions. And number 4 is the relaxation heat. Relaxation heat is only in isotonic. Right? So you have total amount of heat released will be more in isotonic, less in isometric, right? So this is your, what is this relaxation heat? Relaxation heat has been found to be equal to the shortening heat, but what is the exact reason for this relaxation heat? We do not know. But this heat is definitely more in isotonic as compared to isometric, right? This has been found to be equal to shortening heat, but what is the exact reason for it? We do not know. So as far as your MCQ goes, which type of muscle contraction will produce an increase in the heat produced, in more heat production? That will be your isotonic. And which type of heat produced is more in isotonic? And that is relaxation heat, right? So this is your different types of heat which are produced. Next, let's see. What are the sources of energy? What are the sources of energy during a muscle contraction? Now, sources of energy for muscle contraction. Number 1 are your ATP stores. Now, ATP stores in a muscle can sustain a muscle contraction only for 1 to 2 seconds. ATP stores can sustain a muscle contraction for hardly any time. This is just 1 to 2 seconds. Second source of energy is ATP from creatine phosphate. ATP from creatine phosphate, And this ATP stores and ATP from creatine phosphate are together known as the phosphagen system. They are together known as phosphagen system, and this can sustain a muscle contraction for 7 to 8 seconds. Right? ATP stores for 1 to 2 seconds, ATP from creatine phosphate for approximately 6 to 7 seconds. Together, this is known as the phosphagen system, which can sustain a muscle contraction for 7 to 8 seconds. Right? Number 3, you have what is known as glycolytic metabolism. Glycolytic metabolism can sustain a muscle contraction for 1 minute. For 1 minute. Glycolytic metabolism, though please remember, glycolytic metabolism you can generate more ATP as compared to oxidative, but glycolytic metabolism is sort of a self-limiting metabolism. And the reason for that is because now there is glycolytic metabolism, there is production of lactic acid, metabolites, which sort of limit the glycolytic metabolism can continue only for about a minute. Number 4, oxidative metabolism. Whenever muscle contractions are for more than 1 minute, you need oxidative metabolism. They will sustain a muscle contraction during the duration of muscle contraction. It will be the oxidative metabolism which becomes important, right? So if my question says number 1, what is the major source of energy for a muscle contraction? What is the major source of energy for muscle contraction? This becomes oxidative, right? Second question, what is the major source of energy in a marathon runner? Marathon runner will run for 2 hours, maybe half an hour, 1 hour, 2 hours, 2.5 hours, whatever. Again, major source of energy would be oxidative. Now look at another question. What is the major source of energy for a sprint runner? Sprint runner means short distances, but it has to be a short distance, but with a lot of power. He has to run very fast, but for a short distance of time. The major source of energy for a sprint runner becomes glycolytic metabolism. Glycolytic metabolism. Please understand, Jabalpur, that these phosphagen system can sustain a muscle contraction for only about 7 to 8 seconds. Cannot be more than that. So here it becomes glycolytic metabolism. Sprint runners will need a glycolytic metabolism to complete the source, to complete their sprint, right? So this is your different sources of energy. So 3 questions: major source of energy in a marathon runner, major source of energy in a sprint runner. So this is your sources of energy. Next, let's see. Let's see what is the mechanism of an isometric contraction. What is the mechanism of an isometric contraction? See, isotonic contraction was very easy to understand. Isotonic contraction was very easy to understand. What happens in isotonic? Okay. Now, isotonic mechanism is very easy to understand. We said there was sliding of I band into the A band. There is a sliding of I band into the A band, so there is a decrease in the length of the sarcomere. There is decrease in the length of the muscle fiber and the decrease in the length of the muscle itself, right? That is your isotonic muscle contraction. Now, isometric muscle contraction, how does this take place? Let's see this. See, when I look at an isometric muscle contraction, a muscle consists of, there is the belly of the muscle and there are the tendons on the 2 ends of the muscle. In other words, if I depict this in a diagrammatic fashion, I can say that a muscle consists of the contractile component, and on either side of the contractile component is the series elastic component. series elastic component, right? Now, what is this series elastic component? I said a muscle consists of the contractile component here in the belly of the muscle, and on either side is the series elastic component. Series means it is in series with the contractile component. What is this elastic component? See, the tendons are very rich in elastic fibers. Very rich in elastic. What are elastic fibers? Elastic fibers are like our rubber bands. You pull a rubber band, there is a tension which is generated. You leave the rubber band will recoil to its original size and shape. So similarly, in the tendons which are rich in elastic fibers, right, you've got these rubber bands, so to speak, present in the tendons, right? So we call it the series elastic component. So series elastic component is formed by the elastic fibers in the tendons. Elastic fibers in the tendons. Plus, if you remember, I did it at the beginning of this chapter, that there is another protein which is responsible for the elasticity, and that was titin. So elastic fibers in the tendons and titin. These are two which are responsible for the series elastic component. Both of these form the series elastic component, right? Now let's see what happens in an isometric muscle contraction. Now isometric muscle contraction, the length of the muscle is fixed. Length of the muscle cannot change. Length is fixed. Now when I give a stimulus to the muscle will contract. The contractile component will contract. There will be sliding of actin and myosin. The contractile component contracts. But what happens to the two series elastic element on either side? There is a stretch, right? So in this isometric muscle contraction, there is a decrease in the length of the contractile component, but there is a stretch of the series elastic component. And because of this, the total length of the muscle remains the same. Total length of the muscle remains the same. Decrease in the length of the contractile component and stretch of the series elastic component. Right? When I see, very strange na, we call it an isometric and we call it a contraction. So how do I explain? Have you understood? From one side we call it isometric and the other side we call it a contraction. Isometric means the length remains unchanged and contraction means the length decreases. So this sort of a paradox can be explained on this basis, that the total length of the muscle is the same, but there is a decrease in the length of the contractile component. Hence we call it an isometric muscle contraction. Right? So this is your mechanism of isometric muscle contraction. Next, let's go on to the next one, which is the length tension relationship. Length tension relationship. This is also known as our Frank-Starling's law. Now this law states that we can write this down and then we'll try and understand it. What is the length tension relationship? More the initial length of a muscle, more the initial length tension relationship, more the initial length, more is the tension generated, more the initial length, more is the tension generated, but up to a physiological limit. More the initial length, more is the tension generated, but up to a physiological limit. More the initial length, more is the tension generated, but up to a physiological limit, beyond which further increase in length, decreases the tension generated. This is a Frank-Starling's law. More the initial length, more the tension generated, but up to a physiological limit, beyond which further increase in length decreases the tension generated. Further increase in length decreases the tension generated. So this is your Frank-Starling's law, or the length-tension relationship. So let's look at this. Now, what is this Frank-Starling's law? Let's see how do we make this graph. Now, please understand that this length-tension relationship is applicable to isometric contractions only. Isometric contractions only. Now, what is this initial length? First, let us define initial length. Initial length is the length of the muscle fiber, or the muscle before it starts contracting. Length of the muscle fiber or the muscle before it starts contracting. This is known as the initial length, right? Like I said, this is only in isometric muscle contraction. Let's see this. How do I get this? How do we arrive at this graph? Let's say this is some muscle. This is some muscle which I have dissected, which is maintained under laboratory conditions, right? Some muscle which we have dissected, removed it, maintained it under laboratory conditions. And let's say the initial length of this muscle. And what was initial length? Initial length is the length of the muscle before it starts contracting. So let's say the initial length of this muscle is 5 centimeters. Some arbitrary length, 5 centimeters. We fix the length of this muscle. Length of the muscle cannot change. We give a stimulus to this muscle. Now this muscle will contract isometrically. It will contract isometrically. Now the tension generated in this muscle can be recorded, right? I can measure the tension generated in this muscle. The tension generated, now let's see, this gives us our first point on the graph. Let's say if the initial length was this much, the tension was this much, and here I get the first point on the graph. Okay? Now I stop the muscle stimulation, the muscle relaxes, and now I stretch the muscle, increase this initial length of the muscle, and let's make it 7 centimeters. ठीक है? फिर से मसल का लेंथ को फिक्स किया, muscle को stimulus दिया, muscle contracts isometrically, tension generated can be measured, which I measure with the help of certain instruments, right? So I get what do I see? That if the length is this much, the tension generated is this much, so I get my second point on the graph, right? To get the next point on the graph, I stretch the muscle even more. Let's make it 9 centimeters. Again, I fix the length of the muscle. Right? Give a stimulus to the muscle contracts isometrically, and the tension generated is measured. This gives me my third point on the graph. This is my third point on the graph here. Right? So similarly, I kept on doing this, increasing the initial length of the muscle and recording the tension generated. And I find more the initial length, more is the tension generated, but up to a physiological limit. What do I find? More the initial length, more is the tension generated, but up to a physiological limit beyond which more the initial length, more is the tension generated. But like I said, up to a physiological limit, beyond which a further increase in length decreases the tension generated. Decreases the tension generated. This is what is known as the Frank-Starling's law, or the length-tension relationship. Right? Now, if you see over here, there is something known as Tmax. Right? This is your Tmax. Tmax क्या है? This is the maximum tension. Tmax. Maximum tension. Now, the length at which the tension generated is maximum. Length at which the tension generated is maximum. This is called the optimum length. Optimum length. So now let's see what is the definition of optimum length. Optimum length is that initial length. What is optimum length? Optimum length is that initial length at which optimum length is that initial length at which if a muscle contracts isometrically, the tension generated is maximum. Optimum length is that initial length at which if a muscle contracts isometrically, the tension generated is maximum. Right? So this is your optimum length here. Right? This is the optimum length, which I've just pointed it out to you. This is the optimum length, which is the tension generated is maximum. Right? Optimum length is that initial length at which if a muscle contracts isometrically, the tension generated is maximum. Clear? Now let's take this a little. Take this in a little more, depth. Now this is the when I stretch the muscle. See, please understand, when I stretch the muscle, like I told you, the initial length was 5, then 7, then 9. When I increase the length of the muscle at rest, there is an increase in the tension in the muscle even at rest, before it starts contracting. And this increase in tension can be seen over here. This is what is known as the passive tension. What is passive tension? This is the tension in the muscle at rest. That means this tension is because of the stretch of the muscle. The graph that we get when we increase the length of the muscle and make it contract isometrically, this is what is known as the total tension. And what is total tension? Total tension is active tension plus the passive tension. Active tension means the tension generated when the muscle contracts, and passive tension was the tension in the muscle at rest. Means if I want to see what is the graph of active tension, I must subtract the passive tension. I must subtract the passive tension from the total tension. This will give me a graph of the active tension. Right? This is my graph of the active tension. And what is active tension? This is total minus passive. Again, what is passive tension? Passive tension is a tension which is in a muscle due to at rest, because I have increased the length of the muscle, stretched the elastic fiber, so there is a passive tension. What is total tension? Total tension is what you record during an isometric muscle contraction. And this is active plus passive. And what is active? This is total minus the passive. So in other words, if I ask you this question, all of the following are maximum at optimum length except. All of the following are maximum at optimum length except total tension, active tension, passive tension. Right? See, what was total tension? Total tension is, what is passive tension? Passive tension is when you are stretching. Remember how I got this graph? I changed the length of the muscle. Isn't it? When I stretch the muscle, there will be some degree of stretch at rest, some degree of tension at rest, which is called the passive tension. So passive tension, see, it increases like this. At optimum length, which are the types of tension which are maximum? Active is when a muscle is contracting. It includes both total as well as passive, right? This graph is your active tension. See this? The solid line is your total tension. Yes, the solid line is total tension. What is the total tension? This is the tension generated during the contraction plus the passive. And this dashed line, which I've shown over here, this becomes your active tension. Active means during the muscle contraction. So this is total minus passive. Now, which are the ones which you can see maximum? Total tension is maximum, active tension is maximum. But what about passive tension? Passive tension increases, becomes maximum much later. So in this question, my answer becomes maximum at optimum length are all except, and your answer becomes passive tension. Please see the graph, right? Jabalpur, I hope you've understood. It is not Dehradun. It is not the active tension is maximum. Ye dekho. ये active tension है. Active is total minus the passive, right? So this will be maximum here. Whereas here the total tension is also maximum. But passive is not the maximum. Also please remember, at optimum length, I have the maximum number of actin myosin cross bridges. Actin myosin cross bridges, right? Maximum number of actin myosin cross bridges. That means if the length of the muscle fiber is less than optimum length or more than optimum length, then the number of actin myosin cross bridges decreases, right? So you have maximum number of actin myosin cross bridges at this time. Also another important question, and that is, the optimum length corresponds to a sarcomere length of. Optimum length corresponds to a sarcomere length of 2 microns, 3 microns, 4 microns, 5 microns. Your answer is 2 microns. In fact, it is 2 to 2.2 microns. And what does this mean? That means if the sarcomere length is 2 to 2.2 microns and it begins to contract isometrically, the tension generated will be maximum. This is what is the concept of optimum length, right? It corresponds to a sarcomere length of 2 to 2.2 microns. Ludhiana, optimum length is length. Passive tension is tension, isn't it? Yes, they are independent of each other. Now, what is passive tension, Mumbai? Passive tension is when you are stretching a muscle at rest. When you're increasing the initial length of the muscle at rest, there will be some amount of tension generated in the muscle, na? And as you keep on increasing the length of the muscle, more will be the tension generated. At rest, the active active tension and the total tension will be seen when the muscle is, when you make the muscle contract isometrically. Yes, isometrically. Now, when I've already discussed with you what is the reason for increase in tension during an isometric muscle contraction. Because the series elastic element is now stretched because of contraction of the contractile component. Have you got my point, right? So passive means at rest. So as you keep on increasing the length of the muscle, there will be increase in the passive tension. This is how the passive tension goes. This is the graph for total and the active tensions. Total and active tensions are what you're going to see when it starts contracting. So optimum length is not going to be related to passive tension. Optimum length is related to these two, that is active and the total tensions. Right? So this is, I hope I've tried to make it, I hope you've understood what is this, Mumbai and Ludhiana. Naina, Mumbai, please understand, this tension generated is when a muscle contracts. Passive tension will be when the muscle is at rest. So passive tension is not related to the optimum length. It is when the muscle, when you're stretching the muscle at rest, isn't it? Remember we got it, we said 5 centimeters, 7 centimeters, 9 centimeters. When you're increasing the length of a muscle at rest, there will be some amount of tension generated. After all, elastic fibers are getting stretched. So that is called your passive tension. But when it contracts isometrically, now the tension generated will increase. Have you understood? So in that case, it will, that is what is known as total and active tension. And that will be related to the optimum length. Chaliye. The next, then the last point in the muscle, skeletal muscle is, last point in this will be, what do you understand by endurance training? Endurance training and strength training. Now, there are two types of, training that an athlete undergoes. One is known as endurance training and the other is known as strength training, right? Now, endurance training means, this is simply, in simple words, it is said that endurance training increases the stamina. And what does stamina increase? Stamina increase karne ka matlab kya hai? This means it increases the efficiency of its cardiovascular and the respiratory systems to tolerate exercise. That is what is endurance training. Whereas strength training is what you achieve in the gym. Because of weightlifting, because of isometric exercises, there is a hypertrophy of the muscles which you see. Right? Endurance training, this is achieved by isotonic exercises. And strength training is by isometric, primarily isometric exercises, right? What is endurance training? This increases the efficiency. This increases the efficiency of the cardiovascular and the respiratory systems to tolerate exercise. Increases the efficiency of the respiratory and the cardiovascular systems to tolerate exercise. Whereas strength training, this causes a hypertrophy of the muscles. There is a hypertrophy of the muscles which is seen, and primarily which fibers will hypertrophy? Type 2 fibers, because type 2 fibers now increase, there will be hypertrophy of the muscles, and like I said, primarily type 2, because please remember, to increase the bulk of the muscles, the larger diameter fibers have to hypertrophy. Have you got my point? So there is hypertrophy of the type 2 muscle fibers. Yes, this is what happens. I'm sure the boys can vouch for it. After a couple of months of gymming, you end up with increase in the bulk of the muscles. That is because of the hypertrophy of the type 2 fibers. Okay? So this increases the efficiency of the cardiovascular, and that is what is causing hypertrophy. In other words, this increases the oxidative capacity. whereas this increases the glycolytic capacity of the muscles. Increases the oxidative capacity, increases the glycolytic capacity. Mane, in endurance training, the load in endurance training is submaximal. Load is submaximal and the duration, load is submaximal, duration is prolonged. This is a prolonged duration. Load is submaximal and the duration is prolonged. Whereas in strength training, this is a maximal load and the duration is brief. Short duration, right? Brief duration. Load is submaximal. So that means if I ask you a simple question, that's the last question in skeletal muscle. If I want to increase the oxidative capacity of a muscle, what type of a load should I give? Maximal, submaximal? Submaximal. What type of duration should I give this individual? Brief or prolonged duration? बोलिए, ऑक्सीडेटिव कैपेसिटी को बढ़ाना है तो कौन सा ड्यूरेशन बढ़ेगा? बोलिए, prolonged, right? So this is your strength training and Jammu, cardiac muscle will contract both. In fact, all muscles show both isotonic element and an isometric element. Isovolumetric phase of the cardiac cycle is a primarily isometric contraction. Have you got it? Right? So it will be, you will see both isotonic as well as isometric. Even normal physiological, even in normal, all activities, even skeletal muscles will have a component, an isotonic component and an isometric component. Okay? So I hope that brings us to the end of this chapter, and let's take on our next chapter, which is respiratory system. Let's discuss the respiratory system. Bolo, Mumbai, kya question? Bolo, aerobic capacity is oxidative capacity, na? Will be increased by what? Tell me. Submaximal load, prolonged duration. Mumbai? Okay? Active tension equals Tmax, that's right. Mumbai, what is for aerobic capacity, you need a submaximal load and a prolonged duration, right? Now, you have asked me, is it prolonged exercise or repeated short 3-minute exercise? Even 3-minute exercises, but done repeatedly, has been found to increase the aerobic capacity. 3-minute exercise, the compliance is a little better. Have you understood? Short, but this has to be done repeatedly, in the sense that you have to do multiple short 3-minute exercises. Yes? So your best, I'm telling you, Mumbai, ruk to jao beta, 2 minutes zara sun to lo. I said this has a better compliance. So it's easier to achieve repeated 3-minute exercises rather than prolonged. So your best answer becomes the short but repeated, short but multiple 3-minute exercises. Which segment of the muscle decreases during passive stretching? Dekho, Manipal, when you are stretching a muscle, there will be no decrease in length there is no decreased length. When you are passively stretching a muscle, you're causing an increase in the length of the elastic fibers of the muscles, which is going to create some amount of tension. Okay? अब जब you have achieved a certain length of the muscle. Now when you make it contract isometrically, then the contractile component decreases and there is a further increase in the tension of the because of the elastic fibers, elastic component. Beta, I told you Mangalore, the recovery and the relaxation heat, there is what is the exact reason for the relaxation heat? We do not know. I specified that specifically. Have you understood? But it has been found that even it has been found to be equal to the shortening heat. What is the exact reason? We do not know, but it is present in isotonic muscle contractions. That is what I was trying to tell you. Have you understood? We do not know the exact. The differences: isotonic endurance, isometric strength training increases the efficiency of the cardiovascular and respiratory systems. There is a hypertrophy, oxidative capacity is increased, glycolytic, load submaximal, maximal, duration prolonged, duration brief. Chaliye. Next, let's go on to our respiratory system. Now, as far as the respiratory system, the first things first, respiratory system, let's see what are the agents which cause a bronchoconstriction and a bronchodilation, right? Parasympathetic is a bronchoconstriction, sympathetic is a bronchodilation. Use of sympathomimetic drugs. Mumbai, heart rate, BP, peripheral vascular resistance. Beta, let us do it in cardiovascular system. Isn't it? Isotonic, isometric muscle contraction. We will do it in cardio. Once let me do cardiovascular system with you. I will come back to what are the cardiovascular changes in isotonic, isometric. Okay, good. Remind me when we do CVS. Okay. So this is your bronchoconstriction, bronchodilation. This is sympathetic and parasympathetic. Okay. Now you know that sympathetic causes sympathomimetic drugs are used in the treatment of bronchial asthma. So sympathetic causes a bronchodilation. So this is adrenaline and adrenergic drugs. Acetylcholine is bronchoconstriction, right? Now agents such as leukotrienes. Leukotrienes will produce bronchoconstriction. This is an inflammatory chemical which will produce a bronchoconstriction. Leukotrienes. Next is substance P. Leukotriene, substance P. These will produce bronchoconstriction. Adenosine produces a bronchoconstriction. So these are all agents which will produce a bronchoconstriction. Bronchodilation is produced by VIP, vasoactive intestinal peptide. This will produce a bronchodilation. Please understand, VIP is a potent smooth muscle relaxant. Right? Potent smooth muscle relaxant. This is VIP, right? And this VIP is secreted by NANC nerves. This is secreted by NANC nerves. And what is NANC nerves? These are non-adrenergic, non-cholinergic nerves. Non-adrenergic, non-cholinergic nerves. So VIP produces a bronchodilation. Now there's a very simple question now. Which of the agents produces a bronchodilation? Leukotriene, substance P, adenosine, or VIP? So your answer becomes VIP. Clear? This is secreted by non-adrenergic, non-cholinergic nerves. Right? Next, let's see two important definitions. What do you understand by total or pulmonary ventilation? Total or pulmonary ventilation, this is respiratory rate into tidal volume. respiratory rate into tidal volume. This is called total or pulmonary ventilation. Then you have something known as alveolar ventilation. And alveolar ventilation is respiratory rate into tidal volume minus the dead space. This is alveolar ventilation, respiratory rate into tidal volume minus the dead space. Now let's discuss a little bit about dead space. Now as far as the dead space is concerned, right? Dead space is concerned. Now dead space is the volume of air which does not take part in the respiratory exchange process. Dead space is the volume of air which does not take part in the respiratory exchange process, right? Which does not participate in the respiratory exchange process. Now there are two types of dead space. There is a physiologic or the total dead space. Physiologic or the total dead space, this is equal to anatomic dead space plus what is known as a wasted ventilation. Anatomic dead space plus a wasted ventilation. This is your physiologic dead space. Now in normal individuals, there is no wasted ventilation. So normally what do I find? The physiologic dead space is equal to the anatomic dead space. Anatomic dead space means this is the volume of air which is present in our respiratory tracts, isn't it? Trachea, bronchi, it is not participating in the respiratory exchange process. So this is my anatomic dead space. Clear? Now physiologic or the total dead space is anatomic plus a wasted ventilation. Now what do I mean by a wasted ventilation? Let's discuss that. What is a wasted ventilation? Let's see. Now when I say wasted ventilation, see what is an ideal VQ ratio for me? What is an ideal VQ ratio? Ideal VQ ratio is a VQ ratio of 1. माने क्या? At a VQ ratio of 1. At a VQ ratio of 1, I get maximum saturation. I get a maximum saturation. So this is an ideal situation for me, right? Now what do I mean by a wasted ventilation? Now wasted ventilation mane, wasted ventilation can be wasted ventilation happens either number one, if I have over ventilated alveoli. Over ventilated alveoli, this is a wasted ventilation. Over ventilation mane, now the perfusion is normal, but ventilation has become 2 times, 3 times, 4 times normal. Mane, what will happen to the VQ ratio if ventilation has increased? Now this becomes more than 1. So this becomes a wasted ventilation, because this is not going to improve the saturation. Saturation was maximum when the VQ ratio was 1. Now by increasing the ventilation without increasing the perfusion, this is wasting the ventilation. Have you got this? So VQ ratio of more than 1 means it is a wasted ventilation. Now this wasted ventilation is possible if you have over ventilated alveoli, or if I have under perfused alveoli. And what is under perfusion? Under perfused alveoli mane? Now here the VQ ratio, now the ventilation is normal, but the perfusion is half or 1/3 of the normal. So 1 divided by half, half means 0.5. In that case also, what will happen to my VQ ratio? VQ ratio will still be more than 1 under perfusion. Ventilation is normal, let's say perfusion is half of normal. So 1 divided by 0.5 is 2, right? So this is your wasted ventilation. It could be, yes, alveolar dead space, Jabalpur, yes. So this is your underperfused. So you have either an overventilated alveoli or the underperfused alveoli. That means here in this segment of the lung, the VQ ratio is more than 1. That is called your wasted ventilation, right? Wasted ventilation. That means it is not improving the saturation. Have you understood? Saturation is maximum at a VQ ratio 1. Just to reinforce this, let's take a question. This is a diseased area of the lung where the VQ ratio is 10. Something has happened and the VQ ratio is 10. Now this will result in increase in A, anatomic dead space, or will this increase the physiologic dead space? बोलिए, VQ ratio is 10 in this particular area of the lung. It could be because of overventilation or underperfusion. So that means this will result in an increase in the physiologic dead space. Physiologic dead space, right? Now let us see what are the factors affecting dead space. How much is the normal dead space volume? Normal dead space is about 150 ml. And this is half of tidal volume, 1/3 of tidal volume, 1/4 of tidal volume, 1/5 of tidal volume. This has been asked as a question. This is 1/3 of the tidal volume. This was last year's All India PG question. Dead space volume is 1/3 of tidal volume. Now let us see what are the factors affecting dead space. Now when I say factors affecting dead space, the first is of course the age. In an infant, the dead space volume is 3.3 ml per kg body weight, but in an adult it decreases to 2 ml per kg body weight, right? Infant is 3.3 ml per kg body weight, so approximately a 3 kg child will have a dead space of about 10 ml, whereas an adult will have a 70 kg man, dead space is 140 ml, right? So in terms of per kg body weight, this is 3.3 ml, adult is 2 ml per kg body weight. That means if I want to know the dead space volume in an adult, I must multiply his body weight by 2. Whatever is his body weight multiplied by 2, I get that as the dead space volume. Second factor which affects the dead space is the size of the lungs. More the size, more the dead space. We've already discussed infant and adult dead space, right? Infant and adult dead space, right? Number 3, dead space. This also depends upon the posture. In standing position, there is an increase in the dead space. There is increase in the physiologic dead space. There is increase in the physiologic dead space, and the reason for this is standing position, there is some amount of wasted ventilation at the lung apex. Why does this wasted ventilation occur? I'll tell you a little later, right? But there is some amount of wasted ventilation at the apex in the standing position. So posture. Number 4, dead space also depends on the position of the neck. When I do a neck flexion, the dead space decreases. There will be a decrease in dead space, obviously, if I do a neck flexion. But if I do a neck extension, there is an increase in dead space. Dead space will increase, right? See, when you're doing just before intubation, you will always do a neck extension. Advantage, though the dead space increases, but the advantage is that the airway becomes straighter. So it's easier to intubate this patient, right? Number 5, if I do a tracheostomy or an intubation, tracheostomy or intubation, there is a decrease in the dead space. There is a decrease in the dead space. So tracheostomy or intubation. So these are your factors which affect the dead space. Number 1, I told you the age, infants mein kitna hai, adults mein kitna hai, size of the lungs, posture, neck, position of the neck, and tracheostomy and intubation. Clear? Now to measure the anatomic dead space, you have the Fowler's method. Fowler's method. And what is the Fowler's method? This is what is known as nitrogen analysis in the expired air after a single breath. A single breath of 100% oxygen, right? Nitrogen analysis in expired air after a single breath of 100% oxygen, right? This is called the Fowler's method for the anatomic dead space. Aurangabad, in propped up position, the dead space volume, lying down, the dead space volume is the least, right? Dead space volume will increase if you are in the sitting and in the standing up positions. Okay? But of course, in the standing will be the maximum because the effect of gravity, we'll discuss this, ventilation perfusion ratio. Child, does intubation increase dead space? Why would it increase dead space, Aligarh? Please understand, see, when you are intubating the patient, you are bypassing the dead space of that particular individual. Yes, what you're doing is, if you're connecting the tube to a ventilator, you have added the dead space of the tube, but the patient's dead space has been reduced. No, what am I trying to tell you? When you are intubating a patient and hooking him to a ventilator or to an ambu bag or something, you are adding the dead space of the tube. So the total dead space is in fact decreasing, but the dead space increasing, but the dead space of the patient would be decreasing. Yes. See, I'm talking from Respiratory Physiology by John West, so I can give you the reference for what I've just told you, what happens in intubation and tracheostomy, dead space of the individual. This is from Respiratory Physiology by John West, factors affecting dead space. Exact page number, I do not know, but you could refer to that. Okay? And that is what even anesthesia people read for teaching you respiration. But what I'm trying to tell you, please understand that. I said dead space of the individual decreases, but you are adding the dead space of the tube. Have you understood? So if you are hooking him to a ventilator, you've added the dead space of the tube. Chalo. So this is as far as your dead space is concerned. Okay, let's go on to the next one. Let's see what are the different lung volumes and capacities. Lung volume and capacities. Now, as far as lung volumes and capacities is concerned, lung volumes and capacities, this is the first lung volume that we are going to talk about is the tidal volume. And what is tidal volume? Tidal volume definition. Tidal volume is the volume of air inspired or expired. Volume of air inspired or expired during a normal, quiet, Q-U-I-T, respiration. Volume of air inspired or expired. This is our tidal volume. Volume, like what is happening right now. You're doing a quiet respiration, normal respiration. So volume of air breathed in or breathed out. It is not inspired and expired. It is inspired or expired during a normal quiet respiration is termed as the tidal volume. And tidal volume in fact is the only volume which is the same in both males and females. This is 500 ml in males and 500 ml in females. This is your tidal volume, right? This is the same in both males and females. Number 2, second is inspiratory reserve volume. Now, what is inspiratory reserve volume? Definition. Inspiratory reserve volume is the volume of air inspired over and above a tidal inspiration with maximum effort. Over and above a tidal inspiration with maximum effort. What is inspiratory reserve volume? It is the volume of air inspired over and above a tidal inspiration with maximum effort. IRV is 3300 ml in males and 1900 ml in females. This is your inspiratory reserve volume. This is the volume of air inspired over and above a tidal inspiration with maximum effort. Right. इसके बाद आ जाए expiratory reserve volume. What is expiratory reserve volume? This is the volume of air expired. Now this was tidal volume here, right? So this is the volume of air expired over and above a tidal expiration with maximum effort. Volume of air expired over and above a tidal expiration with maximum effort. This is called the expiratory reserve volume, and this is about 1000 ml in the case of males and about 700 ml in the case of females. Next is, what is residual volume? Residual volume is the volume of air which remains in the lungs. Residual volume is the volume of air which remains in the lungs at the end of a maximum expiration. Residual volume is the volume of air which remains in the lungs at the end of a maximum expiration. This is 1200 ml in the case of males and 1100 ml in the case of females. Now comes, these are your different volumes. Now let's discuss capacities. So the first capacity is inspiratory capacity. Inspiratory capacity is IRV plus tidal volume. IRV plus tidal volume is the inspiratory capacity. Mane, this now becomes the inspiratory capacity. Inspiratory capacity is IRV plus the tidal volume. Next is, what is expiratory capacity? Expiratory capacity. Expiratory capacity is ERV plus the tidal volume. ERV plus the tidal volume. Expiratory capacity is ERV plus the tidal volume, right? Then you have what is known as the functional residual capacity. Definition of functional residual capacity: it is the volume of air. It is the volume of air which is present in the lungs, volume of air present in the lungs at the end of a normal expiration, volume of air present in the lungs at the end of a normal expiration. This is functional residual capacity. And what is functional residual capacity? Now, functional residual capacity becomes ERV plus the residual volume. This is FRC, functional residual capacity, ERV plus the residual volume. So let's discuss this here. This is ERV plus the residual volume. This is the volume of air in the lungs. What? I don't know. Functional residual capacity is ERV plus the residual volume. That means this is the volume of air present in the lungs. See what is happening right now. You are doing a tidal inspiration, tidal expiration. Okay? So, जब tidal expiration किया, now the volume of air which is remaining in your lungs at the end of a normal expiration is equal to your FRC. FRC matlab it is ERV 1200 ml in males and ERV 1000 ml in males and residual volume 1200 ml. So that means even at the end of normal expiration, you've got 2200 ml of air present in your lungs. This is the significance of FRC, right? That we are doing a tidal inspiration expiration over a partially inflated lung, right? At the end of normal expiration, your lungs are still partially inflated. In fact, the volume is approximately 2200 ml. In the case of males at least. Okay? So this is your functional residual capacity. Next comes what is vital capacity. Now for vital capacity, how do I record the vital capacity is, I will ask my patient to take a maximum inspiration. I ask him to take a maximum inspiration followed by a maximum expiration. So what is the vital capacity? This becomes now the vital capacity, and vital capacity definition will now be, now this is the vital capacity. That means what is the definition of vital capacity? This is the volume of air expired forcefully, after a forceful inspiration. This is my forced vital capacity. Volume of air expired forcefully after a forceful inspiration is called the forced vital capacity or the vital capacity. Right? And vital capacity becomes IRV plus tidal volume plus ERV. This is my vital capacity. Right? Volume of air expired forcefully after a forceful inspiration. If I add up all the lung volumes, I get total lung capacity, which is 6000 ml in the case of males and 4200 ml in the case of females. Right? This is my total lung capacity. Right? In other words, let's try and answer this question. This question says, vital capacity is equal to, is it inspiratory capacity plus expiratory, inspiratory plus expiratory plus tidal volume, inspiratory plus expiratory minus tidal volume, inspiratory minus expiratory plus tidal volume? Bolo. A, B, C or D. What is vital capacity? Yes, this is inspiratory plus expiratory minus the tidal volume, isn't it? Because inspiratory capacity includes one tidal volume, expiratory capacity includes one tidal volume. And we know that vital capacity is ERV plus tidal volume plus IRV. So you need to subtract one tidal volume. So this is your forced vital capacity or vital capacity, right? Now comes an important point. That was, which are the lung volumes and capacities I cannot record, which cannot be recorded by a routine spirometry, cannot be recorded by routine spirometry. cannot be recorded by routine spirometry, right? This is residual volume, FRC, and total lung capacity. All three cannot be recorded by routine spirometry. For FRC, I have a technique which is known as helium dilution technique. Helium dilution technique. You know that FRC is ERV plus the residual volume. ERV you can get by routine spirometry. So therefore, I can calculate RV. RV is equal to FRC minus the ERV. RV becomes FRC minus the ERV. Please remember, there is no technique. So if I have a question which says helium dilution technique is meant for measurement of residual volume and FRC, then your better answer becomes FRC, right? Some more techniques for the measurement of FRCs. Number one, I told you helium dilution technique. Then you have a nitrogen washout method. Please understand, this is different from the Fowler's method. Fowler's method was nitrogen analysis in expired air after a single breath of 100% oxygen. This is the nitrogen washout method for helium dilution technique, for FRC, sorry. And you also have what is known as a body plethysmography. These can be used to measure the FRC. Right? So this is your techniques to measure FRC, right? One small important point, and that is we've discussed the volumes in males and females. Now supposing gender has not been specified and you are asked what are the lung volumes, then look at, I'll give you some volumes which are sort of not gender specific, and the not gender specific method is volumes is tidal volume 500 to 750 ml, IRV approximately 2 liters, ERV approximately 1 liter, right? And total lung residual volume approximately 1.2 liters, and total lung capacity approximately 5 liters. So these are more sort of realistic values and more non-gender specific, right? 2 liters of IRV, 1 liter of ERV, and 1.2 liters of residual volume, right? So this is as far as your different volumes, lung volumes and capacities is concerned. Next, let's see. Mysore, you've asked me which is the best method. I think you're asking for FRC. Yes? See, FRC, if it is FRC you're asking me, then the body plethysmography. What is the advantage of body plethysmography is that it not only records the FRC, it also records, sometimes, you have air which is trapped in, the bully, for instance. There is air which is trapped. It also helps you to record that, right? So, so based on that, yes, it would be the body plethysmography, which will be more effective in recording not just the FRC, but also the air trapped in the bully. That is your, which cannot be measured by helium dilution technique or by the nitrogen washout method. Right. Let's go on to the next part, and that is let's have a look at ventilation. Now, when I talk of ventilation, there are some important pressures that we need to discuss. And the first pressure that I'm going to discuss with you is what is known as the intrapleural pressure or the pleural pressure or the intrathoracic pressure. Intrapleural or the pleural pressure or the intrathoracic pressure. Now what is this pressure? This is the pressure between the two layers of pleura. This is the pressure between the two layers of pleura. You've got a parietal pleura and you've got a visceral pleura. And the pressure between, please remember, between the visceral and the parietal pleura, there is a potential space. It is not an actual space, it is a potential space, right? And the pressure in this space, this is negative with respect to atmosphere. This is negative with respect to atmosphere. So firstly, let us try and understand, why is it negative with respect to atmosphere? Why is it negative? What do you mean by negative with respect to atmosphere? And the reason why, what do you mean by negative with respect to atmosphere is, see, if I say this pressure is minus 2 millimeters of mercury, and the atmospheric pressure is 760, let's say 760 millimeters of mercury, then this pressure becomes 760 minus 2, that means 758 millimeters of mercury. So it's negative with respect to atmosphere. So if I say the outside pressure is 500 millimeters of mercury, this pressure becomes 498. This is your negative with respect to atmosphere. Now the question arises, why is this pressure negative with respect to atmosphere? Now please remember, the lungs are elastic structures and the chest wall is also an elastic structure. An elastic structure, I have already described to you. What are elastic structures? Are your rubber bands. You stretch a rubber band, there is a tension which is generated, you leave it, it recoils to original size and shape. Original size and shape, right? That is called, so here the elastic fibers of the lung are so arranged that their recoil of the elastic fibers of the lung is towards the inside, and recoil of the chest wall elastic fibers is towards the outside, right? These lung recoil and chest wall recoil are in opposite directions. These are two opposing forces. And these two opposing forces now create a negative pleural pressure, right? So this is negative. The reason for the negative pressure in the pleural space, the first one is lung recoil and chest wall recoil. These are two opposing forces. They are pulling the pleural space in the opposite direction. And this creates a negative pressure in the pleural space. See, the closest I can come to describing this pressure is, if you remember your physiology, if your hematology experiments in the first year, and I just hope that you've done them, yes? Now, we would do DLC, right? And you would take glass slides, right? And so many times it would happen. Remember those microscopic glass slides for DLC and all that? You would do that, right? Now, so many times you would reach the lab and your lab technician has washed the glass slides. He has kept them in a tray. You pick up two slides. There is a thin layer of fluid between the two, and the two glass slides are stuck to each other. Yes? Now, if you remember, it was very easy to move the slides like this. This movement was very easy. Yes, and that is exactly what's happening to our visceral pleura, parietal pleura. There is a thin layer of pleural fluid and very easy to move the pleura like this. That is what happens during respiration. It minimizes friction, minimum friction. But if you remember, if we try to separate the two glass slides, that is exactly what's happening here. The lung recoil is pulling the visceral pleura towards the inside. The chest wall is pulling the parietal pleura towards the outside. They're both being pulled in opposite directions, and that's what creates a negative pressure in the pleural space. So lung recoil and chest wall recoil, these are two opposing forces. Okay? Second reason for this negative pressure in the pleural space is there is a continuous drainage. Continuous drainage of the pleural fluid into the lymphatics. There is a continuous drainage of the pleural fluid into the lymphatics. And this also creates a negative pressure in the pleural space. But please remember, the main reason is this one. Lung recoil and chest wall recoil are two opposing forces. This has been asked as an All India PG question. See, look at a clinical application. Clinical application is fairly easy to understand. See what's happened? See what happens in emphysema. Emphysema, mein, what is the pathology? There is a destruction of the lung elastic fibers. So the lung recoil now reduces. There is destruction of the lung elastic fibers and the lung recoil has now reduced. So what happens now? The chest wall recoil is relatively more. Right? The chest wall recoil is relatively more. So what happens to the shape of the chest in emphysema? What happens to the shape of the chest? It becomes a barrel-shaped chest. Chest acquires an expanded position. Yes, Meerut. It acquires an expanded position and becomes barrel-shaped. Have you understood this point? Right? See what happens in a pleural effusion. Yes? Now what happens now? In pleural effusion, there is now fluid in the pleural space. There is fluid, or let's say pneumothorax for that matter. There is fluid present over here now. So what happens is this lung recoil and chest wall recoil. Now between the two there is water, or there is fluid, or there is air. Pleural effusion or pneumothorax. So what happens is link between the two forces is broken. What is commonly seen in pleural effusion? Lung collapse. Commonly seen in pneumothorax, lobar collapse or maybe a lung collapse. Why? Because now the lung recoil is no longer being opposed by the chest wall recoil, क्योंकि उन दोनों के बीच में क्या आ गया है? बोलिए, fluid या air. Have you got it? So this is your intrapleural pressure or the pleural pressure or the intrathoracic pressure. The synonyms, these are important to know the synonyms. This is the pressure in the two layers of the pleura. Okay? Chalo, I'll take a lunch break. I'll give you 40 minutes for lunch. Yes, 1:20. We'll come back at 2 o'clock and then we'll continue. Right? Okay. Continue from where we left. Let's discuss the next one. We were discussing the intrapleural pressure. Yeah, good afternoon, Karam Sad. We were discussing the intrapleural pressure, and I said that there's a potential space between the visceral and the parietal pleura, and the pressure in this space is negative with respect to atmosphere, and negative with respect to atmosphere, and the reason for this is lung recoil and chest wall recoil. These are two opposing forces, and number 2 was the continuous drainage of pleural fluid into the lymphatics. Okay, you want to be prepared with your doubts before the classes. Okay, Gorakhpur, today we will, let's say till about 6 to approximately 5 or 6 ke beech mein, we'll be able to finish respiratory system. Then we'll start off with kidney. And on next Saturday, I will take up CVS and GI tract on Saturday, and CNS we'll take it on next Sunday. So that's how I'm planning your classes. Okay. So this is how it goes. So you've got respiration and kidney today. Respiration and kidney. Tomorrow would be next Saturday would be GI tract, CVS and GI tract, and the Sunday would be CNS, right? So this is your lung recoil and chest wall recoil are two opposing forces and a continuous drainage of pleural fluid into the lymphatics, right? So now let's see what is going to be the effect of hemat. Hemat is done in physiology, doesn't do hemat. Hemat is covered in pathology. So we do not do him at in physiology. Endocrinology Bilaspur is again, you have one whole day of medicine. Medicine covers endo for you. You have a complete class in endo which they cover the physiology aspect. So if you still feel the need that there is some need for endo, I'll probably take a little short endo class as well on the last day. Okay, let's see how it goes. But I think the physiology aspects are covered in your endo class in medicine, right? So let's see the next one, and that is let's see what happens to the intrapleural pressure. Let's see intrapleural pressure. Now, intrapleural pressure, Alikar, I'll try and recap the lung volumes in the break for you, right? So let's just continue from where we left, right? Now, the intrapleural pressure, let's see what happens to the intrapleural pressure or the pleural pressure or the intrathoracic pressure with respiration, right? This is the midline here. This is inspiration here, and this is expiration, right? Inspiration and expiration, right? Now let us see what happens to this pleural pressure with inspiration and expiration. This is normal tidal volume, right? This is normal tidal volume, and this tidal volume is 500 ml. Now the pleural pressure with respiration, pleural pressure at the start of inspiration, this is minus 2.5 millimeters of mercury. This is with respect to atmosphere, minus 2.5 millimeters of mercury with respect to atmosphere. This is your pleural pressure with respiration, minus 2.5 millimeters of mercury with respect to atmosphere. But of course, in centimeters of water, this has been given as minus 4 to minus 5 centimeters of water at the start of inspiration, and then but your better answer here is minus 5 centimeters of water. In millimeters of mercury, it is minus 2.5. Why is it minus 2.5? I've already told you this is because of lung recoil and chest wall recoil being in two opposing forces, right? Now let us see what happens to this pressure with inspiration. See, which is the main muscle of inspiration is diaphragm, right? So what happens during inspiration is the diaphragm contracts, moves downwards. When the diaphragm contracts and moves downwards, now the diaphragmatic pleura also moves downwards. So what happens to the volume of the pleural space? The volume of the pleural space increases and the intrapleural pressure decreases, right? Intrapleural pressure decreases. Now from minus 2.5 millimeters of mercury at the beginning of inspiration, this pressure now decreases and becomes minus 6 millimeters of mercury at the end of inspiration. Minus 2.5 at the start of inspiration to minus 6 at the end of inspiration. Okay? Now, during expiration, the diaphragm moves upwards. When the diaphragm moves upwards, so does the diaphragmatic pleura. So what happens to the volume of the pleural space? Volume of the pleural space decreases and the pressure increases, right? So this is your, now the pleural pressure will increase and come back to minus 2.5 millimeters of mercury at the end of expiration, right? See this? Inspiration, this is your tidal inspiration 500 ml, expiration 500 ml. So I'm trying to tell you what happens to the pleural pressure with inspiration. That's all that I've covered, Mangalore. So at the start of inspiration, this pressure is minus 2.5. Why is it minus 2.5? I've already described it to you because of the two opposing forces, lung recoil and the chest wall recoil. Now this pressure, minus 2.5 millimeters, this can be written in terms of centimeters of water. This is minus 4 to minus 5 centimeters of water. But like I said, preferred answer would be minus 5 centimeters of water. So it is minus 2.5 at the start of inspiration. Now during inspiration, the diaphragm, which is the main muscle of inspiration, moves downwards. So does the diaphragmatic pleura. So the volume of the pleural space increases and the pressure decreases and the pressure becomes minus 6 millimeters of mercury. Minus 6 millimeters of mercury. During expiration, the diaphragm contracts and moves upwards. The volume of the pleural space decreases and the pressure increases to minus 2.5. This is, now supposing I am using my accessory muscles of inspiration, the scaleni, the serratus anterior, and the abdominal muscles for inspiration. So now what will happen to the volume of the pleural space? It increases even more. So pressure will decrease even more, right? So instead of -6, it may be -10, -15, right up to -30 millimeters of mercury. The pressure will become more negative. Stronger the inspiratory effort, the pressure will become more negative. That is what I'm trying to tell you. And the maximum that it can go down to is -30 millimeters of mercury with respect to atmosphere, right? So this is your pleural pressure, pleural variation of the pleural pressure with respiration. Pleural pressure with respiration, right? -2.5 at the start of inspiration, -6 at the end of inspiration, and this becomes -10, 15, depending upon how strong is the inspiratory effort, right? Now, does this pressure ever become positive? Does this pressure ever become positive? This pressure can become positive during a Valsalva. Okay. So this pressure, like I said, it becomes positive during a Valsalva. And what is Valsalva? Valsalva is, now this becomes positive during a Valsalva. Why Valsalva? Valsalva, what is Valsalva? Valsalva is a forced expiration against a closed glottis. Forced expiration against a closed glottis, right? This is, Mahinder. Okay, so welcome back all centers. Let's see. So I was discussing what happens to the pleural pressure with respiration, and we discussed that the pleural pressure becomes more negative with inspiration and comes back to -2.5 at the end of expiration. So this is the variation, and I said if I take a strong inspiratory effort, this pressure will become even more negative. That is what we had discussed. Then we discussed, is there any physiological condition where this pressure becomes positive? Does this pressure ever become positive? This pressure becomes positive during a Valsalva. And what is Valsalva? Valsalva is a forced expiration against a closed glottis. Forced expiration against a closed glottis. Forced expiration against a closed glottis. Yes, this is a Valsalva. Valsalva is forced expiration against a closed glottis. That means, see, when do we do a Valsalva? Whenever you are straining, for example, to pass stools, you're doing a Valsalva, right? Or if you have lifted a very heavy weight, you are doing a Valsalva. Yes, or a lady is in labor. She is doing a Valsalva, a forced expiration against a closed glottis. So this is the time when this pressure becomes positive. This pressure becomes positive. Right? This pressure becomes positive during a Valsalva, forced expiration against a closed glottis. Right? Now, like I said, this pressure becomes more negative. If it's a stronger inspiratory effort, it will become more negative. Now, next, let's see another pressure which is important, and that is your intrapulmonary pressure, or let's see, this is the next pressure which I need to talk about, and that is your intrapulmonary pressure. Intrapulmonary pressure, or the airway pressure, or the alveolar pressure. Intrapulmonary pressure, or the airway pressure, or the alveolar pressure. This is the next one that we have to talk about. Now, here is your inspiration, and this is your expiration. Inspiration, expiration, right? This is 500 ml is inspired, 500 ml is expired, right? This is your tidal volume, 500 ml, right? So now let's see what is the variation of the airway pressure. Now please understand, which are the airways am I talking about? Which are the airways that we are referring to over here? The airways being spoken about over here are not the trachea and the bronchi, because please understand, the trachea and the bronchi, their diameter does not change with respiration, or at least it does not significantly change with respiration, because they have got cartilage rings, right? So which are the airways which I'm talking about? This is, if this is a cross section of the lung, a diagrammatic representation. So the airways which are present in the lung parenchyma, the airways which are present in the lung parenchyma, these are the ones which I'm speaking about. Now, this is your airway or the alveolar pressure. Now, please see this. The airway or the alveolar pressure at the start of inspiration, this is 0 millimeters of mercury with respect to atmosphere. 0 millimeters of mercury mane with respect to atmosphere mane. This means it is equal to the atmospheric pressure. 0 millimeters of mercury with respect to atmosphere is equal to the atmospheric pressure. Now let's see what happens during inspiration. Now during inspiration, we have already discussed that the pleural pressure becomes more negative, and when the pleural pressure becomes more negative, this has a sort of an expansile force. It creates an expansile force on the lungs, right? And when there is an expansile force created on the lungs, the airway and the alveolar diameter increases. The airway and the alveolar diameter increases, and when the airway alveolar diameter increases, pressure decreases. Pressure becomes slightly negative with respect to atmosphere. It becomes minus 1 millimeters of mercury with respect to atmosphere. Why has this pressure become slightly negative with respect to atmosphere? Because during inspiration, the pleural pressure has become more negative. So it creates a sort of an expansile force on the lungs, increasing the airway and the alveolar diameter, and the pressure increases to minus 1 millimeters of mercury. Now you know this, that air will always go from an area of high pressure to an area of low pressure. When the pressure becomes minus 1 millimeters of mercury, now this pressure is lower than the atmospheric pressure. Lower than the atmospheric pressure, right? So this is now minus 1 millimeter of mercury. So now air comes from the atmosphere into the lungs. From the atmosphere into the lungs, and as air moves into the lungs, into the airways and the alveoli, the pressure starts increasing, and now reaches 0. 0 matlab it has become equal to atmospheric pressure. The moment it becomes equal to atmospheric pressure, the airflow stops. Okay? This is your end of inspiration. So during inspiration, this pressure became minus 1, and at the end of inspiration, this pressure became 0. This pressure became 0. So -1 during inspiration and then becomes 0 at the end of expiration. End of inspiration. Now during expiration, let's see what happens during expiration. Now expiration, the diaphragm moves upwards. When the diaphragm moves upwards, there is a compressive force which is generated on the lungs. There is a compressive force which is generated. It tends to decrease the lung volume. So what happens now? When the volume decreases, the airway and the alveolar pressure increases. Now it becomes +1 millimeters of mercury, slightly positive with respect to atmosphere. When it becomes slightly positive with respect to atmosphere, now air will go from an area of high pressure to an area of low pressure. Now it goes from inside the lungs to outside the lungs. And as it moves outside the lungs, the airway and the alveolar pressure now starts falling, and it now reaches a pressure of 0 at the end of expiration. So let's see this again. The pressure is equal to the atmosphere. The pressure in the airways and the alveoli is equal to the atmospheric pressure at the start of inspiration, which is 0. During inspiration, the airway and alveolar diameter, because of the surrounding pressure becoming more negative, their volume increases, pressure decreases to -1, slightly negative with respect to atmosphere. Air comes from outside to inside the lungs, and the airway pressure now starts increasing, becomes maybe 0.75, -0.75, -0.5, -0.25, and reaches 0. And the moment it reaches 0, airflow will stop. During expiration, a compressive force on the airways and the alveoli, and the pressure increases to +1 during expiration and again comes back to 0 at the end of expiration. This is the variation of the pleural pressure with respiration. And let's see what is the variation of this was the variation of the airway and the alveolar pressure with respiration. Let's see the variation of pleural pressure with respiration, which I've already done with you. -2.5 at the start of inspiration, and now this becomes -6 at the end of inspiration, and then it comes back to -2.5 at the end of expiration. Yes? Okay, Bhatinda and Srinagar, please just see again. The pleural pressure at the start of inspiration is -2.5. End of inspiration becomes more negative, that is -6, and back to -2.5 at the end of expiration. Clear? Now, like I said, Ludhiana, just be careful. I said the pleural pressure becomes more negative with inspiration, and this pressure can decrease up to -30 millimeters of mercury. -30 millimeters of mercury. Now, the airway or the alveolar pressure at the start of inspiration is 0. During inspiration, -1. Then as air moves from the atmosphere into the lungs, this pressure increases and comes to 0 at the end of inspiration. During expiration, the airway or the alveolar pressure becomes +1. And now air moves from the airways from inside to outside the lung, and the airway pressure starts decreasing, and this pressure now the airway pressure starts decreasing and now reaches 0. So 0 at the start of inspiration, -1 during inspiration, 0 at the end of inspiration. +1 during expiration and 0 at the end of expiration. This is the variation of the pulmonary pressure, intrapulmonary pressure, or the airway pressure, or the alveolar pressure with respiration. Now, what we are interested in, see, these are two pressures which we have dealt with separately. Now, what we are interested in is the net effect of these two pressures. After all, they are not going to act in isolation. We are interested in the net pressures. These two pressures are not going to act in isolation, they are acting together. So we are interested in the net pressure. And what is the net pressure? Net pressure is the transpulmonary pressure, also known as the transmural pressure, or the distending pressure. Transpulmonary or the transmural pressure or the distending pressure, right? And this is intrapulmonary minus the intrapleural. Intrapulmonary minus the intrapleural. So let's take the first question. First question is, what is the distending pressure? What is the distending pressure at the start of inspiration? What is the distending pressure at the start of inspiration? Start of inspiration, is it minus 2.5? Is it plus 2.5? Is it minus 6 or plus 6? Yes. What is the transpulmonary pressure at the start of inspiration? So let's see this now. Start of inspiration, the intrapulmonary pressure is 0. The intrapleural pressure is minus 2.5. So it becomes 0 minus 2.5, which is a plus 2.5. Plus 2.5. The answer now here becomes a plus 2.5. Similarly, what is the transpulmonary pressure at the end of inspiration? End of inspiration, let's see again. What happens at the end of inspiration? Now, end of inspiration, intrapulmonary is 0. Intrapleural is minus 6. So it becomes 0 minus 6, which is equal to plus 6. Yes. So now the transpulmonary or the distending pressure is plus 6 at the end of inspiration. Manikya, see, basically, when I said at the start of inspiration, there's a distending pressure of plus 2.5. This you must be able. This you should be able to appreciate, because remember I told you that the tidal inspiration expiration is on a partially inflated lung. It is not on a totally collapsed lung. Remember when I was discussing FRC with you, I said that the tidal inspiration expiration is on a partially inflated lung. So now you know that this lung is partially inflated with a distending pressure of plus 2.5 millimeters of mercury. During inspiration, this distending pressure increases to plus 6 millimeters of mercury. Manika, the stronger the inspiratory effort, more negative the pleural pressure, more positive will be the distending pressure. If the pleural pressure is minus 10 millimeters of mercury, then the distending pressure or the transpulmonary pressure becomes 0 minus 10, so which is plus 10. Supposing I've taken a yet more strong inspiratory effort, the pleural pressure is minus 20 millimeters of mercury, distending pressure will become 0 minus 20, which is plus 20. Have you got my point? More negative the intrapleural pressure, more positive will be the transpulmonary or the distending pressure. And how do I calculate transpulmonary or distending pressure? It is the intrapulmonary minus the intrapleural. Intrapulmonary minus the intrapleural. So this is as far as your transpulmonary pressure is concerned. Why minus 13, Karamsad? I told you it was minus 30, 30 millimeters of mercury. Yeah. Okay. Next, let's look at what is the ventilation perfusion gradient in the standing posture. Ventilation perfusion gradient in the erect or the standing posture. What is the ventilation perfusion gradient in the standing posture? Now see this, to remember this, we have to understand that when one is in a standing position, ventilation and perfusion is not uniform throughout the lungs. There is a gradient which has occurred, right? And how do I remember this? Small thing, this is BAD, base to apex, there is a decrease in intrapleural pressure. There is a base to apex, there is a decrease in ventilation. Base to apex, there is a decrease in perfusion. But the VQ ratio increases from base to apex, right? You need to remember this. Base to apex, there is a decrease in intrapleural pressure, there is a decrease in ventilation, there is a decrease in perfusion, but VQ ratio increases from base to apex, right? Dehradun, a positive pressure ventilation. Yes, that means you are exerting a positive pressure from outside. Have you understood? Right? You are increasing the positive pressure from outside. You are pushing the air into the lungs with the help of a positive pressure. Have you understood? So this is what is that. So we are increasing here. Please understand, you are increasing the airway pressure because your pressure, you are exerting, you're pushing air into the lungs under positive pressure. Okay? Now this is your base to apex, which are decrease in intrapleural pressure, ventilation, perfusion, but VQ ratio increases from base to apex. Now what does this mean? This means, of course, that base per intrapleural pressure is less, apex per zyada. Base per intrapleural pressure is more, apex per less. Base per ventilation is more, less at apex. Base per perfusion is more, less at apex. But VQ ratio, less at base, more at apex. Firstly, can this occur? Is this possible that ventilation is decreasing from base to apex, perfusion is decreasing from base to apex, but VQ ratio is increasing from base to apex? And this is possible if you look at the relative change in ventilation and perfusion. Absolutely right, Vishakhapatnam. If the decrease in perfusion is much more than the decrease in ventilation, this sort of a change in ratio is a possibility. Yes, and what do we know? What do we see? Look at this. This is your lungs here. The VQ ratio, let's see. I'm just going to take some arbitrary example. Right? Some arbitrary values. These are not the true values. We find that the ventilation is 2.5 liters per minute at the base. Perfusion is 5 liters per minute at the base. So my VQ ratio becomes 0.5. And in fact, this is true. VQ ratio at the base is approximately 0.5. Right? This value is correct, but of course these two values are just arbitrary values which I've given you. Now let's see what happens at the level of the third rib. Now this is at the level of the third rib. Now at the level of the third rib, both ventilation and perfusion will decrease. Now ventilation at the third rib becomes 2 liters per minute. Perfusion is also 2 liters per minute. So my VQ ratio here is 1. Ventilation has decreased from 2.5 to 2. Perfusion has decreased from 5 to 2. There is a larger decrease in perfusion, so there is an increase in the VQ ratio. At the apex, now the ventilation is 0.5 liters per minute. Perfusion is ventilation is 1.5 liters per minute and perfusion is 0.5 liters per minute. So what will happen to my VQ ratio here now? Now my VQ ratio has become 3. Right? And this is exactly, see what has happened to the ventilation has decreased from 2.5 to 1.5, but the perfusion has decreased much more. So the VQ ratio has increased from base to apex. VQ ratio at the base is 0.5, at the third rib it is 1, and at the apex it is 3. In fact, the average VQ ratio for the whole lung, this is 0.8. 0.8. Average VQ ratio for the whole lung is 0.8. Remember, I told you a little while back that in the standing position, there is some amount of wasted ventilation which happens at the apex. You remember that? We discussed that there is a there in the standing position, the physiologic dead space increases because, like I told you, in the standing position there is increase in the there is some amount of wasted ventilation which happens at the apex, and I've just told you how there is some amount of wasted ventilation happening at the apex, right? So we said that the VQ ratio increases from base to apex. So just to answer this question now, arterial oxygen tension will be maximum at base or apex. Arterial oxygen tension is maximum at base or apex. Arterial oxygen tension. Please understand, arterial oxygen tension will depend on the VQ ratio. Wherever VQ ratio is maximum, arterial oxygen tension will be maximum. Please understand, this is your arterial oxygen. What is the level of dissolved O2 in the blood? This depends on the VQ ratio. Wherever VQ ratio is maximum, you'll get the maximum arterial oxygen tension. So maximum arterial oxygen tension will be at the apex, right? And that is why tuberculosis is common at apex. Tuberculosis is common at apex. What has happened at the apex? Tuberculosis is common at the apex. Why? Because what has happened at the apex? See, Mycobacterium is an air-loving organism. Hematogenous spread of tuberculosis will occur to those areas where the arterial oxygen tension is maximum, and arterial oxygen tension is maximum at the apex. So tuberculosis is common at the apex. Tuberculosis is common at the apex. This is arterial oxygen tension maximum at apex, right? Please understand, what determines the arterial oxygen tension is the VQ ratio. Wherever the VQ ratio is maximum, you'll get a maximum arterial oxygen tension. Chaliye, let's have a look at what do you understand by dynamic compression of airways. Dynamic compression of airways. Now, what is dynamic compression of airways? See, whenever I do, please understand again, which are the airways which I'm going to talk about is the intrapulmonary airways. Intrapulmonary airways. So normally, now what is dynamic compression of airways? So whenever I do a forceful expiration, please understand, it is not a tidal expiration. You will not see compression of airways in tidal expiration. It is in forceful expiration. Now, in forceful expiration, there is a tendency for the airways and the alveoli to collapse. During expiration, there is a tendency for the airways and the alveoli to collapse. Forceful expiration, please understand, not in tidal expiration. Now, the alveolar collapse is not possible because it has the presence of surfactant. But the airways will now show the airways will tend to collapse with forceful expiration. And at the end of forceful expiration, the airway collapse is complete. Yes, This tendency for the airways to collapse during expiration is known as dynamic compression of the airways. Please understand, where do I see this dynamic compression? This is seen during forceful expiration. During forceful expiration, there is a tendency of the airways to collapse. This is known as dynamic compression of the airways. And at the end of forceful, when I do a maximum expiration, the airway collapse is complete. And when the airway collapse is complete, air is trapped in the alveoli, and this air which is trapped in the alveoli is known as the residual volume. Have you got my point? See, during forceful expiration, when you're doing a forceful expiration, there is a tendency for the airways to collapse. Alveolar collapse is not possible because they have surfactant which prevents a complete collapse of the alveoli. See, isn't it? Clinical application, early bronchial asthma, the wheeze that we hear is the expiratory wheeze. Expiratory wheeze is heard before the inspiration, isn't it? As the disease advances, you also hear inspiratory wheeze, but first you hear the expiratory wheeze. Early stages of disease, expiratory. Why expiratory wheeze? Wheeze because the airways are inflamed, plus in expiration there is, as it is, a narrowing of the airways. So expiratory wheeze is commonly heard in early stages of the disease, right? Now, like I said, at the end of a forceful expiration, this is at the end of maximum expiration, the airway collapse is complete. When airway collapse is complete, air gets trapped in the alveoli. And that air which is trapped in the alveoli is known as the residual volume. This entire thing, this collapse of the airways during expiration is known as dynamic compression during forceful expiration, and residual volume. So that means if I ask you a question, what is this residual volume due to? This residual volume is due to, and your answer becomes dynamic compression of airways. Dynamic compression of airways. Airways, now, why would they collapse? Why would they collapse during expiration? You're now compressing the lungs, now. When you're doing a forceful expiration, you're compressing the lungs. So airway diameter will tend to decrease, alveolar diameter will tend to decrease. Expiration हो रहा है, so we're compressing the lungs. Yes. So airway will tend to collapse during expiration. There is a tendency for the airways to collapse during expiration. Yes. Now this is tendency of the airways as well as the alveoli. There's a compressive force being generated. But alveolar collapse is not possible because, like I said, they've got the presence of surfactant. Right? So this is your forceful expiration, dynamic compression of the airways. Next, let's see what do we understand by lung compliance. What is lung compliance? Now, lung compliance refers to compliance is the distensibility of the lungs. Compliance has been defined as the distensibility of the lungs. And the definition of compliance is change in volume per unit change in transpulmonary pressure. Change in volume per unit change in Karam Sir, what is the definition of residual volume? Residual volume is the lung volume at the end of maximum expiration. ठीक है? See, maximum expiration जब आप करते हैं, we should expect that the alveolar volume should have been 0. Yes? But you have certain amount of, at the end of maximum expiration, you have air in the alveoli. And this air in the alveoli is residual volume. What is this residual volume due to? This is because the collapse of the airways leading to the alveoli are collapsed. When you do an airway leading to the alveoli is collapsed, there will be air trapped in the alveoli, which is known as residual volume. So residual volume is due to dynamic compression of the airways. ठीक है? चलो. So compliance is delta V by delta P. This is the change in volume per unit change in transpulmonary pressure. Change in volume per unit change in transpulmonary pressure. This is lung compliance. Yes, in fact, elastance. Elastance is the reciprocal of compliance. Elastance is the reciprocal of compliance. This is delta P by delta V. Delta P by delta V. Now it is change in pressure per unit change in volume. That is the definition of elastance. Compliance were change in volume per unit change in transpulmonary pressure. That was compliance, and this is elastance, right? Now, how much is the value, normal value of lung compliance? Lung compliance normal value. This is 0.2 liters per centimeter of water pressure. 0.2 liters, that is 200 ml per centimeter of water pressure. This is the normal value of lung compliance. The compliance of the lung plus chest wall. Lung plus chest wall. This is reduced. This is 0.1 liters per centimeter of water pressure. Of only the lung or only the chest wall is 0.2 liters per centimeter of water pressure, but lung and chest wall together is 0.1 liter per centimeter. That means, what does this mean? That the change in volume, when you change the transpulmonary pressure by 1 centimeter of water, the change in the lung compliance will be 200, the change in lung volume will be 200 ml. This is what it means, right? Now, this is as far as your definition of compliance is concerned. Now, Ludhiana says, for practical purposes, VQ ratio, 1 or 0.8, 0.8. Na? Average VQ ratio for the whole lung is 0.8. Merit says dynamic compression occurs at the level of matlab? Merit, level of matlab? Upper zone, mid zone, what is that? What level of? Right, so this is your compliance of the lung. Remember, I just told you dynamic compression is for all the intrapulmonary airways. It's the intrapulmonary airway pressure which will change with respect to the intrapulmonary airways, where the diameters can change, because these are the ones which are present inside the lungs, not the trachea and the bronchi. Yes, there will be some amount of, the different, what you're trying to ask me, at which level of the bronchioles. Now, see, you've got the, you will see it in all the bronchioles present within the lung parenchyma. Have you understood? Within the lung parenchyma. Even the ones which have got small amounts of cartilage in it, because please remember, they are subject to the changes in pressure. If time permits, could you please explain the spirometry patterns for flow volume curves? Flow volume curves, Mumbai, will be done, or may have been done already in respiratory medicine. So probably that's what sir told me, that he has already covered these flow volume curves with you. That is why I'm not doing them with you. But if possible, definitely I'll try and explain them to you. Right? This is your Vishakhapatnam, ruk to ja beta. We are on compliance. We have to talk about surfactant. Without surfactant, how will my lung physiology be complete? Yeah, okay. So compliance of lung and chest volume is 0.1 liters per centimeter of water pressure. Okay? This is your lung compliance. So let's see what are the factors affecting lung compliance. Let's take the factors affecting lung compliance. Yes? Factors affecting lung compliance. Number one is the size of the lung. Larger the lung, more is the compliance. That means if I compare the compliance of a lung of a child and lung of an adult, the adult lung will have a better compliance. Larger the lung, more the compliance. Right? Now, supposing I actually want to compare the compliance of two lungs and I want to eliminate size as a factor. Please remember, size is a confounding factor here. I do not want size to come in. Then the better term to compare is a specific compliance. What is specific compliance? This is compliance divided by FRC. Specific compliance is compliance divided by the functional residual capacity. This is a better term to compare in the case of two lungs. If I want to compare the compliance of two lungs, I must do specific compliance, which is compliance divided by FRC. See, let's see what happens in emphysema. Now, emphysema, we've discussed that there is a lung, in emphysema, there is destruction of the elastic fibers of the lungs. When there is a destruction of the elastic fibers of the lungs, now it is easier to inflate this lung. There is decreased resistance to expansion. Easier to inflate this lung, right? But at the same time, the lung recoil decreases. So what happens in emphysema? Because it is easier to inflate the lung, there is increase in lung compliance. But at the same time, because like I said, the recoil is less, there is a decrease in the FRC, functional residual capacity. Increase in compliance, but a decrease in, increase in compliance, but just see this once again, increase in compliance, and there is a increase in FRC. Why increase in FRC? Because the lung recoil is less. If recoil is less, the functional residual capacity now increases. But remember, the increase in FRC is more than the increase in compliance. Manika? Compliance has increased, but increase in FRC is more than the compliance. So what happens to my specific compliance? It shows a decrease, right? So this is a question which has been asked: in which lung condition there is increase in compliance but a decrease in specific compliance? And your answer becomes emphysema. Emphysema में compliance बढ़ा. Compliance क्यों बढ़ा? Because there is destruction of elastic fibers, easier to inflate this lung, easier to obtain a larger change in volume. But because the elastic fibers have got destroyed, there is a decreased recoil. There is a decreased recoil, and because of that, there is increase in FRC, but the increase in the denominator is more than the increase in the numerator. Therefore, the specific compliance decreases. So, increase in compliance, but a decrease in specific compliance is seen in emphysema. The second factor which can affect compliance, is compliance is less. Compliance is less at extremes of lung volume. Compliance is less at extremes of lung volume. Extreme inflation, extreme deflation. Compliance is less at extremes of lung. Extreme inflation, extreme deflation, there will be less compliance decreases. Number 3, compliance is more in deflation than inflation. During expiration, it's more than an inspiration. Deflation than inflation. Number 4, compliance is inverse to the surface tension. And surface tension is inverse to the amount of surfactant. Surfactant lowers the surface tension. Lower the surface tension, more will be the compliance. Right? So compliance is inverse to the surface tension. Surface tension is inverse to the surfactant. In other words, compliance becomes directly proportional to the amount of surfactant. Compliance is proportional to the surfactant. Clear? So these are the factors affecting compliance. So let's see compliance once again. What are the factors affecting compliance? Number one, I told you size of the lungs. More larger the lung, more the compliance. Then I discussed with you what is specific compliance. Compliance divided by FRC. Emphysema, Vishakhapatnam, just listen to this very carefully. There is increase in compliance. Why? Why is there increase in compliance? Because what has happened is elastic fibers have got destroyed. Right? So there is less resistance to expansion. Right? Now, when there is less resistance to expansion, it's easier to inflate this lung. So compliance increases. Now you get a larger change in volume for the same change in pressure. But because the elastic fibers have got destroyed, now the recoil is also less. You remember I told you about elastic fibers? Pull them, there is a tension generated. Leave them, there is a recoil. So because the recoil is less, now there is increase in FRC. So specific compliance in fact now decreases. Numerators increased, FRC has increased much more. Therefore there is a decrease in the specific compliance. And like I said, this is what has been asked. Increase in compliance but a decrease in specific compliance is emphysema. Second factor, compliance is less at extremes of lung volume. Third, compliance more in deflation than inflation. And like I said, compliance is inverse to surface tension. Surface tension is inverse to surfactant. So compliance becomes directly proportional to the amount of surfactant, right? Now let's do surfactant in detail, right? Surfactant in detail. So let's discuss this. Surfactant secreted by type 1, type 2 pneumocytes. ये तो सबने बोल दिया. Surfactant secreted by type 2 pneumocytes. Yes? Now tell me this question. Which is more numerous? Which is more numerous? Type 1 या type 2? अब देखो. The one which is more numerous is in fact the type 2 cells. Type 2 cells are more numerous than type 1. Have you understood? The surfactant secreting cells are in fact more numerous. They are 60% of the cells, whereas type 1 are only 40%. But if I look at the alveolar surface area, if I ask you which cells form the majority of the alveolar surface area, type 1 cells form almost 95% of the alveolar surface area and type 2 cells form just 5% of the alveolar surface area. So in terms of number, it is type 1, but in terms of alveolar surface area, in terms of number, it is type 2, but in terms of alveolar surface area, it is type 1 cells. So please be careful with these two questions. One is asking you the number, the other is asking you alveolar surface area. Right? Now, surfactant, the chemical composition is of course dipalmitoyl phosphatidylcholine. Dipalmitoyl phosphatidylcholine. This is the chemical composition of the surfactant. Okay? Now let us see what is the function of surfactant. What does surfactant do? Surfactant, like I said, it lowers the surface tension. It decreases the surface tension. Surfactant lowers the surface tension. Now let us see what is surface tension. Let's discuss the surface tension. Now this is liquid here, right? Now this is liquid, this is air, right? So whenever there is a liquid air interface, please understand there has to be a liquid air interface. A liquid air interface has to be present. What is liquid air interface? That means there is a sort of liquid and air. So there's an interface, right? So liquid air interface. Now in this liquid air interface is present. Now the molecules on the topmost layer, they will be attracted towards one another. Molecules on the topmost layer will be attracted towards one another, and this is what creates the surface tension. Molecules on the topmost layer will be attracted to one another, and thus this creates a surface tension. That is why, see, when you see the shape of the meniscus, meniscus is like this, right? Liquid air interface. So molecules on the topmost layer are attracted to one another, and the meniscus is like this. Look at the shape of a raindrop. Raindrop is spherical in shape. Liquid air interface. A raindrop is spherical. There is a surface tension. There is a tension which is created on the surface, because of which the raindrop becomes spherical. Now, what is the role of surfactant? Now, what does surfactant do? Surfactant is a kebab mein haddi. And who do you call a kebab mein haddi? Kebab mein haddi is anyone or anything, actually anyone, who comes between two people and decreases the attractive forces between two people. And this is exactly what your surfactant does. Surfactant goes and sits between the water molecules. It goes and sits between the water molecules like a kebab mein haddi and decreases the attractive forces between the water molecules, and now decreases the surface tension. Is it clear? It breaks these attractive forces between the water molecules and reduces the surface tension, right? So if we've understood what is the action of surfactant, I'm going to do one conceptual question with you. What is this question? Now, this was an AIMS question. This is completely a conceptual question. So do not ask me, how can this happen? How is it even possible? I'm just asking you this question to test your concept of surface tension and surfactant. Now, there are two types of lungs. There are two types of lungs. There is a saline ventilated lung. Saline ventilated lung, that means it is ventilated with saline. So that's why I said, don't ask me how is it possible. There is a saline ventilated lung, and there is a normal air ventilated lung. Normal air ventilated lung, matlab there will be a thin layer of alveolar fluid, and a thin layer of alveolar fluid is present, and of course you've got air here. So you've got a saline ventilated lung and an air ventilated lung. Now my question is, which one has a better compliance? Let me see how many of you can answer this. Great, I'm so happy. You've got quite a few of you have got this answer as A. Why? Mangalore and Jabalpur, just listen to me. Why does this is the saline ventilated lung will have a better compliance? Now, why is this? See, because please understand, in the saline ventilated lung, there is no liquid air-liquid interface. When there is no liquid air interface, therefore there is no surface tension. If no surface tension, and remember I said compliance is inverse to the surface tension. If there is a no surface tension force, so compliance becomes more. Whereas in the case of an air ventilated lung, there is a liquid air interface which is present, and that means there is a surface tension and compliance is inverse to the surface tension. There is a surface tension force present over here, so therefore it has a lower compliance as compared to the saline ventilated lung. Karamsad, very good. No fluid air interface. But hydrothorax mein, what you're trying to tell me, I think what you're trying to indicate is what happens if there is a pulmonary edema. What happens to compliance? Pulmonary edema, now you will have a liquid air interface now. It is not a completely liquid ventilated lung. I'm talking about a complete liquid ventilated lung. Let's say the closest we can come to, closest physiological condition where you have a liquid ventilated lung is, let's say, a fetal lung, right? Fetus in the amniotic fluid, now the lung is completely filled with liquid, right? So that is the only physiological situation where, so this is just a conceptual question where you are ventilating the lung with saline. I mean, we've taken lungs, dissected lungs out, and we're ventilating them with saline. In that case, compliance will be better of this particular lung because there is no liquid air interface, right? So let's have a look at which are the hormones which affect. Let's see the hormones which affect the surfactant secretion. Hormones which affect the surfactant secretion. Now, hormones which affect the surfactant secretion. The first one is thyroid hormones. Thyroid hormones, they increase the type 2 pneumocyte activity. So they increase the production of surfactant. That is why our hypothyroid patient is more likely to develop RDS, respiratory distress syndrome. Our hypothyroid patient is more likely to develop RDS. Number 2, insulin. Insulin decreases the type 2 pneumocyte activity. That is why babies born to diabetic mothers, these are more likely to develop respiratory distress syndrome, isn't it? If the mother is a diabetic, the baby will also, mother has got hyperglycemia, babies will also have hyperglycemia, and then, but baby or the fetus has hyperglycemia, and fetus also has hyperinsulinemia. And insulin, I just told you, decreases type 2 pneumocyte activity. So after birth, this child is more likely to develop respiratory distress syndrome. Number 3, steroids. Glucocorticoids, right? Glucocorticoids, what is the action of glucocorticoids? Glucocorticoids, they increase the type 2 maturation. This is glucocorticoids. Steroids will increase the type 2 maturation. So these are your hormones, thyroid hormones, insulin and steroids, right? So these increase the type 2 maturation. Next, let's see. Other factors which decrease surfactant. What are these? Which decrease in surfactant can be seen in the following conditions. Number 1, surfactant decrease will be seen whenever decrease in surfactant is seen whenever there is an obstruction of a main bronchus. This causes a decrease in surfactant. Number 2, if there is an obstruction of a branch of pulmonary artery. Number 3, obstruction of a main bronchus, obstruction of a branch of pulmonary artery. Number 3, prolonged inhalation of 100% oxygen. Number 4, cigarette smoking. This will also decrease the surfactant, right? So these are decrease in surfactant will be seen whenever there is obstruction of a main bronchus. For example, in lobar collapse or a lung collapse, the collapsed area will always have a decrease in surfactant. Obstruction of a branch of pulmonary artery, prolonged inhalation of 100% oxygen, and cigarette smoking. These are the conditions where there is a decrease in surfactant. Clear? Right. Next, let's see 2 important questions. This is Allahabad. This is prolonged inhalation of 100% oxygen. It has been found that whenever we give 100% oxygen for a very long time, right, that causes a decrease in surfactant. Exactly what is the reason for this? We cannot say, but yes. Belgam, what did I say? Thyroid hormones increase the type 2 pneumocyte activity. Hypothyroidism, there will be less type 2 pneumocyte activity, so RDS becomes common. This is what you're saying? Ma'am, why does increase type 2 pneumocyte? Hypothyroidism means decreased है ना? So RDS becomes common. ठीक है? So these are your conditions which will affect surfactant secretion. Clear? Next, let's see two important questions here which always create a confusion. And what are these? The first ones are, when you look at the first question is, what is responsible for maintaining alveolar stability? Alveolar stability is maintained by surfactant, or B, is it alveolar interdependence? Is maintained by surfactant or alveolar interdependence, right? Please understand both of these. What is alveolar stability? Alveolar stability matlab the alveoli do not collapse at the end of maximum expiration. We've discussed that already. Alveoli do not collapse at the end of expiration. How is this prevented? Is it because of surfactant, or is it because of the alveolar interdependence? Right? I said both. What is alveolar interdependence? Alveolar interdependence is, like, for instance, when at the end of maximum expiration, the smaller alveoli will tend to collapse. But what prevents these is there is a pull exerted by the larger alveoli, which prevents the collapse of the smaller alveoli. That is called alveolar interdependence. Both these contribute to maintaining an alveolar stability, but when you have to choose between the two, your better answer becomes surfactant. Right? Both of them are helping to maintain alveolar stability, alveolar interdependence plus surfactant. But please understand that if you want to mention one, then it has to be surfactant. Yes, Jammu. Good. All right. So this is your alveolar interdependence. Okay. Now this is next question. This is alveoli are kept dry. Alveoli are kept dry due to A, is it surfactant, or B, is it a negative interstitial fluid pressure? Negative interstitial fluid pressure. Please understand again both. Even surfactant helps to keep the alveoli dry. Right? But if I have to choose between the two, my better answer becomes negative interstitial fluid pressure. And the reference here is Guyton. Right? B more than A. Absolutely right, Dehradun. This is negative interstitial fluid pressure, which helps to keep the alveoli dry. Negative interstitial fluid pressure in the lungs is it's almost -8 millimeters of mercury. And this helps to keep your alveoli dry. Right? Most other tissues, it's about -1, but in the lungs, it's about -8, right? This helps to keep our alveoli dry. So this is as far as your compliance is concerned. Next, let's see. Let's go on to what is the, how do I calculate? Now next, let's see. How do I calculate the partial pressure of oxygen in the atmosphere? What is the partial pressure of oxygen in the atmosphere? Now, to calculate partial pressure of oxygen in the atmosphere, I must know what is the percentage of oxygen, which is 21%, into whatever is the barometric pressure. Right? 21 upon hundreds. You know this, in atmospheric air, the percentage of oxygen is 21%, and expired air expired air is 16%. Yes, in atmospheric air, the percentage of oxygen is 21%, and in the expired air is 16%. 5% has been used up by the body. This has been a question which was asked that in mouth-to-mouth respiration, mouth-to-mouth respiration, the percentage of oxygen is 16, right? That is also sufficient to revive a dying man. Have you understood? 16%, but it's quite good to revive a dying man. That is your mouth-to-mouth respiration. Now, what is PB? This is barometric pressure. So how do I calculate the partial pressure of oxygen in the atmosphere? I multiply the percentage into the barometric pressure. Now, supposing the barometric pressure is 760, you are at sea level, pressure is 760. This becomes 21 upon 100 into 760, which gives me a value of approximately 160 millimeters of mercury, right? But supposing you are at some high altitude, let's say the barometric pressure is now 500. Please remember, as we go into high altitude, the percentage of oxygen remains the same. Have you understood? The fractional concentration of oxygen will remain the same, that is 21%. But the barometric pressure, atmospheric pressure, will decrease. Right? So this, now what happens? Now 21 upon 100, now this gives me a partial pressure of oxygen approximately 105 millimeters, approximately in fact 105 millimeters of mercury. माने जब आप sea level पर है, the partial pressure of oxygen is 160. At high altitude where barometric pressure is 500, the partial pressure of oxygen has been reduced to 105. Right? Now I am interested. Now see, when we inspire, now this is, right? Now when we inspire, there are some changes which take place before the air reaches the alveoli. There are some changes which take place, like I said, before the air reaches the alveoli. Now, as your air passes through the respiratory tracts, it is warmed, brought to body temperature, and water vapor is added. And the partial pressure of water vapor which is added is 47 millimeters of mercury. And this is irrespective of altitude. Makes a lot of sense now. Why is it irrespective of altitude? Why is it irrespective of altitude? Because please remember, this water vapor is not coming from the atmosphere. This is coming from your respiratory tracts into the inspired air. So this is irrespective of altitude, right? It is irrespective of altitude. It is irrespective of the environmental temperature. It has nothing to do with the environmental temperature, nothing to do with the altitude. Because this partial pressure of water vapor is now, this water vapor is being added by your respiratory tracts to the inspired air. But yes, this partial pressure of water vapor, this can depend on your body temperature. If you are febrile, then obviously this partial pressure of water vapor will increase. It will depend on your body temperature. It can depend on the environmental humidity, right? See, if the air is already 100% humidified, then there's going to be no addition of water vapor over here. Have you understood my point? So this will depend, it is irrespective of altitude, irrespective of environmental temperature. In other words, can I say the addition of water vapor has diluted the inspired air? Have you understood? We have humidified the inspired air and we have diluted the gases present in the inspired air. You have added water vapor. Okay? So let's see. Now what happens? Now this air manages to reach the alveoli. These are the two lungs here. Yes. The air has reached the alveolus. Now, in the alveolus, there is addition of CO2. And this CO2 further dilutes the alveolar air. It further dilutes the alveolar air. So basically, what am I trying to tell you? I've been giving you the background information because I want to know how much is the alveolar O2. What changes have taken place by the time the air has reached the alveoli? There has been addition of water vapor. There is addition of CO2. Okay? So there is some changes in the atmospheric air which have occurred. So I want to know, is the alveolar O2 the same or has it decreased? Has it changed? And this alveolar O2 is given by what is known as the alveolar gas equation. Alveolar gas equation. Right? See, you must understand, because for me, this alveolar O2 is very important. This alveolar O2 is very important because this will determine how much is the arterial O2. Have you understood? Alveolar O2 will determine the arterial O2. Have you understood? Your arterial O2 will be equal to the alveolar O2. So whatever is the alveolar O2 will now, this is based on the Henry's law. And what does Henry's law state? Henry's law states that the concentration of a gas in a solution here is plasma. The concentration of gas in a solution will be equal to the partial pressure of the gas above the solution. The partial pressure of the gas above the solution here is the alveolar O2. So my arterial O2, small a is arterial, capital A is alveolar. My arterial oxygen tension will be determined by the alveolar O2. So for me to know what is the alveolar O2 becomes very important. See, one thing I can do is either I take an alveolar sample and measure the alveolar O2, which is not easy to do. And the other way is to calculate the alveolar O2 with the help of alveolar gas equation. Clear? So let's see what is the alveolar gas equation. Alveolar gas equation. Now this equation states that alveolar O2 in mm of Hg, this is equal to barometric pressure minus the water vapor pressure into the fractional concentration of oxygen minus alveolar CO2 divided by R, right? So this is barometric pressure minus the water vapor pressure into the fractional concentration of oxygen minus alveolar CO2 divided by R. This is your alveolar gas equation. Now your barometric pressure, normal barometric pressure is 760. Water vapor pressure is 47. And fractional concentration of oxygen, you know, is about 21 upon 100. 21 upon 100. Now, alveolar CO2, please remember, is the same as the CO2 in the venous blood. And CO2 in the venous blood is 45. Now comes what is R. So let's discuss what is R. R is the respiratory, this is the respiratory quotient. And what is the respiratory quotient? This is the volume of CO2 produced divided by volume of oxygen consumed. Volume of CO2 produced divided by volume of oxygen consumed. Now, this respiratory quotient depends on your diet, right? If you are on a carbohydrate diet, your RQ will be equal to 1. That means the amount of oxygen being consumed to metabolize carbohydrates will be equal to the amount of the CO2, or the amount of CO2 produced after metabolism of carbohydrates is equal to the amount of oxygen consumed. But on a mixed diet, RQ is equal to 0.8. In other words, there was a question which had come, and let me see if you can answer this question. What is the RQ of the brain? RQ of the brain, is it 0.7, 0.8, 0.9 or 0.97 to 0.98? Bolo. A, B, C or D? D, isn't it? Because after all, what is the major fuel for the brain? It is, what is the major fuel for the brain? This is carbohydrates, glucose. Brain can use keto acids during starvation. But otherwise, the major fuel for the brain is glucose. Major fuel for the heart is fatty acids. Have you got my point? But for the brain, it's glucose, and brain can use keto acids during starvation. So this is your respiratory quotient. So let's go back to what I was trying to do with you, and that is your alveolar gas equation. So let's try and solve this equation now. Now let's see. Alveolar O2 is equal to 760 minus 47 into 21 upon 100 minus 45, and in this equation we take RQ equal to 1. Make life simple for us. Yes? And when I solve this, I get an alveolar O2 of 105 millimeters of mercury. 105 millimeters of mercury. Are we clear? So what has happened? Atmospheric air, the O2 was 160. By the time the air reaches the alveoli, in the alveoli, the PO2 has been reduced to 105 millimeters of mercury. Clear? अब इसका एक question लेते हैं. Let's try and reinforce this. This alveolar gas equation was asked in 2012, All India PG 2012. Let's see if you can answer this question. This says, there is a person who is breathing 100% oxygen at 4 times atmospheric pressure. 100% oxygen at 4 times atmospheric pressure. 100% oxygen at 4 times atmospheric pressure. How much is his alveolar O2? Is it 1000 millimeters of mercury, 2000, 3000, or 4000 millimeters of mercury? Firstly, let's see which situation, where do you get 100% oxygen at 4 times atmospheric pressure? What is this situation? Where do you get something like this? Let me see if any of you can answer this. नहीं, विशाखापट्टनम, intermittent positive pressure ventilation नहीं. Atmospheric pressure is 4 times. The surrounding pressure is 4 times. Okay, Mysore, great. You've managed to impress me. You've got this answer. But I wanted to ask, not even CPAP. You must understand it is the atmospheric pressure which I'm talking about, not the pressure with which you're ventilating the lungs. Have you understood? So this is, okay, underwater. That means in a diver. Yes. Anything else? At sea level, can I get some situation like this? Is it possible to get a situation like this at sea level? Underwater, got it. Diver, yes, got it. Can I? No? Think about it, Mysore. Have you heard of hyperbaric oxygen chambers? Hyperbaric oxygen chamber. This comes as a short note in final MBBS. Isn't it? Hyperbaric oxygen, oh yes. Hyperbaric oxygen chamber is, what are the indications for hyperbaric oxygen therapy? Gas gangrene, carbon monoxide poisoning, gram-negative septicemias, non-healing ulcers. Yes, so these are the indications of a hyperbaric oxygen chamber. So this is a hyperbaric oxygen. See, basically hyperbaric oxygen chamber, right? Hyperbaric oxygen therapy, like I said, the indications are gas gangrene, gram-negative septicemias, non-healing ulcers, carbon monoxide poisoning, right? And so let's calculate how much is the alveolar O2. Now 4 times atmospheric pressure means it's 4 into 760. 4 into 760, which is 0.43040 mm of Hg. So let's see the alveolar gas equation. Alveolar O2 is equal to 3040 minus 47 into 100 upon 100 minus 45 divided by 1. Minus 45 divided by 1. This is my alveolar gas equation. So 100 upon 100 obviously gets cancelled, right? So my answer now becomes 3000 millimeters of mercury. Mane, when he was inspiring atmospheric air, his alveolar O2 is 105. Like in the moment I give him 100% oxygen at 4 times atmospheric pressure, there is a 30 times increase. 30 times increase. It now becomes 3000. And why is this value important for me? Because like I told you, if the alveolar O2 is 3000 millimeters of mercury, then by the Henry's law, the dissolved O2 or the arterial oxygen tension will also be 3000 millimeters of mercury. And this becomes important. See, gas gangrene, Clostridium. Clostridium is anaerobic. So if I increase the arterial oxygen tension, I will kill all the anaerobic bacteria. Have you got it? Gram-negative septicemia, I increase the oxygen tension from 100 to 3000, again I will kill all the anaerobic bacteria. Have you got this point? So this is the idea behind giving hyperbaric oxygen. Okay? Chalo. Now we've done the alveolar gas equation. Next, let's go on to respiratory exchange. Let's discuss respiratory exchange. Yeah, Mumbai, we will still abuse the Henry's law because this, please remember, this it's a semi-permeable membrane, but it allows almost a free movement of the respiratory gases. Remember, we've done this yesterday when I said that simple diffusion, respiratory gases move by simple diffusion. So it's permitting almost a free diffusion of of course, varies for different gases because we know that the physical properties of different gases are different, but it is almost allowing a free movement of gases across this membrane. That is why Henry's law will become applicable over here. Good, intelligent question, Mumbai. Right. So this is your, now let's see respiratory exchange. Respiratory exchange. Now before I do the respiratory exchange with you, let's just discuss a few values so that things become a little more clear, and then we'll do the details of respiratory exchange. Like for instance, in the case of oxygen transport, now there are two things that you have to know. There is a PO2 and there is an SO2. Okay? Bikaner, in respiratory system you have a flow volume curve which like Mysore has already informed me will be done in medicine, so don't worry about it. Flow volume curves and flow volume loops. Pressure volume curve and pressure volume loops, I will do it in CVS. Those are related to the left ventricular pressure and volume, right? So what you have here in the respiratory system are flow volume curves. Okay, so which has been informed, which I have been told has already been covered in medicine. So if you have a medicine class coming up, it will probably be covered over there. Okay, that's a flow volume curve and flow volume loops. Pressure volume curves and pressure volume loops, I'll do it in CVS. Okay, so now this is PO2. Now PO2 is in millimeters of mercury, and SO2 is a percentage, right? PO2 is the partial pressure of oxygen in millimeters of mercury. SO2 is the saturation of hemoglobin, right? Now, in the arterial blood, the PO2 is 95 and SO2 is 97 to 90. What do I mean by saturation? Please understand, each hemoglobin has got 4 heme. There are 4 heme present. Each heme can bind with 1 oxygen each. When all the 4 heme have got 1 oxygen each, it is said to be 100% saturated. When only 3 have got an heme and 1 does not, it is 75% saturated. If only 2 have got an oxygen, it is 50% saturated. If only 1 has got oxygen, it is 25% saturated. This is your percentage saturation. Arterial blood is almost 100% saturated. Venous blood, on the other hand, is 40 is the dissolved oxygen, partial pressure of the dissolved oxygen, and the saturation is 75%. So just these 2 important values. And how do we measure PO2? See, PO2 is measured by arterial blood gas analysis. If I use an arterial sample, I'll get an arterial value of PO2. If I use a venous sample, I'll get venous PO2. Saturation I can measure with the help of pulse oximetry. You know, the pulse oximeter which you put on the finger. But please remember, the finger pulse oximeter only gives us arterial saturation. Clear? If I want to know, the blood gas analysis will also give me saturation. If you look at the printout that you get when you see the computerized blood gas analysis, it also gives you saturation. Okay? So be careful. Like I said, pulse oximeter which you attach over here gives you the value of only the arterial saturation, not the venous. Okay? Now let's see what is the respiratory exchange. Now, this is the alveolus here, and we had decided that the alveolar O2 is 105 millimeters of mercury. 105 millimeters of mercury, and this is the pulmonary capillary. Now the PO2 on the venous side of this pulmonary capillary, that means in the pulmonary artery, which is actually the venous side of the pulmonary capillary, this will be PO2 is 40. Blood which moves into the pulmonary capillary has a PO2 of 40. Now O2 will move from the alveolus into the pulmonary capillary because there's a difference in the partial pressure. So PO2 will move from the alveolus into the pulmonary capillary. So in the pulmonary vein, which is the arterial blood now when I take the PO2, it is 100 millimeters of mercury. It's not exactly 105. It should have been 105, but it is not exactly 105. Now let me tell you why. Why is it that in the pulmonary vein, which is the arterial blood, the PO2 is not exactly 105, it's slightly less, 100 millimeters of mercury. Why has this happened is, because please remember, not all the alveoli are ventilated equally. We've already discussed this. In the standing posture, for instance, there is a gradient, right? So not all the alveoli are ventilated equally. And number 2, not all the pulmonary capillaries are functioning at the same time. So what will happen when I take the blood sample from the pulmonary vein? The PO2 now reduces slightly. It should have been the same as the alveolar O2, but PO2 now, because the average comes down slightly, isn't it? Because number 1, all alveoli are not functioning at the same time, and number 2, no, this is not physiological shunting, Jumbo. This is simply that not all the alveoli are not functioning at the same time, and all the pulmonary capillaries are not open at the same time. So PO2 has come down to 100. This is not physiological shunting. Okay? Now this pulmonary blood from the pulmonary vein now goes into the left atrium, left ventricle, and comes into the aorta. Now, in the aorta, when I take a sample from the aorta, the PO2 in the aorta is now 95 millimeters of mercury. And this decrease, pulmonary vein, the blood was, PO2 was 100. Raste mein, there was only left atrium, left ventricle. And in the aorta, when I took a sample, the PO2 has been reduced to 95. This is because of physiological shunting. Jammu. Right? This decrease in PO2 is because of physiological shunting. And what is physiological shunting? Physiological shunting means, Jabalpur, good. This is your venous blood from the bronchial circulation, venous blood from the coronary circulation, not the complete venous blood, only a small fraction of the venous blood from the coronary circulation, from the bronchial circulation, drains directly into the left atrium. Drains directly into the left atrium. So deoxygenated blood is mixing with the oxygenated blood. So the PO2 reduces to 95 millimeters of mercury. This is your physiological shunting. See, venous blood should go to the right side of heart. But venous blood is going towards small fraction of the venous blood from the coronary circulation, bronchial circulation is coming directly into the left atrium. Clear? So this is what is known as physiological shunting. Now blood from the aorta, arteries, arterioles, it goes into the tissue capillaries. Tissue capillaries on the arterial side. On the arterial side, tissue capillaries, arterial side, PO2 is 95. Then of course oxygen goes into the tissues. PO2 on the venous side is reduced to 40. It now goes to the right atrium, right ventricle, pulmonary, artery, and so on. This is your complete circulation, pulmonary as well as the systemic circulation. Right? Now my first question, the question which I want to know over here, largest change in PO2. Largest change in PO2. Which capillary is Visakhapatnam? Largest change in the PO2. It's in front of you and you're still writing tissue capillaries. Visakhapatnam, Hyderabad, Allahabad, Karamsad, all of you are writing wrong. The values are in front of you, in the board in front of you. Largest change in PO2 is at the level of tissue capillaries or the pulmonary capillaries. Look at this. Pulmonary capillaries, the change is 40 to 100. Tissue capillaries is 95 to 40. Where do you see the largest change in PO2? Thank you, Hyderabad. Finally, this is your pulmonary capillaries, not the tissue capillaries. Is it okay? Yes. 40 to 100 at the level of the pulmonary capillaries and 95 to 40 at the level of the tissue capillaries. Right? So largest change in PO2 is at the level of the pulmonary capillaries. Okay. Now, please understand, I said the alveolar O2 is 105, but the pulmonary vein, the PO2 is 100. Why this difference is? Because I told you all the alveoli are not being equally bent. If you take a single alveolus, single pulmonary capillary, then the PO2 at the end of this pulmonary capillary will be 105. But like I said, not all the pulmonary capillaries are active at the same time. Not all the alveoli are being ventilated equally. Therefore, this reduces to 100. Somebody had asked me this question. So be careful. There is a small amount of difference in the PO2. But this difference, 195 here between the pulmonary vein and the aorta, this is because of physiological shunting. Please understand, there is a normal alveolo-arterial gradient. And how much is this alveolo-arterial gradient? It should be less than 5 to 10 millimeters of mercury. Alveolar arterial gradient माने alveolar में 105 है, arterial blood में 95 है. So this is your normal alveolar arterial gradient. Yeah, because Dehradun, whether I can actually call it wasted ventilation? No, because see, there is, what is the average VQ ratio for the whole lung? It is 0.8. Have you got it? It's not 1 throughout the lungs. So there will be some amount of decrease in the diffusion here, right? Okay. So this is alveolar arterial gradient. What is the alveolar O2? Alveolar O2 is 105. Arterial is 95. Difference between the two should be less than 10. Less than 5 to 10 millimeters of mercury. If it is more than 10 millimeters of mercury, that means there is obviously some problem in diffusion of gases. Either there is a VQ mismatch, or there is fibrosis, or something has happened. Right? So there is a normal, it should not exceed more than 10. That is what you mean by alveolar arterial oxygen gradient. Alveolar arterial oxygen gradient. Chalo, I'll take a 10-minute break. Okay? Let me have a cup of tea and get back, and then we have to do the balance of the respiratory system. Okay? Chalo, okay. Right? O2 transport. O4. O2 transport. Let's see O2 transport. Now you know that oxygen is transported in 3 ways. There are in 2 ways. 2 ways for transport of oxygen. And how is oxygen transport occur? How does it occur? Number 1, in combination with hemoglobin. In combination with hemoglobin, and number 2, as the dissolved O2. Oxygen transport will be in 2 ways, that is with hemoglobin and as dissolved O2. Mane, if I want to know what is the oxygen content of the blood, this is oxygen content of the blood will be oxygen in combination with hemoglobin and number 2, the dissolved O2, right? Dissolved O2. So oxygen in combination with hemoglobin plus the dissolved O2, this will give us the oxygen content of the blood. Now, how much is the oxygen in combination with hemoglobin? This is hemoglobin in grams per deciliter into 1.34. Now, why did I multiply hemoglobin into 1.34? Because you know that 1 gram of hemoglobin can transport 1.34 ml of oxygen. 1 gram of hemoglobin can transport 1.34 ml of oxygen. So this is hemoglobin in grams per deciliter into 1.34 into a third factor, and that is the percentage saturation. This will give me oxygen in combination with hemoglobin. For example, if I have a patient whose hemoglobin is 10 grams and I want to calculate how much is the oxygen being transported by the hemoglobin. So see, if it is an arterial blood sample, the oxygen transported will be 10 into 1.34 into percentage saturation of arterial blood is 100 upon 100, right? Almost 100 upon 100. So that means now the oxygen being transported by the arterial blood becomes 13.4 ml per 100 ml of blood, right? On the other hand, if it's venous blood, it becomes 10 into 1.34 into 75 upon 100. The percentage saturation of venous blood the percentage saturation of the venous blood, this is less, this is about 75%. So it's 10 into 1.34 into 75 upon 100. So please remember, to calculate the oxygen content, you need to have hemoglobin in grams per deciliter multiplied by 1.34 and multiplied by the percentage saturation. Hemoglobin in grams per deciliter into 1.34 into the percentage saturation. Clear? Now, plus I said there is the, see what is the oxygen carrying capacity of the blood? How do I calculate the oxygen carrying capacity of the blood? I have to multiply the hemoglobin into 1.34 into 100 upon 100. That will give me the maximum amount of oxygen that can be carried by the arterial blood. Right? Now, for example, see what is anemia. Let's take another example. What is anemia? Now, how do I define anemia? Anemia is nothing but, what is anemia? Anemia is a decrease, it's a quantitative or a qualitative decrease in hemoglobin and or RBC count, resulting in a decreased oxygen carrying capacity of the blood. Right? What is anemia? Anemia is a quantitative decrease in hemoglobin. What do I mean by quantitative decrease? Either the hemoglobin itself is reduced. Yes, that is like your iron deficiency anemia, megaloblastic anemia, hemolytic anemia. That hemoglobin itself is reduced. So quantitative decrease in hemoglobin or a qualitative decrease in hemoglobin. What do I mean by qualitative decrease? That means hemoglobin is normal, but the percentage saturation of the arterial blood, instead of being 100%, is now reduced, right? So this is a qualitative decrease. Percentage saturation is less. For example, the examples where hemoglobin is normal but the percentage saturation is less is methemoglobinemia. Methemoglobinemia. See what has happened in methemoglobinemia? You've got 4 heme. Each of these have got iron in the ferrous form. But let's say one heme has got iron in the ferric form. So oxygen will combine with the 3 heme which have got iron in the ferrous form but cannot combine with the third one. So what has happened to the percentage saturation of the arterial blood? It is, instead of being 100% saturated, it's now only 75% saturated. So oxygen being transported by the arterial blood will decrease. Oxygen carrying capacity decreases. Right? Or if you have a situation of carbon monoxide poisoning. Carbon monoxide poisoning. What's happened in carbon monoxide poisoning? Again, carbon monoxide has formed. Let's say carbon monoxide has a very strong affinity for heme. Carbon monoxide forms an almost irreversible combination with heme. So that means this heme is not available to combine with oxygen. Let's say carbon monoxide is combined with 2 heme. These are not available to combine with oxygen. That means now oxygen can combine with only the remaining 2 heme. So percentage saturation of the arterial blood has been reduced to 50%. So oxygen carrying capacity reduces. Right? So these are your, this is how you define anemia. Either a quantitative decrease in hemoglobin or a qualitative. Qualitative means now the percentage saturation is reduced. Either way, there will be a decrease in the oxygen carrying capacity. Right? So this is your oxygen in combination with hemoglobin. But there is a second factor here which is important, and that is your dissolved O2. Now dissolved O2, we have said, we have measured dissolved O2 in the, we have measured dissolved O2. We have measured dissolved O2. Dissolved O2 by PO2. Isn't it? Blood gas analysis gave us PO2 in millimeters of mercury. Now I want the answer. I want to know how much is the dissolved O2 in ml per 100 ml. And to do that, to convert the PO2 in millimeters of mercury into ml per deciliter, I must know what is the solubility of oxygen. what is the solubility of oxygen? Solubility of oxygen, this is 0.003 ml per deciliter per millimeter of mercury. This is the solubility of oxygen. Solubility of oxygen is 0.003 ml per deciliter per millimeter of mercury. Right? Now, what is this solubility of oxygen? 0.003 ml per deciliter per millimeter of mercury. What does this mean? That is, if PO2 is 1 millimeter of mercury, then the dissolved oxygen is 0.003 ml per deciliter of blood. This is what is meant by solubility is 0.003 ml per deciliter per millimeter of mercury. So it means if PO2 is 1 millimeter, dissolved oxygen is 0.003 ml per 100 ml of blood. That means if PO2 is 100 millimeters of mercury, then the dissolved oxygen is 0.3 ml per deciliter of blood. Right? If PO2 is 90 millimeters of mercury, dissolved oxygen is 0.27 ml per deciliter of blood. If PO2 is 80 millimeters of mercury, it is 0.24 ml per deciliter of blood. 70 hai to 0.21 ml per. So it's not difficult to calculate at all. Right? If you remember the solubility is 0.003 ml per deciliter per millimeter of mercury. This is your solubility of oxygen. Clear? Now this is right. Now let's take the first question related to this topic and let me see if you can answer this first question. The first question states that we've done a part of this question already, so I'm now let's see what is this question. It says, if a person is breathing 100% oxygen at 4 times atmospheric pressure, how much is his dissolved oxygen per 100 ml of blood? Is it 3 ml? Is it 6 ml? Is it 9 ml? Is it 12 ml? Answer this now. 100, we've already done a part of this question. We had reached over here that PO2 here is 3000. And remember I said if this is 3000, then this will be maximum difference can be 10 millimeters of mercury. We've already discussed that. So this will approximately be 3000 or maybe 2990. So what is going to be my maximum? This is now 9 ml. Right? So you have to multiply 3000 into 0.003, which gives you 9 ml per 100 ml of blood. Right? 9 ml per 100 ml of blood. So this is your, what is the dissolved oxygen. Let's discuss one AIMS question related to this topic. Yes, let's discuss this AIMS question. What does this question say? Let's see this. Now this question says that there is an intern. Yes, I think most of you must be interns. Yes. So there is an intern who takes a blood sample and he finds that the PO2 is 80 millimeters of mercury. The question says, how much is the dissolved oxygen per ml of blood? Is it 0.24? Is it 0.024? Is it 0.0024 or 2.4? Bolo. You know, this is, let me see, Mysore, Gulbarga, Hyderabad, Aligarh, Ludhiana, Srinagar, Mumbai. All of you, I don't know if you remember this, your moms have always been telling you right from the time you started school that question ko dhyan se padhna. And that is exactly what is applicable today itself. Please please read the question very, very carefully. Mumbai, Mysore, all wrong. Ludhiana, wrong. Meerut, wrong. Right? So see this very carefully. It says PO2 is 80 millimeters of mercury. How much is the dissolved oxygen per ml of blood. Per 100 ml was 0.24. So per ml of blood becomes 0.0024. Is it clear? Okay, Miraj, no. Miraj, Agartala got it, Srinagar got it, Karamsad got it, right? Now, of course, everybody will get it. Yes? Please remember, please read the question carefully. What your moms has been telling you right from school day onwards is even more important now, right? Read the question. If it was per 100 ml, it would have been 0.24. That was the easier one. But it says per ml of blood. So you need to divide this value 0.24 by another 100 ml. So that makes it 0.0024 ml per ml of blood, right? So this is your Belgam says, is amount of dissolved oxygen influenced by any other factor? Which are the factors which can influence the dissolved O2? Is primarily, what is the alveolar O2? Henry's law. What is the thickness of the respiratory membrane? Supposing there is some problem in diffusion. Supposing there is, let's say, lung fibrosis, let's say interstitial lung disease. So thickness of the respiratory membrane has increased. In that case, now the PO2 will be different. Or if there is a VQ mismatch, that can also give rise to a decrease in PO2. Or if there is a shunting. Have you understood? So this will give rise to Belgam. The effective pH is on the oxygen which is combined with hemoglobin, not on the dissolved O2. Have you understood? So if you look at it, dissolved O2 will depend only on these factors. What is the alveolar O2? What is the diffusion? What is the state of the respiratory membrane? Thickness of the respiratory membrane? Are there any VQ mismatches? Is there a shunting? Yes. So that will give us our O2, right? So this is your O2. Now next, let's see oxygen hemoglobin dissociation curve. Now, what is this oxygen hemoglobin dissociation curve? For this curve, now we've got PO2 on the x-axis. PO2 is in millimeters of mercury on the x-axis and SO2 is on the y-axis. This is the percentage, right? Percentage. So this is the oxygen hemoglobin dissociation curve. PO2 on the x-axis and the SO2 on the Mohinder? PO2 on the x-axis and SO2 on the y-axis, right? PO2 on the x-axis and SO2 on the y-axis. So let's take some values which you already know, right? Values which you already know. Okay. Like for instance, I told you PO2, SO2, arterial blood we have already done. PO2 in millimeters of mercury is 95. The saturation is 97 to 98%. Venous blood, the PO2 is 40 and the saturation is 75%. There is another value which we need to understand, and that is a PO2 of 60 corresponds to a saturation of 90%, right? This is PO2 and SO2. We've already discussed what is SO2 and what is PO2. PO2 is the dissolved oxygen. SO2 is the saturation of the hemoglobin with the oxygen, right? So these are three values which are very important for us. And why is this value very important? Because please understand, an arterial oxygen tension less than 60% or an arterial saturation less than arterial oxygen tension less than 60 millimeters of mercury or an arterial saturation less than 90% is taken as hypoxic hypoxia. Have you got it? So this value becomes very important. See, if you've got a pulse oximeter And the pulse oximeter shows a value of 85. That means arterial saturation is 85%. That means your patient is severely hypoxic. On the pulse oximeter, the saturation should be more than 90. If it becomes less than 90, means the PO2 is less than 60, which is a severe hypoxia. Have you got this point? Right? So PO2 and SO2. So you've got these three values, and let's plot them on the graph. Let's see on the graph now. This is 50. This is 100. This is 50. This is 100. Right? So now when the PO2 is 95, when the PO2 is 95, when the PO2 is 95, the percentage saturation is 97 to 98%. This is your arterial blood. And when the PO2 is 40, this is 40, 10, 20, 30, 40, percentage saturation is 75. And when the PO2 is 60, 60, 70, 80, 90. So when the PO2 is 60, percentage saturation is 90. So at least you've got these 3 points on the graph. And again, similar, this is your normal physiological range. Arterial blood and venous blood, this is what it varies between. Now, then at different PO2s, we took under experimental conditions, we measured what is the saturation. So here we now get a sigmoid shaped curve. This is a sigmoid shaped curve. And what is this? That means it is like the Greek letter sigma. Sigma is like this, right? Greek letter sigma. So it's a sigmoid shaped curve. And what is the sigmoid shaped curve due to? This is due to the phenomena of positive cooperativity. And what is positive cooperativity? You know that each hemoglobin has got 4 heme. Binding of the first heme with oxygen increases the affinity of the second, which increases the affinity of the third, which increases the affinity of the fourth. So you've got these. So this is known as positive cooperativity. Most difficult or least affinity is of the first heme for oxygen. Then as oxygen keeps on adding to the hemoglobin, the affinity for oxygen increases. That is called the positive cooperativity. In fact, positive cooperativity is defined on the basis of Hill's coefficient. Hill's coefficient is used to define this cooperativity, Hill's coefficient. When Hill's coefficient is equal to 0, it means no cooperativity. When Hill's coefficient is more than 1, it is a positive cooperativity. When Hill's coefficient is less than 1, this is a negative cooperativity. No cooperativity. When Hill's coefficient is equal to 1, this is a no cooperativity. More than 1 is a positive cooperativity. Less than 1 is a negative cooperativity. Right? So this is Hill's coefficient. Hill's coefficient for the combination of heme and oxygen. Hill's coefficient is equal to 3. That means this is a positive cooperativity. And I've already discussed it with you. What is positive cooperativity? It means that the that binding of the first heme increases the affinity of the second, which increases the affinity of the third and fourth. So Hill's coefficient for hemoglobin and oxygen is 3. Right? That means there is a positive cooperativity. Hill's coefficient equal to 1, no cooperativity. More than 1, positive. Less than 1, negative. Right? Now let's see what is P50. What is P50? And P50 is the partial pressure at which hemoglobin is 50% saturated. This is the partial pressure at which hemoglobin is 50% saturated. This is P50. Partial pressure at which hemoglobin is 50% saturated. This is 23 to 25 millimeters of mercury. This is the partial pressure at which hemoglobin is 50% saturated. Please please understand, Dehradun, myoglobin, only one molecule of myoglobin combines with one molecule of oxygen. I'm coming to myoglobin, right? 1 second. Just give me some time. So this is P50. P50, what is P50? This is the partial pressure at which hemoglobin is 50% saturated, and the normal value of P50 is 23 to 25 millimeters of mercury, right? And this is also equal to 3.6 kilopascals. 3.6 kilopascals, right? This has been asked as a question in terms of kilopascals, yes? And please remember, 1 kilopascal, you can write it in some corner somewhere, 1 kilopascal is equal to 7.5 millimeters of mercury. 1 kilopascal is 7.5 millimeters of mercury. Are we okay with that? Right? So this is your sigmoid shaped curve. This is your normal. Now let's see if the curve shifts to the right. Supposing there is a rightward shift of the curve, what will happen to P50? Increases or decreases? Bolo. If there is a rightward shift of the curve, is there an increase in P50 or a decrease in P50? Increase, isn't it? The P50 now increases. The P50 is now this much. Money? Increase in P50 money, there is a decrease in affinity. Why decrease in affinity? Because the partial pressure at which hemoglobin is now 50% saturated has increased, has become more. So there is a decrease in affinity. Similarly, if the curve is shifted to the left, if the curve is shifted to the left, this means there is a decrease in if the curve is shifted to the left, it means there is if the curve, this was rightward shift of the curve, so I said increase in P50, but supposing there is a leftward shift of the curve would mean a decrease in P50 and an increase in affinity. Next, let's see what are the factors which cause a shift of the curve to the right and shift of the curve to the left. Let's see this. Shift of the oxygen hemoglobin dissociation curve, oxygen hemoglobin dissociation curve to the right. Shift of the oxygen hemoglobin dissociation curve to the right. This, to remember this, please remember FDR. Rightward shift means increase in P50 and a decrease in affinity. And to remember this, remember FDR. What does FDR stand for? It says favors delivery. A shift to right. It decreases affinity, therefore it increases the delivery. Right? Decrease in affinity means there is an increase in delivery. So decrease in affinity, shift to right. The conditions which cause a shift to right are increase in PCO2, increase in H+, decrease in pH, increase in temperature, increase in 2,3-DPG and hemoglobin S. PCO2, H+, pH, temperature, 2,3-DPG and hemoglobin S. Now what are the conditions which will cause an increase in 2,3-DPG? 2,3-DPG is increased, the mnemonic is GITA. GITA here stands for growth hormone. Increase in growth hormone increases 2,3-DPG. Exercise increases 2,3-DPG. Increase in thyroid hormones increases 2,3-DPG. Anemia, high altitude and androgens. These are the conditions which increase the 2,3-DPG. Growth hormone, exercise, thyroid hormone, anemia, altitude, androgens. Right? So these are the conditions which cause a shift of the curve to the right. Next, let's see what happens if the curve is shifted to the left. So let's see shift of oxygen hemoglobin dissociation curve to the left. And leftward shift means there is a decrease in P50 or an increase in affinity. And this can be remembered with the help of this mnemonic FAL. What is FAL? Favors affinity. a shift to left. Favors affinity, a shift to left. Right? Favors affinity, a shift to left. And which are the conditions which favor affinity? If there is a decrease in PCO2, decrease in H+, increase in pH, hypothermia, decrease in 2,3-DPG, hemoglobin F, myoglobin, and banked blood. Banked blood means stored blood. Right? In all these conditions, there is a shift of the curve to the left. Shift of the curve to the left. Now let's try and understand why is there a shift of the curve to the left. See, in banked blood, why is there a shift to the left? In banked blood, glycolysis will be inhibited. You're storing the blood at low temperature. You're storing it at 4 degrees Celsius. So the glycolysis is inhibited. So this decreases the 2,3-DPG. And if this decreases the 2,3-DPG, what will happen to the curve? Curve shifts to the left. Clear? Next, hemoglobin F. Now why hemoglobin F? See, hemoglobin F is alpha 2 gamma 2. Now the gamma chain of HbF, this has a lower affinity for 2,3-DPG and a higher affinity for oxygen. Please understand, 2,3-DPG and oxygen bind at the same place. So if the HbF has a lower affinity for 2,3-DPG, it will have a higher affinity for oxygen. They're both binding at the same place. But the point to remember is that in the case of 1 mole of oxygen combines with 4 moles of oxygen combine with 1 mole of hemoglobin. But in the case of 1 mole of oxygen combines with 4 moles of oxygen combined with 1 mole of hemoglobin. But in the case of 2,3-DPG, it is 1 mole of 2,3-DPG combines with 1 mole of hemoglobin. Have you got this? Right? So this is your, the 2,3-DPG and oxygen are binding at the same place, but there's a difference. 4 molecules of oxygen bind with 1 molecule of hemoglobin, but 1 molecule of 2,3-DPG binds with 1 molecule of hemoglobin. So, right? On the other hand, if you see these curves, the myoglobin and oxygen dissociation curve and the HbF and the oxygen dissociation curve, let's see them. Right? Here you are. If this is a normal oxygen hemoglobin dissociation curve, the myoglobin and the HbF and the oxygen dissociation curve, there is a shift to the left. This is the HbF curve. And if you look at the myoglobin and the oxygen dissociation curve, it's a parabola. It's an extreme shift to the left. This is the myoglobin and the oxygen dissociation curve. Right? And what is the unique feature of myoglobin? 1 molecule of myoglobin combines with 1 molecule of oxygen. And please remember, myoglobin is present. We've done it in the morning. The types of muscle fibers which have got myoglobin. Myoglobin is present in your type 1 muscle fibers, oxidative metabolism, isn't it? And it is a storehouse of oxygen. And this myoglobin will surrender its oxygen only when the oxygen in the tissues, in the skeletal muscles is very, very low. And this is also asked as a question. Myoglobin will surrender its oxygen. Myoglobin gives up its oxygen only when the PO2 in the muscles is less than PO2 in the muscles is less than 1 kilopascal, 1 kilopascal or less than 5 mm of Hg, less than 1 kilopascal or less than 5 mm of Hg. Right? These are your, of course we know that 1 kilopascal is 7.5, but it has to be approximately 0.75 kilopascals or less than 5 millimeters of mercury. So this is this is your myoglobin oxygen dissociation curve and the HbF oxygen dissociation curves. Now let's see what happens in the case of carbon monoxide poisoning. What happens in carbon monoxide poisoning? Now let's see this. This is a normal oxygen hemoglobin dissociation curve. Now there are two curves here. Both are showing a reduction in the height. This is curve A, this is curve B. And reduction in height means the SO2 has decreased, saturation has decreased. Now I'm trying to ask you which curve. This is oxygen hemoglobin dissociation curve. Yeah. Arey Baba Jammu, 7.5 millimeters of mercury is the exact value, right? But when you look at in terms of kPa, when you look at the graph, it looks as though it is less than 1 kPa, right? But in terms of millimeters of mercury, better answer becomes 5 millimeters. Yes, 7.5 is 1 kPa, right? But just in terms of better answers, so to speak, is your 5 millimeters of mercury. Okay. Now let's see this A and B. Which graph represents carbon monoxide poisoning? Which graph represents carbon monoxide poisoning? A or B? A, isn't it? This is your carbon monoxide poisoning curve. Now what has happened is, please understand, in the presence of carbon monoxide poisoning, we have already discussed that there is a decrease in the SO2. So the height of the curve is reduced. But please remember, in the presence of carbon monoxide, the oxygen which is present is now tightly bound. See, how is the saturation reduced? We have already said that carbon monoxide poisoning, carbon monoxide forms an almost irreversible bond with heme. So this heme is not available to combine with oxygen. So saturation reduced, and that is seen by the decrease in the height of the curve. Plus, please remember, the oxygen which is present is also now tightly bound. The oxygen which is present is also now tightly bound. So carbon monoxide, because of the presence of carbon monoxide, there is a tight binding of oxygen with hemoglobin. So there is a shift of the oxygen hemoglobin dissociation curve to the left. So A curve represents carbon monoxide poisoning. Chaliye. Next, let's see what do you understand by the Bohr's effect. What is the Bohr's effect? Bohr's effect. Yes. Bohr's effect is the shift of the oxygen hemoglobin dissociation curve based on these PCO2 levels, right? Based on PCO2 levels, right? For example, in the tissues. Now, in the tissues, there is increase in PCO2. Obviously, tissues are going to oxygen, the PCO2 levels, there is increase in PCO2 in the tissues because obviously oxygen, because of metabolism. Increase in PCO2 now shifts the oxygen hemoglobin dissociation curve to the right. And when there is a shift to the right, And remember, I told you favors delivery shift to right. So this now causes increased delivery of oxygen to the tissues, and what you have described, Dehradun, as an unloading of oxygen. So this is unloaded. Oxygen, there is more delivery of oxygen to the tissues. Increased delivery of the oxygen to the tissues because the curve shifts to the right. Similarly, in the case of the lungs. Now, in the lungs, the carbon dioxide has moved from the blood to the alveoli, isn't it? So, here you have the movement of CO2 in this fashion. So, there is a decrease in the PCO2 in the blood. And when there is a decrease in PCO2, it shifts the oxygen hemoglobin dissociation curve to the left. And we have already discussed shifting to the left means it favors affinity. There is increased affinity of heme and oxygen in the lungs. Yes, so there is increased oxygen affinity. So that means there is more loading of oxygen in the lungs, unloading in the tissues, Dehradun, and loading in the lungs. This is your Bohr's effect. Now, please understand the movement. This is an important point to consider, and that is the movement of oxygen in the lungs or in the tissues. Movement of oxygen in the lungs means from the lungs to the blood, and in the tissues from the blood to the tissues. The movement of oxygen is primarily because there is a difference in the partial pressure of oxygen. Bohr's effect, please understand, Bohr's effect is responsible for an additional 1 to 2% of the oxygen delivery to the tissues. 1 to 2% of the oxygen unloading and 1 to 2% of the oxygen loading. That's it. So that means if I ask you a question which says, what is the major mechanism of oxygen delivery to the tissues? Is it because of a difference in partial pressure? Or B, is it because of the Bohr's effect? Then my answer becomes, obviously, the difference in partial pressure. Have you understood? Bohr's effect is responsible only for an additional 1 to 2%, additional 2% of the oxygen delivery. That's about it. 1 to 2% of the oxygen delivery to the tissues. This is your Bohr's effect. Clear? चलो. इसके बाद आ जाओ CO2 transport पर. We've done oxygen transport. Now let's discuss the CO2 transport. Now what is the CO2 transport? Now CO2 transport occurs in 3 ways. Number 1, as bicarbonates. This is the maximum. Maximum is in the form of bicarbonates. Number 2, it combines with hemoglobin to form carb amino compounds. Carb amino compounds, right? This is your CO2 transport. So as bicarbonates and with hemoglobin to form carb amino compounds. And number 3, as the dissolved carbon dioxide. As dissolved carbon dioxide. Physical solution of carbon dioxide in the plasma. So as bicarbonates with hemoglobin to form carb amino compounds and as dissolved carbon dioxide. Right? Now, if I look at the total CO2 in the arterial blood and the venous blood. So let's look at this. So you've understood this. This is an important, frequently asked question. What is the maximum CO2? The maximum transport of CO2 is in the form of bicarbonates. Now let's see arterial blood and the venous blood. Arterial blood and the venous blood. The PCO2 in the arterial blood is 40 mm of Hg. In the venous blood, it is 45 mm of Hg. Please understand, this is PCO2 I'm talking about, right? Now, if I look at the total CO2 in ml per deciliter, in the arterial blood, the total CO2 is 49 ml per deciliter. 49 ml in the arterial blood. Now, as the blood goes through the tissues, 3.7 ml is added to 100 ml of blood. 3.7 ml of CO2 is added to 100 ml of blood. Mane venous PCO2 will now become 52.7 ml per deciliter of blood. Isn't it? Have you understood this? 49 ml per deciliter is the total CO2 content. 3.7 ml is added as the blood flow through the tissues. So 49 plus 3.7 becomes approximately, or it in fact becomes 52.7 ml per deciliter of blood. Are you able to appreciate this? Yes. Arterial PCO2, dissolved partial pressure of CO2 in the dissolved state is 40 mm. Venous blood 45. This is the total CO2 content. On the other hand, if I compare oxygen levels, I told you PO2 is 95 millimeters of mercury in the arterial blood and 40 millimeters of mercury in the venous blood. We've already done that. But the total amount of CO2 which is carried, total O2 which is carried, of course this will depend upon the hemoglobin. Yes, because you know that already. But let's say take an average hemoglobin of 14. In that case, I get an arterial PO2 of 19 ml per 100 ml of blood and 14 ml per 100 ml of blood. That means the body has used 4 ml of oxygen from 100 ml of blood. Have you understood this point? Right? O2 5 ml has been used up. CO2 3.7 ml has been produced. You remember the RQ? RQ was volume of CO2 produced divided by the volume of oxygen consumed, which gave us an average RQ of 0.8. 3.7 divided by 5 gives us an average RQ of 0.8. Have you got this? So these are important values to remember, right? 19 ml per deciliter of blood and 14 ml per deciliter of blood. Okay. Next, let's see what do you understand by Haldane's effect. What is Haldane's effect? Now, Haldane's effect is based on, this is involved in CO2 transport. Haldane's effect. And what is Haldane's effect? It says that Haldane's effect is based on the fact that deoxyhemoglobin can bind more CO2 than oxyhemoglobin. Deoxyhemoglobin can bind more CO2 than oxyhemoglobin, and makes a lot of sense again. Blood goes to the tissues, deoxyhemoglobin is formed, and deoxyhemoglobin can bind more CO2. Loading of CO2 will be more. Blood goes to the lungs, oxyhemoglobin is formed, unloading of CO2. Have you got this point? So this is Haldane's effect. This is based on the fact that deoxyhemoglobin can bind more CO2 than oxyhemoglobin. So deoxyhemoglobin is what you get in the tissues, and oxyhemoglobin is what you get in the lungs. And again, this is a helpful effect. This is called Haldane's effect. चलिए आगे देखें. Let's now go on to what is known as regulation of respiration. Regulation of respiration. Regulation of respiration. Visakhapatnam, sorry, what does it say? See, what was respiratory quotient? It was volume of CO2 produced divided by the volume of oxygen consumed. So you're adding 3.7 ml of oxygen to 100 ml of blood, and you've used 5 ml of oxygen. I'm just talking about normal conditions. So 3.7 divided by 5 is approximately 0.8. So this will become your, remember I told you, if on a mixed diet, your RQ is approximately 0.8, and that's exactly what I'm trying to tell you. Okay? Now, regulation of respiration. Now, regulation of respiration, there are two major mechanisms for regulation of respiration. Number one is a neural mechanism and the other is the chemical mechanism. Neural and chemical. Chloride shift, Agroha. Okay. Okay. Let me describe chloride shift, what Agroha is asking me. Yes, it will take 2 minutes. Let me just describe it for you. Now, what is chloride shift? Right? Chloride shift. Now, chloride shift is what you can see in the venous RBC. Venous RBC. Now, this is your RBC here. Yes. Now, in the venous RBC, CO2 plus water forms H2CO3, and this is in presence of an enzyme which is called carbonic anhydrase. Carbonic anhydrase. Please remember, RBCs are very rich in carbonic anhydrase, right? And carbonic anhydrase, like I said, it catalyzes this reaction between CO2 and H2O to form H2CO3. Now, this H2CO3, this splits to form H plus HCO3 minus. Now, this H plus, the H plus is buffered by the hemoglobin. The H plus attaches to the hemoglobin. So, it's buffered by the hemoglobin. But this bicarbonate which is generated, now this bicarbonate leaves the cell, leaves the RBC, right? It leaves the RBC, and to maintain electrical neutrality, you have the movement of chloride from outside. Bicarbonate moves out of the cell, comes into the plasma, and to take its place, chloride goes into the RBCs, and this exchange of bicarbonate and chloride, this occurs with the help of a special protein which is called anion exchanger 1. Anion exchanger 1. What is anion exchanger 1? This is a protein which is responsible for a mutual exchange of anions. Mutual exchange of anions based on their concentration gradients. Based on their concentration gradients. Right? There is a mutual exchange of anions based on their concentration gradients. This is anion exchanger 1. And yes, Mumbai, this or Meerut, sorry, this was earlier known as the band 3 protein. This was earlier known as the band 3 protein, but today we call it, our anion exchanger 1. Anion exchanger 1. Right? So this is your, this is responsible for the, this is what is known as chloride shift. Now see, chloride is osmotically active, right? Chloride is osmotically active. So when chloride enters into the RBC, water also enters into the RBC. So what do I find? I find that the venous RBC is slightly larger. Venous RBC is slightly larger than the arterial RBC. You know that venous hematocrit is more than the arterial hematocrit. And makes a lot of sense now. Why is it more than the arterial hematocrit? Venous hematocrit is more than the arterial hematocrit. Why? Because the venous RBC is larger than the arterial RBC, almost a 2% larger than the arterial hematocrit. Right? This is as far as your, which is going to be Mysore, which is going to be a better buffer, has to be a deoxyhemoglobin. Na? deoxyhemoglobin will be a better buffer. So there is now an increase in the size of the venous RBC because of chloride shift. This chloride shift is also called the hamburger effect. Because now the venous RBC has become like a burger patty. So this is called your hamburger effect, right? Okay, so now let's go on to our important topic of respiratory system, and that is regulation of respiration. Now, regulation of respiration में you have a neural mechanism for the regulation of respiration. Neural mechanism for the regulation of respiration. There are different centers. Neural mechanism में there are different centers which are involved in the regulation of respiration. This is along with chloride, water will also come. Okay. So, neural control of respiration. You've got some centers in the medulla. Medulla has, number one, medulla has the pacemaker of respiration, and the pacemaker of respiration is the Pre-Potzinger complex. Pre-Potzinger complex. This is the pacemaker of respiration, and this has been asked as a question. What is the pacemaker of respiration? This is called the Pre-Potzinger complex. Manipal, what is the problem? Manipal, see, bicarbonate is moved out, but chloride is moved in, na? So the chloride is osmotically active. Bicarbonate is moved out, so what? But the cell has gained one osmotically active particle. Bicarbonate is moved out, but chloride is moved in. So that is gaining an osmotically, CO2 was not osmotically active, but the moment CO2 combines with water, you got H2CO3, which splits into water and which splits into HCO3 and H plus. H plus got buffered. Bicarbonate was osmotically active. It moves out, but it is replaced by another osmotically active substance, which is chloride. Right? So that is your so that should take care of Manipal and Dehradun, Mumbai. Are you okay with that? Right? That is what is responsible for your chloride shift. Chloride shift is responsible, right? Okay. So let's go back to the neural control of respiration. So I said medulla. Medulla has got the pacemaker, which is the Pre-Potzinger complex, right? And medulla also has two more areas, and this is dorsal respiratory group of neurons and ventral respiratory group of neurons. Dorsal respiratory group of neurons and ventral respiratory group of neurons, right? Now what has happened in what is dorsal respiratory group of neurons? This has got only the I neurons. Meaning it can send impulses only for inspiration. Whereas the VRG has got I as well as E neurons. It can send impulses for both inspiration and expiration, right? Now, DRG functions in normal quiet respiration, whereas this functions whenever the requirement increases. Whenever the requirement increases, DRG functions whenever in normal quiet respiration, whereas VRG functions whenever the requirement increases. This is your DRG and VRG. Yes. See, basically, what is the DRG doing? DRG is your respiratory center in the medulla. DRG will send impulses to the phrenic nerve nucleus. After all, which is the main muscle of respiration, is your diaphragm. So DRG sends impulses to the phrenic nerve nucleus. Phrenic nerve nucleus is in the cervical spinal cord. And from the phrenic nerve nucleus, the impulses will go to the diaphragm. Diaphragm moves downwards. Inspiration. During expiration, there are no impulses from DRG. Why? Because DRG has got only I neurons. So when there are no impulses during expiration, the diaphragm relaxes, moves upwards, and expiration. So this is how but whenever the requirement increases, for example, when does the requirement increase? For example, during exercise. Now the VRG will take over. VRG will send impulses for both inspiration as well as expiration, right? So VRG functions whenever the requirement increases. These are your medullary centers. Medullary centers, like I told you, there is the pacemaker and there is DRG and VRG. Next, you have some centers in the pons. You have some centers in the pons. In the lower pons, right? Lower pons, you've got the apneustic center. And in the upper pons, you've got the pneumotaxic center. Lower pons are the apneustic center, upper pons is pneumotaxic center. Merit, pre-Botzinger complex is the pacemaker. Now, there are some authors believe that pre-Botzinger complex and DRG are two different things. But some authors say pre-Botzinger, there's nothing known as pre-Botzinger, it is the same as DRG. Whatever it is, you need to understand, you get an MCQ which has already come once, and that is pacemaker of respiration. Kaun hai? You should be able to tell us that this is the pre-Botzinger complex. Is it clear? Some people have said pre-Botzinger and DRG is the same thing. Have you understood? VRG, please understand, will take over only when the requirement increases, right? So this is as far as then the pneumotaxic center. Now pneumotaxic center, pontine centers. Pontine centers are the pneumotaxic and the apneustic center. Pneumotaxic and the apneustic. Apneustic center is in the lower pons, pneumotaxic is in the upper pons, right? Now let us see what is the effect of these on respiration. And to remember this, what is the effect of each of these on respiration? Please write inspiration here, right? And when I talk in terms of inspiration, please understand this is controlling the depth of inspiration. Now write that vagus inhibits inspiration, depth of inspiration. Pneumotaxic center decreases the depth of inspiration, but apneustic center strongly stimulates the inspiration, right? Strongly stimulates. There are two pluses, one minus for pneumotaxic, one minus for vagus. Yes? Then number 3, you also have the higher centers. Higher centers, you know that voluntarily also, the higher centers will include the cerebral cortex. Cerebral cortex, you know this, na? Voluntarily also you can increase the rate and depth of ventilation up to a certain limit, of course, but I can ask you to change the rate as well as the depth of ventilation. So higher centers do have a role in playing in regulating the respiration. So these are your 3 neural areas. This is your neural control of respiration. The first one which I did with you was medulla, which has got the pacemaker for respiration, pre-Botzinger complex. Then I said the medulla also has the DRG and the VRG. DRG functions in normal quiet respiration. This takes over whenever the requirement increases. Pons, lower pons, apneustic center, upper pons, pneumotaxic center. What is the effect of these? Inspiration, vagus inhibits, pneumotaxic center inhibits, apneustic center strongly stimulates, and plus you have the higher centers. So this is your basic information about the neural regulation of respiration. Now what I'm going to do with you is, I'm going to neural control and regulation of respiration. The next thing which I'm going to do with you is questions, so that this topic is completely reinforced. So not to worry, we will do all possible questions which can come from this topic. Okay? So let's take the first one. First one, I said, if I do a vagal stimulation, what will happen to the depth of ventilation? Vagal stimulation, what will happen to the depth of ventilation? Yes, this will decrease. Remember, I told you vagus inhibits the inspiration, right? We've discussed this already. We said that the vagus inhibits the depth of inspiration. So vagal stimulation kiya, so it will cause a decrease in the depth of ventilation. So there is a decrease in depth. And if the depth decreases, So just hang on for 5 minutes. Let me explain this to you. Now, vagal stimulation is decreased depth. Now, if there is a decrease in depth, if the depth decreases, what will happen to the rate? If depth decreases, the rate will automatically increase. But please understand, the primary effect is on the depth. Effect on the rate is a secondary effect. In other words, this gives rise to what is known as a rapid, shallow respiration. Rapid, shallow respiration. Next, let's see. If I do a strong vagal stimulation, if I strongly stimulate the vagus, this can give rise to apnea. Apnea means a complete cessation of respiration. There is a cessation of respiration. Respiration stops because now this is respiration stops. This is complete cessation of respiration. Strong vagal stimulation causes apnea. There is a cessation of respiration. Next question. Supposing I do a bilateral vagotomy. Now what will happen to the depth? If I do a bilateral vagotomy, what will happen to the depth? Let's see this. Bilateral vagotomy matlab, ab now this, the effect of vagus is gone. 1 minus is gone. So you've got 2 pluses of apneustic center, 1 minus of pneumotaxic center. So what is going to happen to my depth? Depth increases. And if the depth increases, what will happen to the rate? If the depth increases, what will happen to the rate? The rate decreases. Please understand, primary effect is on the depth. Rate is a secondary effect. In other words, this is a slow, deep respiration. Next question. You know that this is the spinal cord, this is the medulla here, this is the pons here, these are your higher centers. Okay? We already know that the respiratory center, which is in the medulla, this is the respiratory center here. This respiratory center sends impulses to the cervical spinal cord and then the contraction of diaphragm and so on. Right? Now, supposing I do a section at the upper border of the pons. Upper border of the pons. Now, the higher centers cannot affect the respiratory center. So, section at the upper border of pons means there is loss of voluntary control. There is loss of voluntary control. Makes sense now? Because now the higher centers can no longer send impulses to the respiratory center. They can no longer influence the respiratory center. So there is loss of voluntary control. Upper border of pons. If the section is at upper border of pons, there is a loss of voluntary control. Clear? Next. Let's see the next one. Again, spinal cord, medulla, pons, higher centers. Now, you know in the lower pons is the apneustic center, upper pons is the pneumotaxic center. Now, supposing I do a section here, a mid-pontine section. And if you remember, we wrote a little while back. Now, mid-pontine section मतलब कौन से areas है जो respiration को influence नहीं कर पाएगा? This is cerebral cortex and the pneumotaxic center. Both cannot affect the respiratory center. Where is the respiratory center? In the medulla. Right? Both these areas cannot affect the respiratory center. So remember what we wrote for this? We said inspiration, vagus inhibits, pneumotaxic center inhibits, apneustic center strongly stimulates. Mane, when I do a midsection of pons, pneumotaxic center influence is gone. Vagus is intact. Again, what will happen now? This increases the depth and therefore decreases the rate. Primary effect on the depth. So this is a slow, deep respiration. Slow, deep respiration. Right? Pneumotaxic influence is gone. Vagus is present. two pluses of apneustic center, so increase in depth and a decrease in rate. So slow, deep respiration, right? Next is, supposing I do a mid-pontine section. Mid-pontine section with a bilateral vagotomy. Mane, pons is gone and vagus is gone. Now what will happen? Now it's only apneustic center which is acting. And apneustic center, what is the effect on apneustic center? What is the effect of apneustic center? It strongly stimulates. There are no effect. ऐसे कैसे? Apneustic center तो intact है ना? Right? Apneustic center तो present है. ये देखिए. ये apneustic is present. Right? So what do I get? The pattern of breathing is known as apneusis. And what is apneusis? Apneusis is like this. There are prolonged inspiratory spasms. In fact, prolonged inspiratory spasms, because there is no negative control now. Neither is there the vagus, inhibitory control of vagus is gone, inhibitory control of pneumotaxic center is gone. So it's prolonged inspiratory. In fact, see, the name apneustic center was given after we first discovered the pattern of respiration. We saw that some patients of injury, head injury or whatever, they had this particular, maybe tumors or whatever, they had this particular type of breathing. Then on post-mortem, it was seen that the lesion there was a mid-pontine section with influence of vagus was also gone. So the pattern of this type of breathing was called apneustic breathing. They named the pattern first. Then they replicated this lesion in animals. And they found that there must be some center in the lower pons which is producing this type of a stimulation of respiration, right? So it was the pattern of breathing was discovered first. Then we discovered what is the lesion. Then we named the center in the lower pons as apneustic center. Have you got that? Right? पहले हमने breathing देखा patients में. फिर हमने realize किया this is because of section in mid pons with a bilateral vagotomy. And पहले breathing का नाम दिया, फिर देखा ये section है कहाँ पर. Then we said there must be some sort of a center in lower pons which is stimulating the respiration. So this is your mid-pontine section with bilateral vagotomy. This produces a pattern of breathing known as apneusis. Apneusis. ठीक है? Apneusis. ठीक है? Next question. Now supposing, this is your spinal cord here, medulla here, pons here, higher centers here. Now supposing you of course know that the medulla is where your respiratory center is. In fact, all the important centers are in the medulla, right? Now supposing I do a section at the upper border of medulla. माने ना तो cerebral cortex का influence है, ना pontine centers. Both of these cannot affect the medulla now. So that means now let us see what is the type of respiration which is produced. In this situation, rhythm of respiration is present. Rhythm of respiration is present, but it is an irregular and a jerky kind of respiration. It is highly irregular respiration. Highly irregular. How come? Let's say, now, let us see what happens with vagotomy and without vagotomy. That means if vagus itself has also been removed, then what is the pattern of respiration that we see? It is a highly irregular and gasping kind of respiration. Jerky ki jagah, the better term to use is a gasping kind of respiration. There is irregular and with gasps in between. You can see this. It's highly irregular. Sometimes the rate is more, sometimes the depth is more, and there are gasps. deep gasps. But if vagus is present, it continues to be irregular. But if you can understand, these kind of deep gasps are not seen. Though it is still irregular and gasping, but the gasps are much shallower as compared to when vagus was also removed. This is if the section is at upper border of medulla. Upper border of medulla. Rhythm of respiration is present, but it is irregular and a gasping type of respiration. Irregular and gasping type of respiration. Yes, I'm repeating all of these. Chalo, see what are the questions that I've done with you so that this topic is completely reinforced. Let's do the first question. First, I said what happens when you do a vagal stimulation. Vagus, remember, inhibits inspiration. So we said it is a decrease in depth and therefore an increase in rate. Rapid, shallow respiration. When I do a strong vagal stimulation, vagus inhibits inspiration. जब बहुत strongly आपने vagus को inhibit किया, it completely suppresses the eye neurons. So there is now apnea, cessation of respiration. This came last year, All India PG. If I do a bilateral vagotomy kiya, one negative influence is removed. So there is an increase in depth and a decrease in rate. And why increase in depth? Because if you remember the little thing which we made, inspiration, apneustic center strongly stimulates. Pneumotaxic center was 1 minus, vagus was 1 minus. So 1 minus is removed. So it becomes a slow, deep type of respiration. Next question, supposing I do a section at the upper border of pons. Upper border of pons. Yes, what happens in upper border of pons? Here there is a loss of voluntary control. Loss of voluntary control. What happens in loss of voluntary control is, now that the cerebral cortex is no longer able to affect the respiration, right? So this is loss of voluntary control. This is what you see in decerebration, right? There is no voluntary control. Next, let's see. Next, this is midsection of pons. Midsection of pons without midsection of pons without vagotomy. If vagus is intact, then there is increase in depth, decrease in rate. Mid-pontine section with bilateral vagotomy gives us an apneustic type of breathing, which is prolonged inspiratory spasms. Then when I do a section at the upper border of medulla, now the rhythm of respiration is present, but this is irregular and gasping. And with vagotomy, very deep, with vagotomy, deep gasps are seen. Without vagotomy, it continues to be gasping, but those deep gasps are not seen. See what happens, Eradun, in Kussmaul's? Kussmaul's is what you see in metabolic diabetic ketoacidosis, isn't it? Acidotic breathing. Now, what is acidotic breathing? I'll do it with you when we do chemoreceptors. Similarly, what is Kind-Stokes is also better would be able to explain once we do chemical control of respiration, right? Now, what is Ondine's curse, Jammu? See, Ondine's curse, now what is this? Ondine was a Raja. Okay? So this is a kahani that you have to listen to this. Now, what is Ondine's curse? He was a Raja and his daughter, so he cursed his daughter's boyfriend. And he told him, see, this is what fathers can do, so be careful. He cursed the daughter's boyfriend and he said that, as Ondine was this Raja, and he cursed him and he said that as long as this boyfriend was awake, as long as he was awake, there would be voluntary control of respiration. But the moment he sleeps off, he will not, his involuntary control, he damaged the involuntary control of his respiration. In other words, that means as long as he is awake, he's alive, he'll be able to breathe. But the moment he goes off to sleep, voluntary control is gone, and now his involuntary should have taken over, but involuntary he cursed that his involuntary respiration is gone. So probably, now this is a kahani. It's a story, right? Now, probably the lesion must have been in the pre-Potzinger complex, right? Some curse has gone and hit the pre-Potzinger complex, right? That means his voluntarily, as long as he was able to breathe, as long as he was awake, he's able to breathe. But the moment he sleeps, his breathing is lost, right? So that is your on-nines curse. Okay, so my last question related to this, I still haven't finished this. My last question related to this topic was, which was asked last time. Apnea will be seen if section is at. Apnea is seen if section is at. Is it, if it is at upper medulla, upper pons, lower pons, upper medulla, lower medulla? What is your answer? A, B, C, or D? Lower medulla, isn't it? Complete cessation of respiration will be seen if the section is at lower medulla. Clear? So these are all your questions. We've tried to reinforce this neural control. Go through these questions. It will not be such a big problem. Okay? Ruk ja, Mumbai ruk ja. Abhi chemical control is left, right? So let us just see chemical control. Now, chemical control of respiration, this is done with the help of chemoreceptors. Chemoreceptors, right? Beta, refresh kar do. हो गया? Okay. So chemical control of respiration, there is chemoreceptors. This is with the help of chemoreceptors, and there are two types of chemoreceptors. There is peripheral chemoreceptors and there is central or medullary chemoreceptors. Right? Peripheral chemoreceptors are of two types. There is peripheral chemoreceptors are of two types. There is carotid bodies, and aortic bodies. Now, where are these aortic bodies? Aortic bodies are along the arch of aorta. They are along the arch of aorta. These are your aortic bodies present over here. The sensory fibers from the carotid bodies are the 9th nerve, and from the aortic bodies are the 10th nerve, right? Glossopharyngeal and the vagus. Glossopharyngeal and the vagus. Okay? Okay. So this is carotid bodies and aortic bodies. So these are your afferent nerves, that is 9th nerve and 10th nerve. Please understand that whenever the chemoreceptors are stimulated, stimulation of the chemoreceptors, whether it is peripheral or central, the impulses go to the respiratory center and they increase the rate and they increase the depth of ventilation. They increase both the rate and the depth of ventilation. Mumbai, see this. Increase the rate, increase the depth of ventilation, right? Now let us see what are the stimuli for the peripheral chemoreceptors. Let's see the stimuli for the peripheral chemoreceptors. Let's see this. Yes, the stimuli for the peripheral chemoreceptors are decrease in PO2. This is the most potent stimulus for the peripheral chemoreceptors. Most potent stimulus for the peripheral chemoreceptors is hypoxia, decrease in PO2, right? Number 2, it can also respond to increase in PCO2, increase in H+, acidosis, decrease in pH. Remember we said acidotic breathing. This will be because now it stimulates the chemoreceptors. In chemoreceptors there is an increase in the rate and depth of ventilation. Right? So in decrease in O2, increase in CO2, increase in H+, decrease in pH. It could also be because of increase in lactic acid, increase in K plus. This is important. This is responsible for exercise-induced hyperventilation. One of the reasons for exercise-induced hyperventilation is increase in potassium. Serum potassium levels get elevated in exercise, and this could be one of the reasons for exercise-induced hyperventilation. Also in cyanide poisoning, cyanide causes a direct stimulation of the peripheral chemoreceptors. Direct stimulation of peripheral chemoreceptors. Also agents such as nicotine, lobeline. These are agents which produced histotoxic hypoxia. Cyanide, nicotine, lobeline. These are agents which produce histotoxic hypoxia. Right? And they can directly stimulate. Cyanide is, even in small quantities, can stimulate the peripheral chemoreceptors. Nicotine and lobeline have to be in sufficient amounts. Sufficient quantities, sufficient amounts to stimulate the peripheral. But all three of them, please understand, they produce a histotoxic hypoxia. So this is your, these are your peripheral chemoreceptors and the stimuli, right? Most important stimulus for the peripheral chemoreceptors is decrease in PO2, right? Hypoxia. So let's try and answer this question. This says peripheral chemoreceptors will be stimulated by increase in PO2, decrease in PCO2, decrease in H plus, or by cyanide. Which of these can stimulate the peripheral chemoreceptors? Bolo. D, isn't it? Cyanide will directly, please understand, increase in PO2 will not stimulate. It is a decrease in PO2. Decrease in PCO2 will not stimulate. It is an increase in PCO2. Decrease in H plus nahi, increase in H plus. But cyanide can directly stimulate the peripheral chemoreceptors. Okay? Next, let's see what are the medullary or the central chemoreceptors. Medullary or the central chemoreceptors. Now these medullary or the central chemoreceptors, they are on the surface of the medulla. This is your central chemoreceptors on the surface of the medulla. Now my first question is, the most potent direct stimulus for the central chemoreceptors, is it H plus or is it CO2? The answer is H plus. H plus is the most potent direct stimulus for the central chemoreceptors. But the problem is, if H plus increases in the blood, supposing there is H plus increase in the blood, this H plus cannot cross the blood-brain barrier. We've already discussed the blood-brain barrier in the morning or yesterday. I said blood-brain barrier is formed by astrocytes or by tight junctions between the endothelial cells. Of course, it is both, right? But major contribution is by the tight junction. So this H plus cannot cross the blood-brain barrier. On the other hand, CO2 being a gas can easily cross this blood-brain barrier. I've already told you CO2 can move across, gases can easily move across these cell membranes. So CO2 crosses the blood-brain barrier. Now CO2 reaches the surface of the medulla, it combines with water to form H2CO3, which splits up into H plus and HCO3 minus. Now this H plus will stimulate the respiration. It stimulates the respiration. Please understand, the most potent direct stimulus is H plus. But H plus cannot cross the blood-brain barrier. CO2 can. CO2 crosses the blood-brain barrier, combines with water to form H2CO3, which splits to form H plus and HCO3 minus. Right? And the H plus stimulates the ventilation. So my next question is, what is the most potent stimulus for the central chemoreceptors in the blood? H plus या CO2, then my answer becomes CO2, right? Next question, what is the most potent stimulus for the central chemoreceptors in the CSF? In the CSF, is it H plus or is it CO2? Bolu? H plus. Now there is no question of a blood-brain barrier, so it becomes H plus, right? And my last question related to this, not really my last question actually, which is the most potent stimulus for respiration? Is it hypoxia or is it increase in PCO2? Now my answer becomes most potent stimulus for respiration is increase in PCO2, because please remember that PCO2 affects both peripheral as well as central chemoreceptors. Also answer 2 more questions or 3 more questions. Let's see this. Supposing I give a patient IV lactic acid. When I give, now why have I given this patient IV lactic acid? Don't ask me. We've given him IV lactic acid. Now when I give him IV lactic acid, there is increase in the rate and there is increase in the depth of ventilation. And this is due to action on the central chemoreceptors or the carotid bodies. Bolo? Nahi na, Dehradun, it cannot act on the central chemoreceptors. Why it cannot act on the central chemoreceptors? Because of the blood-brain barrier. But it can act on the carotid bodies. If you remember, we've done this, that the stimuli for the peripheral chemoreceptors is H plus and lactic acid. Have you got it? We've done the stimuli for the peripheral chemoreceptors. There is H plus or lactic acid. But it cannot help my it will not stimulate the central chemoreceptors. Why? Because like I said, there is the blood-brain barrier. Next question. Supposing I remove the peripheral chemoreceptors. Supposing I remove the peripheral chemoreceptors, what will happen to the hypoxic drive? Hypoxic drive decreases by 100%, 70%, 50% or 30%. Hypoxic drive decreases by. Bolo. What is hypoxic drive? Hypoxic drive money? Hypoxia as a stimulus for respiration. Good, Karamsad. It is loss of hypoxic drive is by 100%. If I remove the peripheral chemoreceptors, the CO2 drive decreases by. Please understand, CO2 acts on both peripheral as well as central chemoreceptors. So decrease in the CO2 drive will be by 100%, 70%, 50%, 30%. 50% made it is a smart answer, but not the answer. Bolo. It's actually 30%. That means the CO2 action of CO2 is 30% on peripheral chemoreceptors, 70% of the action of CO2 is on the central chemoreceptors. Right? So this is your, as far as your chemical and neural and chemical control of respiration is concerned. Yes? Let's have a look at, let's have a look at the respiratory reflexes. Respiratory reflexes, my next topic, respiratory reflexes. Now, respiratory reflexes, the first one is what is known as the Herring-Brewer inflation reflex. What is Herring-Brewer inflation reflex? Please understand, whenever the lung inflation is more than 1 to 1.5 liters, it stimulates the pulmonary stretch receptors. It stimulates the pulmonary stretch receptors, and when the pulmonary stretch receptors are stimulated, impulses go via the vagus nerve to the respiratory center, and now they decrease the they inhibit the inspiration and increase the expiration time. Inhibit inspiration and increase the expiration time. Please understand, this is whenever lung inflation is more than 1 to 1.5 liters. Herring-Brewer inflation reflex. It stimulates the where are these pulmonary stretch receptors? Please understand, these pulmonary stretch receptors are not located in the lung parenchyma. In fact, they are in the tracheobronchial tree. Tracheobronchial tree has got the pulmonary stretch receptors. Pulmonary stretch receptors are present in the tracheobronchial tree. Then via the vagus nerve, they inhibit inspiration and increase the expiration time. Next is the Herring-Brewer deflation reflex. Now, whenever there is a marked deflation of the lungs, it now decreases the expiration time. This is Herring-Brewer deflation, reverse of the inflation reflex, right? It decreases the expiration time. Third, you have what is known as the Head's paradoxical reflex. Now, Head's paradoxical reflex, why is it called a paradoxical reflex? Is because over here, inflation produces a further inflation. Inflation is producing a further inflation. Inflation of the lung produces a further inflation. That is called, because remember I just did Herring-Brewer inflation reflex where I said inflation in fact inhibits inspiration and stimulates the expiration. Right? Here the inflation is producing a further inflation. Now, what is the exact physiological significance of this reflex? We do not know. But we do know that the first breath of a newborn is because of the Head's paradoxical reflex. First breath of the newborn is because of the Head's paradoxical reflex. This is your Head's paradoxical. Inflation produces a further inflation. First breath of the newborn is because of the Head's paradoxical reflex. Last reflex is the J receptor reflex. Now, the J receptors are very important for us, especially in India. This is because J receptors were discovered in India, right? J stands for juxtacapillary receptors. These are your juxtacapillary receptors, J receptors, right? They were discovered in India. These receptors are in the alveolo-capillary junction. Alveolo-capillary junction. And like I said, they were discovered in India. There is one Dr. Avtar Singh, actually was one. Dr. Avtar Singh Paintal, very famous Indian physiologist, who discovered the J receptors in 1970. 1970. There are very few things which are discovered in India, and this being one of them, that is J receptors. Yes, so this is in 1970, Dr. A.S. Paintal, Avtar Singh Paintal, he discovered the J receptors. Now, what is this J and what does J? J stands for juxtacapillary. They are in the alveolo-capillary junction. Now, what are the stimuli for the J receptors and what is this J receptor reflex? Let's see that. Whenever, exactly mirrored, pulmonary edema, whenever there is a hyperinflation of the lungs, whenever there is pulmonary congestion, whenever there is pulmonary edema or pulmonary embolism, these situation will stimulate the J receptors, and when the J receptors get stimulated, there is a reflex which is seen. And what is these are the conditions. Please understand, this is different from the Herring-Brewer. Herring-Brewer was pulmonary stretch receptors, and that was at a lung inflation of 1 to 1.5 liters. This is hyperinflation, right? Pulmonary congestion, pulmonary edema, pulmonary embolism, it stimulates the J receptors, and what do I get? The response is, first there is an apnea followed by rapid shallow breathing. Apnea followed by rapid shallow breathing. This has been asked as a question, which are the receptors for rapid shallow breathing? So your answer becomes J receptors, rapid shallow breathing, apnea followed by rapid shallow breathing. Then there is a hypotension and there is a bradycardia. Bradycardia. So this is your J receptor reflex. And see, clinical application of these, sometimes when you have a patient of pulmonary edema, he's not responding, he has hypotension, which is not responding to treatment. You've started him on pressors, you've started him on pressor agents to increase the blood pressure, but for some reason he's not responding. And that is the time when they say this is because of the J receptors. J receptors are producing a hypotension, right? So this is your J receptor reflex. There's apnea followed by rapid shallow breathing, and like I said, there's a question which has been asked, what are the receptors for rapid shallow breathing? My answer becomes J receptors, hypotension and bradycardia. This is your J receptor reflex, right? So these are the four reflexes which I spoke about: Herring-Brewer inflation, Herring-Brewer deflation, Head's paradoxical, and the J receptor reflex. Clear? Now, just a last few questions and then we'll wind up with respiratory system and we'll go on to the next one. And that is, number one, the first question is, what are the receptors which are responsible for tidal respiration? For tidal respiration, which are the receptors responsible for tidal respiration? Is it the chemoreceptors? Can't be, na? Chemoreceptors are stimulated only when, Mysore, I'm just coming to it. Chemoreceptors will be stimulated only when there is a decrease in PO2 or an increase in PCO2. So tidal respiration, this does not happen. So chemoreceptor, no. Could it be pulmonary stretch receptors? Again, no, because they also will be stimulated when the lung inflation is about 1.5 liters. So no. Could it be J receptors? Again, no. Why? Why not J receptors? Because they're stimulated only under those four conditions: hyperinflation, pulmonary congestion, edema, embolism. Right? Therefore, is it the muscle spindles in the diaphragm and the intercostal muscles? In fact, these are the ones which are responsible for tidal respiration. Muscle spindles in the diaphragm and the intercostal muscles. Right? So this is what is your tidal respiration, right? Next, let's see what happens during exercise. Let's see what happens during exercise. I'll just tell you my soul. Now, during exercise, especially if it is mild to moderate exercise, see, mild to moderate exercise, there is mild to moderate exercise, the PO2, the PCO2, the H+, the pH are all normal. There is no change in the PO2, though there is increased requirement of oxygen. But please remember, in exercise, you also hyperventilate. There is increase in rate and increase in the depth of ventilation. So there is more supply of oxygen. So PO2, PCO2, H+, though CO2 production has increased, but because the rate and depth of ventilation increases, CO2 levels remain normal. So in mild to moderate exercise, PO2, PCO2, H+, and pH remains normal. So that means mild to moderate exercise, which are the receptors which are responsible? Which are the receptors? Mild to moderate exercise, you know there is an increase in the rate, there is increase in the depth of ventilation. So which are the receptors which are responsible? The receptors here which increase the rate and depth of ventilation, these are in fact the joint proprioceptors. See, when we are exercising, when you're running, your joint proprioceptors are getting stimulated. These joint proprioceptors, they send impulses to the respiratory center. They send impulses to the vasomotor center. They send impulses to the respiratory center. Please remember, they are both located in the medulla. They are very close to each other, and Mysore, this is where you see the spillover of impulses. Vasomotor center is getting stimulated, impulses spill over to the respiratory center. So what do I get? Now, because of this, vasomotor center, there is increase in blood pressure, increase in heart rate. In respiratory center, there is increase in the rate and increase in the depth of ventilation. So it is the joint proprioceptors which are involved, right? But if there is a severe exercise, there is a fall in PO2, increase in PCO2, increase in H plus, and some decrease in pH. Please remember, now there's going to be a mismatch between the demand and the supply. And this stimulates the chemoreceptors. And whenever the chemoreceptors get stimulated, there is an increase in the rate, there is increase in the depth of ventilation. So please remember, in severe exercise, it is joint proprioceptors plus the chemoreceptor response. Plus, I also told you that increase in K plus, which is responsible for exercise-induced hyperventilation. Mirrored, so exercise-induced hyperventilation. Right? So this is your, please understand, mild to moderate exercise, joint proprioceptors are the primary reason, because of which there is increase in blood pressure and heart rate and increase in rate and depth of ventilation. The severe exercise, the joint proprioceptors ke saath saath there is stimulation of the chemoreceptors, which also increase the rate and depth of ventilation. Plus, like I said, there may be a role of potassium, exercise-induced hyperventilation. Right? So I think that takes care of Mysore. Mysore also has asked me ramp signal. See, what is ramp signal? This I've already told you. What is the ramp signal? But your flow volume curve, Batinda, it will be done in medicine. That is why I'm not doing it with you. If you feel that you need to do it, if you have not done it in medicine, or if you still feel, we can go over it to the next weekend. Let this weekend be, right? Now, like I said, what is the ramp signal, Mysore? Basically, remember I said the DRG cells, this is the eye neurons. The DRG, which has got eye neurons, it sends impulses to the phrenic nerve nucleus. Phrenic nerve nucleus now causes contraction of diaphragm, right? So when the DRG, this sort of starts firing, the eye neurons of the DRG start firing, right? So this now stimulates the phrenic nerve nucleus. Phrenic nerve nucleus now causes contraction of the diaphragm, and when the diaphragm contracts, there is the lung inflation, right? Lung inflation. Then now the eye neurons, when the eye neurons stop firing, now there are no impulses from the DRG, right? This is being controlled. Of course, DRG is being controlled by a pacemaker. Pacemaker sends impulses to the DRG. DRG neurons start firing. So it's like a ramp. There is a steady increase in the firing rate of the DRG. Impulses go to the phrenic nerve nucleus, and from the phrenic nerve nucleus then it causes contraction of diaphragm. Now, when the DRG stops firing, no impulses to the phrenic nerve nucleus now, and the diaphragm now moves upwards. So there is expiration, right? Again, the pre-Botzinger complex tells the DRG to increase the firing rate, then again stops. So this looks like a ramp signal, and that is why it's called an inspiratory ramp signal. Ludhiana, potassium comes from the muscles. Muscles release potassium during severe exercise, and this has a role to play in. What is this, Bikaner? What is the intrapulmonary pressure volume loop? Ye kaun sa hua? Manipal, potassium, please remember, peripheral chemoreceptors ko stimulate kar raha hai. Remember? Chennai, normal lung capacity. I gave you the values in the morning. Total lung capacity 6 liters in males, 5 liters in females, and if the gender is not 6,000 ml in males, 4,200 in females. But if gender is not specified, then stick to 5 liters. Chennai. So it's 1 liter. Normal lung capacity. Total lung capacity beta is 6 liters in the case of males, 4.2 liters in the case of females, total lung capacity, and 5 liters approximately. Repeat the ramp. See, what is ramp? Nothing. Ramp क्या होता है? Ramp is क्या? Chennai, I don't understand your question. Chennai, you have to be more specific. I cannot understand your question. Now, what is DR? What is this ramp signal? I said DRG, the eye neurons have started firing, right? Impulses go to the phrenic nerve nucleus, contraction of diaphragm, lung inflation. Yes? This is the DRG responds to the pacemaker. Pacemaker is your pre-Botzinger. ठीक है? Pre-Botzinger ने DRG को बोला आप अपनी firing rate बढ़ाएं. So firing rate increases. Then pacemaker cells now stop. The firing rate has to stop. So firing rate stops. The DRG stops firing. No impulses from DRG to the phrenic nerve nucleus. If no impulses to the phrenic nerve nucleus, the diaphragm relaxes. This is expiration. Then again under the influence of the pacemaker cells, there is increase in the ramp, increase in the signals from the DRG, which is again looks like a ramp. So this is called the inspiratory ramp signal. Inspiratory ramp signal. Chennai, what does it say? Hemoglobin. I'll take a break for 10 minutes. What is Chennai saying? What reflex? Hemoglobin reflex occurs when lung capacity. Oh, Chennai, nahi bhai. एक पानी का बॉटल दूसरा मंगाना. Okay, so let's see this. We had finished with respiratory system, but of course one part which was left was hypoxia. So let's do the different types of hypoxia. Now as far as the hypoxia goes, दूसरा पेन दूसरा. Hypoxia. Now what are the different types of hypoxia? Let's see this. The different types of hypoxia are hypoxia. Number one is hypoxic hypoxia. Hypoxic hypoxia. पेन देख बेटा कहाँ है? दूसरा पेन किधर है? दो थे ना यहाँ पर. वो ले गया. क्यों? उसके हाथ में देखा. ये वाला पेन ले गया. ये क्यों लेके गया? उसे वापस ले. तो ये इधर टेक नोट का है ना. हाँ तो वो ले गया. ये तो मैं hypoxic hypoxia. Hypoxic hypoxia. This is due to number one, it could be due to high altitude. This is high altitude, right? High altitude, I've already discussed with you why in high altitude there is hypoxic hypoxia, because as we ascend upwards, the barometric pressure decreases and the PO2 reduces. So high altitude, or it could be due to hypoventilation due to any cause. Hypoventilation due to any cause. Maybe there is a bulbar infarct. Bulbar infarct. This will give rise to what is known as a hypoxic hypoxia. Hypoventilation due to any cause. Right? This is hypoxic hypoxia. Right? Now, as far as how do I diagnose the hypoxic hypoxia? Do a PO2. If PO2 is less than 60 millimeters of mercury, that means this is hypoxic hypoxia. Next point, is there going to be any peripheral chemoreceptor stimulation in hypoxic hypoxia? Yes, obviously, because the PO2 is reduced. So there will be stimulation of peripheral chemoreceptors in hypoxic hypoxia. Second type of hypoxia is the anemic hypoxia. And anemic hypoxia, I have already discussed it with you. It could be either because of a reduction in hemoglobin, or it could be due to a reduction in saturation. You remember we discussed this. This is methemoglobinemia, or it could be because of carbon monoxide poisoning. Methemoglobinemia or a carbon monoxide poisoning. This will cause what is known as anemic hypoxia. Now, how do I diagnose anemic hypoxia? Now, for anemic hypoxia, I must do hemoglobin. I must do the saturation, because remember I said either hemoglobin is less or the saturation is less. For example, in methemoglobinemia or in carbon monoxide poisoning, right? But here the PO2 is normal. PO2 is normal. The dissolved oxygen is normal. Will there be any peripheral chemoreceptor stimulation? No. Why will there be no any peripheral chemoreceptor stimulation? Because the PO2 is normal. Please remember, we have already discussed this, that PO2, the arterial oxygen tension will depend upon the alveolar oxygen tension. Yes, and alveolar partial pressure of oxygen, PaO2, right? So this is nothing to do with hemoglobin. Whereas in anemic hypoxia, the problem is with hemoglobin. Either the hemoglobin itself is reduced or its saturation is less. So PO2 over here is normal. The third type of hypoxia is a stagnant hypoxia. Stagnant hypoxia. Now, what is stagnant hypoxia? This is seen in cardiac failure, for instance. There is a stagnation of the blood flow. Stagnation of blood flow. Yes. Now here, how do I diagnose stagnant hypoxia? I do an arteriovenous oxygen difference. Your normal arteriovenous oxygen difference, this is 95, this is 40. Your normal arteriovenous oxygen difference, this is 95 and 40, right? But in stagnant hypoxia, this arteriovenous oxygen difference increases. There is increase in the arteriovenous oxygen difference. This is stagnant hypoxia. Stagnant hypoxia. Will there be any peripheral chemoreceptor stimulation? Right? Arteriovenous oxygen difference increases. This is in stagnant hypoxia. See, normal is 95 and 40. Now what has happened is blood flow is very slow. Blood flow is very slow. So more time for the oxygen to diffuse into the tissues. Tissues are continuously using up oxygen. So the venous O2 reduces. Arteriovenous oxygen difference increases. This is stagnant hypoxia, right? Now, is there going to be peripheral chemoreceptor stimulation over here? And the answer is, in spite of the fact that, please understand, arterial O2 is normal. In spite of that, there is a peripheral chemoreceptor stimulation. Now, why does this peripheral chemoreceptor stimulation occur? Because please remember, the peripheral chemoreceptors, they have a very, very, very high rate of blood flow. They have an extremely high rate of blood flow. And when they have a very, very, very high rate of blood flow, see what is the blood flow? The brain has a blood flow rate of 52 ml per minute per 100 gram of tissue. And the peripheral chemoreceptors have a blood flow rate of 2000 ml per minute per 100 gram of tissue. If you can appreciate, there is almost a 40 times increase in the blood flow rate to the peripheral chemoreceptors. Huge, very, very high rate of blood flow. But please remember, the peripheral chemoreceptors by themselves are hardly 2 to 3 grams. They're very small. They're 2 to 3 grams. But the flow rate, when I want to compare the flow rate, I have to make it per 100 gram of tissue, because only then I can compare it. Have you understood? So this is brain and the peripheral chemoreceptor, very high rate of blood flow. Now, whenever there is cardiac failure, whenever there is stagnant hypoxia, what has happened now? Now, this decreases the total, there is a decrease in the total volume of blood delivered. There is a decrease in the total volume of blood delivered to the peripheral chemoreceptors in unit time. There is a decrease in the total volume of blood delivered to the peripheral chemoreceptors in unit time. Yes, the carotid and the aortic bodies are used to an X amount of oxygen reaching them in unit time. Now, the total volume of blood reaching the chemoreceptors in unit time has decreased. So, the peripheral chemoreceptors will interpret this as hypoxia, and that is why there is an increase in it stimulates the peripheral chemoreceptors. Let's have a look at all the three types of hypoxia. The first one I said is hypoxic hypoxia because of high altitude and because of hypoventilation. How do I diagnose hypoxic hypoxia? Whenever the PO2 is less than 60. Peripheral chemoreceptor stimulation? Yes, because obviously PO2 hypoxia is a PO2 less than 60 is a very powerful stimulus for the peripheral chemoreceptors. Anemic hypoxia. How do I, anemic hypoxia, either a problem with hemoglobin or a problem with saturation. This is seen in methemoglobinemia or in carbon monoxide poisoning. How do I diagnose this? I do a hemoglobin, a saturation, a PO2. PO2 is normal here. Therefore, there is no stimulation of the peripheral chemoreceptors. In stagnant hypoxia, how do I this is seen in cardiac failure. How do I diagnose? There is an increase in the arteriovenous oxygen difference. Normal is 95 and 40. Here now it may be 95 and 20. Yes, there is now, there is more time for the oxygen to be used up by the tissues. So there is an increase in the arteriovenous oxygen difference. Peripheral chemoreceptors, yes. Why? Why is there a stimulation of the peripheral chemoreceptors in spite of the fact that arterial O2 is normal? This is because the total volume of blood being delivered to the peripheral chemoreceptors in unit time decreases. So oxygen delivery to these to the peripheral and to the peripheral chemoreceptors reduces. It stimulates them. The last type of hypoxia is what is known as histotoxic hypoxia. Histotoxic hypoxia. And histotoxic hypoxia is seen in cyanide poisoning, nicotine poisoning, and lobeline poisoning. And how do I diagnose? So what is histotoxicity? That means now the tissues are unable to utilize oxygen. Right? Tissues are unable to utilize oxygen. So what happens over here? Here the arteriovenous oxygen difference decreases. Instead of being 95 and 40, it may be 95 and 70 because the tissues are not being able to utilize the oxygen. So there is a decrease in the arteriovenous oxygen difference. Will there be any peripheral chemoreceptor stimulation? Yes, no. Answer. Will there be any peripheral chemoreceptor stimulation in histotoxic hypoxia? Meerut, Hyderabad, Karamsad, all wrong. Aligarh, wrong. Chennai, wrong. Vishakhapatnam, yes. There will be a peripheral chemoreceptor stimulation. Please remember, I told you at that time when I gave you the stimuli for the peripheral chemoreceptors. Remember, I told you that cyanide, nicotine, lobeline can directly stimulate the peripheral chemoreceptors. We did a question also. Do you realize how volatile our memories are? So volatile. So please, 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 when you go back, my request to you is whatever we have done today, you must please revise whatever is done, isn't it? So otherwise, all this what we've done is not going to be helpful at all, isn't it? So please ensure that you revise. The moment you put a gap, you've got some time to revise, but I suggest maybe not today, but definitely tomorrow in the morning, sit down for 2-3 hours and go through your notes. So that would help you the maximum. That will help to consolidate whatever you have learned, right? More the gap between what we do today and your revision, and you'll go back to resting membrane potentials. Yes, so there will be all this will be difficult to remember then, right? So this is, so my Asan side question now. Which type of hypoxia there is no peripheral chemoreceptor stimulation? In which type of hypoxia there is no peripheral chemoreceptor stimulation? No peripheral chemoreceptor stimulation. Is it histotoxic, anemic, stagnant? Yes, this is the anemic hypoxia. Anemic hypoxia, there is no peripheral chemoreceptor stimulation. Okay? Another question, carbon monoxide poisoning is a type of. Carbon monoxide poisoning is a type of. Is it anemic hypoxia or is it histotoxic hypoxia? Actually, please remember, it is both. But I will mark anemic as the answer. Why not histotoxicity? But please remember, histotoxicity is seen only when the carbon monoxide levels are 1000 times above the lethal limit. 1000 times above the lethal limit. Only then you see histotoxicity in carbon monoxide poisoning. Okay? So this is your carbon monoxide poisoning, which will result in, which is primarily causes, and the patient will die of anemic hypoxia, not of histotoxicity. Histotoxicity is seen only when the carbon monoxide levels are 1000 times above the lethal limit. Right? Okay. Next, let's see. Next, let's see what are, I know there is somebody who asked me what are perfusion and diffusion limited. Chennai, I was trying to overlook that. Yes. Bulbar infarct means bulb. What is the bulb? Bulb is the medulla. So there's an infarct in the medulla. So that is why it's causing hypoventilation. That means the respiratory center has been affected. What is hypoxemia? Anoxia means, of course, complete lack of oxygen. But what is hypoxemia? Hypoxemia is defined at a PO2 of less than 80. PO2 of less than 80. Pulse oximetry. Pulse oximetry क्या है? Pulse oximetry, what happens to pulse oximetry readings in carbon monoxide poisoning will be low, na? Saturation. See, what is your pulse oximetry telling you? Saturation. So saturation means saturation is low. We've already discussed that. There will be decrease in the height of the curve. So there is a decrease. So it's a low value. Okay? Okay. Now diffusion and perfusion limited substances. Let's see this. What is diffusion and perfusion limited? Please listen to this very carefully. Again, there is a lot of understanding involved here. Chennai has asked me diffusion and perfusion limited. So let me just solve it. Let me just solve this confusion, or at least help you sort it out. What is this? See, we have already discussed that you've got the, this is the alveolar O2 and this is the pulmonary capillary over here, right? We know that oxygen moves from the alveolus into the pulmonary capillary and CO2 moves in this direction. Yes. Now, if I look at what is the pulmonary capillary transit time? What are pulmonary capillary transit time? What is pulmonary capillary transit time? Now, pulmonary capillary transit time, that means for how long does the blood stay in the pulmonary capillary is It is 0.75 to 0.8 seconds. This is the time for which the blood stays in the pulmonary capillary. That means this is the time available for the diffusion of oxygen and carbon dioxide. Now, this is a pulmonary capillary, this is the 0.8 seconds pulmonary capillary transit time. This is the arterial end of the pulmonary capillary. Arterial end means that is the pulmonary artery or the venous end of the pulmonary capillary, right? And that means, and this is the venous end of the pulmonary capillary. This is the arterial end of the pulmonary capillary. Arterial means pulmonary vein. So what do I find? I find that the PO2 rapidly rises. It reaches a point of equilibrium with the partial pressure of oxygen in the alveolus. It reaches a point of equilibrium and then it flattens out like this. Yes? That means I can say this is PO2 levels. That means I can say the PO2 levels, the diffusion of oxygen is complete. And when do I say that the diffusion of oxygen is complete? When it reaches a point of equilibrium. And this is the point of equilibrium, right? And to reach a point of equilibrium, oxygen is taken only 0.3 seconds, right? Diffusion of oxygen is complete in 0.3 seconds. Okay? Now let us see PCO2 levels. PCO2 levels have to decrease, and in fact the diffusion of carbon dioxide is even more rapid, and carbon dioxide also reaches a point of equilibrium well within the 0.3 seconds. In other words, I can comfortably say I can comfortably say that the PCO2 levels and the PO2 levels, or the diffusion of oxygen and carbon dioxide, is complete in the first 0.3 seconds, right? The first 0.3 seconds, this is because of like I said, the diffusion is complete within the first 0.3 seconds because they've both reached a point of equilibrium. In other words, now, what is the advantage? See, the diffusion of gases is taking only 0.3 seconds, but the capillary transit time is 0.8 seconds. The diffusion is only taking 0.3 seconds, right? So what is this advantage of the remaining 0.5 seconds? The advantage is that whenever supposing there is increase in cardiac output, the blood flow rate increases. If blood flow rate increases, then the pulmonary capillary transit time decreases, right? Pulmonary capillary transit time decreases. And if the pulmonary capillary transit time, instead of 0.8 seconds, it becomes 0.5 or 0.6 or 0.5 seconds, it will not affect the diffusion of my gases. Why? Because the diffusion of gases is taking only 0.3 seconds. Have you understood? Right? So this is the advantage of the remaining 0.5 seconds. If there is an increase in the cardiac output, if there is increase in the blood flow rate, the capillary transit time will reduce. In spite of that, there will be no effect on the diffusion of gases, right? Because diffusion of gases is taking only 0.3 seconds. Okay? Now, let us see how does there are two types of gases now. There are perfusion or flow limited gases. Perfusion of flow limited gases, and there is a diffusion limited gas. Perfusion of flow limited gas is a typical example is nitrous oxide. Nitrous oxide. Now nitrous oxide is a gas which diffuses very rapidly and has a very low reactivity with hemoglobin. So what do I find? This is the pulmonary capillary transit time of 0.8 seconds. The partial pressure of nitrous oxide very rapidly reaches a point of equilibrium. And the time taken by nitrous oxide to reach a point of equilibrium, this is 0.1 second. This is 0.1 second. It reaches a point of equilibrium in 0.1 second. This is the partial pressure of nitrous oxide. It very rapidly reaches a point of equilibrium in 0.1 seconds. This is the perfusion or the flow limited gases, right? Why is it called a perfusion limited gas? Because please remember, the partial pressure of nitrous oxide in the blood is not limited by diffusion. Diffusion took hardly any time. 0.1 second. And supposing I want to increase the nitrous oxide delivery to the tissues, what can I do? How can I increase the delivery of nitrous oxide to the tissues? This can only be done if I increase the flow rate, right? More the flow rate, more will be the amount of nitrous oxide reaching the tissues. So this now becomes the amount of nitrous oxide reaching the tissues in unit time. This becomes limited by flow, not by diffusion, because the diffusion took hardly any time, right? In the diffusion limited gas, a typical example of a diffusion limited gas is carbon monoxide. Now carbon monoxide, please see this. This is the pulmonary capillary transit time of 0.8 seconds. Now carbon monoxide diffuses from the alveolus into the plasma. And then carbon monoxide has a very high reactivity with hemoglobin. So it immediately reacts with hemoglobin. So more carbon monoxide diffuses from the alveolus into the plasma. Very high reactivity with hemoglobin, reacts with hemoglobin. Again, carbon monoxide diffuses from the alveolus into the plasma. Very high reactivity with hemoglobin. So what happens is, we find in that 0.8 seconds, carbon monoxide fails to reach a point of equilibrium. In other words, I can comfortably say that carbon monoxide has not completed, it has not reached a point of equilibrium. So that means the amount of carbon monoxide in the blood is limited by diffusion. The diffusion of this gas itself is not complete in the 0.8 seconds that the blood stays in the pulmonary capillary. So this is my diffusion limited gas. See, diffusion iska complete ho jayega. Point of equilibrium achieve ho jayega. When all the hemes have combined with carbon monoxide. When hemoglobin is 100% saturated with carbon monoxide. That will take a much longer time. That is not possible in this 0.8 seconds. Right? So this is a diffusion limited gas. The diffusion of this gas itself is not complete. Whereas this is a perfusion limited gas. Diffusion is taking hardly any time. CO2 and oxygen are also perfusion limited gases. They've taken more than nitrous oxide, but their diffusion is also complete well within the capillary transit time of 0.8 seconds. See what happens, why am I calling it a flow limited gas? See what happens during exercise. Exercise mein oxygen requirement of the tissues is increased. And how does the body supply more oxygen to the tissues? By increasing the flow rate. So oxygen and CO2 these are your flow limited gases. So easy is our question. Which of the following is a diffusion limited gas? Diffusion limited gas, is it nitrous oxide? Is it oxygen? Is it carbon monoxide? Or is it carbon dioxide or carbon monoxide? So this is, of course, your answer is carbon monoxide, right? But please understand, supposing there is severe exercise, yes? Supposing the capillary transit time is now less than 0.3 seconds. Supposing it is less than 0.3 seconds. Mane, now this is happening in severe exercise. Cardiac output is very high. Pulmonary capillary transit time is less than 0.3 seconds. Now the diffusion of oxygen and carbon dioxide itself is not complete. That means both these gases have now become diffusion limited gases. See, there's a line which is given in your textbook which states that the carbon dioxide and oxygen are perfusion limited gases, but in certain situations they become diffusion limited gases. So which is the certain situation? The certain situation is when the pulmonary capillary transit time is less than 0.3 seconds. Right? So these are your diffusion and perfusion limited gases. Okay, so all right, so this is what, okay, welcome, right? So this is your perfusion and diffusion limited gases. One small question here, which I forgot to tell you at that time. What is the systemic capillary transit time? This was asked last year in All India PG. Systemic capillary transit time is 1 to 2 seconds. Systemic capillary transit time is 1 to 2 seconds. Right? This is this is systemic capillary transit time of 1 to 2 seconds. Okay? Pulmonary capillary transit time 0.75 to 0.8 seconds. Belgaum, the essence of what we have done is these: nitrous oxide, oxygen, carbon dioxide. These are flow limited gases. Why do I call them flow limited gases? Because they're able to complete the diffusion well within the pulmonary capillary transit time of 0.8 seconds. Carbon monoxide is called a diffusion limited gas. Why? Because it is not able to complete its diffusion within the pulmonary capillary transit time of 0.8 seconds. That is the simplest, and the question that will be asked is just this. Diffusion limited gas is carbon monoxide. It is not able to complete its diffusion in the capillary transit time of 0.8 seconds. Chaliye, let's now go on to a little bit about environmental physiology, and the first one which I'm going to take up with you is high altitude. Let's see what happens in high altitude. Now, high altitude, I've already discussed with you. This is what causes hypoxic hypoxia. Yes. Now, as you go to high altitude, the barometric pressure decreases, so PO2 in the atmosphere decreases. Now, when there is a decrease in PO2, the moment you go into the high altitude, it will now stimulate the peripheral chemoreceptors. There is peripheral chemoreceptors get stimulated, and remember, we've just discussed this, that whenever there is stimulation of the peripheral chemoreceptors, there is increase in the rate and there is an increase in the depth of ventilation. Increase in rate, increase in depth of ventilation, right? And this is known as hyperventilation. Hyperventilation. There is increase in rate, increase in depth. Now, whenever there is increase in rate and depth, this produces what is known as carbon dioxide washout. Carbon dioxide washout. Yes? Please understand, what is this carbon dioxide washout? See, whenever we do an increase in rate and depth of ventilation, there is increase in total ventilation, there is increase in alveolar ventilation. And what is total ventilation? What is alveolar ventilation? I've discussed it right in the beginning of respiratory system. Right? And please remember, the PCO2 levels are inverse to the alveolar ventilation. Mane, if I increase the alveolar ventilation, PCO2 levels decrease. In simple words, this is called a carbon dioxide washout. Have you got my point? Like, for instance, if I ask you if the alveolar ventilation has been doubled, what will happen to PCO2 levels? PCO2 levels become half, right? So this is known as carbon dioxide washout, right? And whenever there is a carbon dioxide washout, it causes what is known as respiratory alkalosis. Respiratory alkalosis. Now, what is respiratory alkalosis? Respiratory alkalosis. Now, what is the effect of alkalosis on the peripheral chemoreceptors? Alkalosis inhibits the peripheral chemoreceptors. Have you understood? Alkalosis begins to inhibit the peripheral chemoreceptors. See, now you've gone to high altitude. As it is, the oxygen supply is less, right? And if the peripheral chemoreceptors start getting suppressed, inhibited because of the alkalosis, now there is going to be, now as it is, the oxygen in the atmosphere is less, and if your respiratory system stops responding, that means now, how do the tissues get oxygen, right? So in fact, this is going to be detrimental. Respiratory alkalosis inhibiting the peripheral chemoreceptors. This is not a favorable situation at all. Have you understood? So now, to overcome this inhibitory effect of alkalosis on the peripheral chemoreceptors, the body now undergoes a process of acclimatization. Now, what is acclimatization? Acclimatization. Acclimatization, the minimum time required for acclimatization is 2 to 3 days. Acclimatization minimum time required is 2 to 3 days. Right? The first process which occurs in, this has been asked as a question, how much of time is required? See, if you go to high altitude, the first thing which you are told is, do not undertake any undue physical exercise. Why? At least in the first 2 to 3 days. Why? Because as it is, there is hypoxia in the environment. And your respiratory drive is suppressed because of the alkalosis. So if you undertake any kind of physical exercise, now what will happen? It will worsen the hypoxia. And there is a risk of you going into mountain sickness. Right? So first 2 to 3 days you are advised that please do not undertake any kind of physical exercise. Yes? So like I said, the minimum time required for acclimatization is 2 to 3 days. Now what happens in acclimatization is, number one, there is an increased renal excretion of bicarbonates. There is increased renal excretion of bicarbonates. Right? See, whenever there is a respiratory cause of acidosis or alkalosis, the compensation is renal. So the kidney now starts excreting bicarbonates. So what is the advantage of when there is an increased renal excretion of bicarbonates? The pH now tends to normalize. pH tends to normalize, and when pH tends to normalize, so at least this inhibitory effect of alkalosis is removed. pH tends to normalize so that now hypoxia will be able to drive. Now this drives the ventilation. Hypoxia will now stimulate the ventilation. Right? Increased renal excretion of bicarbonates, pH tends to normalize, and hypoxia drives the ventilation. Are we clear? This is the first thing which will happen in acclimatization. Now tell me, how do I, now this is a very simple question which I'm going to ask you. How do I know, now there's let's see, I've gone to high altitude, yes, or somebody has gone to high altitude and he's trying to ask you, you've told him don't do any physical exercise in the first 2 to 3 days. Minimum physical activity. Physical activity that you allow him is from the bed to the toilet and toilet to the bed. That's it. Because you tell him, you try to explain it to him that your body has to acclimatize. Now he asks you, how do I know that acclimatization is taking place? How do I know that renal compensation is occurring? Bolo, kaise? That there is increased renal excretion of bicarbonates. How do we come to know? How can I? Urinary bicarbonates. Urinary bicarbonates, simpler than that. Simply by taking a history. Simply by asking something. And that is, if he gives you a history of increased urinary output, if he tells you that he is passing more urine, because please remember that there is Mysore, normalization of respiratory rate is very difficult. It is a subjective feeling. It is not possible to quantify on the basis of normalization of respiratory rate. But please remember, increased renal excretion of bicarbonates. See, if there is increased renal excretion of loss of bicarbonates, there will also be increased loss of water. So if he gives you a history of polyuria, there will be a history of polyuria. That means acclimatization is taking place. Have you got my point? Simply that increased renal compensation, by simply if he gives you a history of increased urinary output, that will tell me an indication that there is an increased renal excretion of bicarbonates. Number 2, what happens in acclimatization? Let's see what happens in acclimatization. Number 2. What happens in acclimatization? Number 2, there is now, and we've already discussed this, there is, we haven't. There is increased secretion of erythropoietin. Erythropoietin is secreted from the kidney. 85% of the erythropoietin comes from the kidney. And 15% of the erythropoietin comes from the liver. 85% and 15%. Increased secretion of erythropoietin. See, erythropoietin is a glycoprotein, and what are the stimuli for erythropoietin is number 1, hypoxia. Hypoxia is a very potent stimulus for erythropoietin secretion. Hypoxia, alkalosis, cobalt salts, androgens. These are very potent stimuli for erythropoietin secretion. 85% of the erythropoietin are 100% of erythropoietinogen is from kidney, 85%, and liver 15%. Now, which are the cells in the kidney which are responsible for the secretion of erythropoietin? And that is the interstitial cells in the peritubular capillary bed. Interstitial cells in the peritubular capillary bed. Whereas liver, it is the perivenous hepatocytes. Perivenous hepatocytes, which are responsible for the secretion of erythropoietin. So 15% is from the liver and 85% from the kidney. Interstitial cells in the peritubular capillary bed. That is why, like I said, that is why a patient of renal disease, chronic renal disease or chronic renal in CRF, a patient always has an anemia, and we need to give him erythropoietin injections. So this is and see this, androgens increase erythropoietin. This is one of the primary reasons why the hemoglobin in males and females is different. Males have a higher hemoglobin as compared to females. Primary reason is this: androgens increase the secretion of erythropoietin. Right? It is the of course, there is some amount of menstrual blood loss which occurs in females in the reproductive age group, but the primary reason is because of secretion of androgens. Now, whenever you go to high altitude, there is hypoxia. This stimulates the release of, whenever you go to high altitude, there is hypoxia, and this hypoxia now stimulates the release of erythropoietin from the kidney, primarily from the kidney, and this is what causes the increase in RBC count. Yes. The third, of course, is the oxygen hemoglobin dissociation curve is shifted to the right. Oxygen hemoglobin dissociation, that is not a very helpful, that is really not very helpful. I've done it with you. That altitude, there is increase in 2,3-DPG synthesis, there is increase in 2,3-DPG, and this causes a shift of the oxygen hemoglobin dissociation curve to the right. This is not very helpful because please remember, rightward shift means now there is an increased delivery of oxygen, but rightward shift also means a decrease in affinity. So when the hemoglobin reaches the lungs, it will pick up less oxygen. So this is the least helpful. The shift of the oxygen hemoglobin dissociation curve to the right, like I said, is the least helpful. Then some more changes in the high altitude. There is increased capillary density at tissue level so that the tissues can utilize more oxygen. There is increase in mitochondria. There is increase in the cytochrome enzymes. But these, please remember, are not going to occur in us. These are seen in people who live in high altitude. So these are more at a genetic level. These are seen in people who live in high altitude for a long time. So this is acclimatization. And the questions which have been asked from here is time required for acclimatization. Minimum is 2 to 3 days. Next, let us see. Now, supposing there is some problem in acclimatization. Supposing the hypoxia persists. Hypoxia persists. there is some problem with acclimatization. Now the patient goes into what is known as an acute mountain sickness. And the acute mountain sickness has got two dangerous forms. Two dangerous forms. Two dangerous forms. This is high altitude cerebral edema and high altitude pulmonary edema. High altitude pulmonary edema. High altitude cerebral edema and high altitude pulmonary edema. Now the time required for development of acute mountain sickness, time usually which is taken, this is 1 to 4 days. 1 to 4 days. Generally, but you can also see mountain sickness within the first few hours of landing up in high altitude. Right? Now, the question arises, at what altitude? Like what you've asked me, Jammu, at what altitude? Now, there is no fixed altitude as such. Because please remember, it depends upon a number of things. It depends upon, did you undertake any physical activity? Right? Supposing you were asked not to undertake physical activity in the first 2 to 3 days, but you decide, no, I'm going to ignore these warnings, and you undertake some sort of, you maybe decide, you've reached Leh and Leh Ladakh, and you decide, no, I'm going out for a run on the first day itself. So you've undertaken physical activity and you've worsened the hypoxia. So more chance of developing pulmonary edema, right? Or pulmonary or cerebral edema. Or it also depends upon your rate of ascent. Rate of ascent. If it is a rapid, for example, if you've taken a flight from Delhi to Leh Ladakh, to Leh, the rate of ascent has been very rapid. But if you take a road, you take a journey by road, your rate of ascent has been slower. Yes? But if you decide to walk, then of course it is the slowest rate of ascent. So that means you have more time to acclimatize. This is your rate of ascent. And third, of course, it depends upon your individual susceptibility. Individual susceptibility. Right? Now, what is the reason for this? What is the pathogenesis of the high altitude cerebral edema or high altitude pulmonary edema? Now, what happens in high altitude cerebral edema is, please understand, hypoxia is a very potent stimulus for cerebral vasodilation. It causes vasodilation everywhere, including the cerebral vessels, and whenever there is a cerebral vasodilation, it increases the capillary hydrostatic pressure. We've done this yesterday. If there is increase in capillary hydrostatic pressure, it increases the tissue fluid formation. It increases the tissue fluid formation, and increases the tissue fluid formation means there is now edema. Right? So this is your pathogenesis of cerebral vasodilation, high altitude cerebral edema. There is hypoxia, cerebral vasodilatation, increase in the capillary hydrostatic pressure, increase in the tissue fluid formation, edema. How do I treat a patient of high altitude cerebral edema? Bolo, let me see. Cerebral edema, what can be the treatment? Bolo. Mannitol, okay. Yes, Haldwani, you have to give him oxygen. See, that is the reason why reverse the hypoxia, because hypoxia is the reason why you're getting edema, isn't it? So you need to give him oxygen, immediately give him oxygen. Yes, mannitol. And evacuation of the patient becomes very important. See, the very reason why has he developed high altitude cerebral edema is because he's decided to come to Leh Ladakh for a holiday from Delhi. Send him back. Let him go home. Evacuation of the patient. Of course, he's not going to walk and go back home. You have to evacuate your patient. Mysore carbonic anhydrase inhibitors are more useful in the prevention prevention of high altitude cerebral and pulmonary edema. And why do prophylaxis? Yes, more for prevention. Carbonic anhydrase inhibitors, acetazolamide is a very good drug for prevention, because please remember, the carbonic anhydrase inhibitors or acetazolamide, what does it do? It causes acidosis. One of the side effects of this drug is, besides being a mild diuretic, this causes acidosis. And when there is acidosis, how does it help me in this situation of acidosis? How does acidosis help me? Because acidosis will stimulate his peripheral chemoreceptors and increase his ventilation. So acidosis will be helpful in this situation. That is why you prefer carbonic anhydrase inhibitors to prevent it. But if you have a full-blown case of cerebral edema, acetazolamide is not going to help. You need to give him mannitol, you need to give him oxygen, you need to evacuate your patient. Now, high altitude pulmonary edema. Let's see the pathogenesis of high altitude pulmonary edema. Now, high altitude pulmonary edema would be, see, in hypoxia, in the pulmonary circulation, it produces a pulmonary vasoconstriction. Now, this is unique. Hypoxia produces a vasodilation everywhere, but in the lungs, it produces a vasoconstriction. Why is that so? Because hypoxia, wherever it is producing vasodilation, the channels which are present are ATP-dependent potassium channels. So there is a cerebral vasodilation. But in the lungs, the channels which respond to hypoxia are oxygen-sensitive potassium channels. The pulmonary vasculature has got oxygen-sensitive potassium channels, so they respond by constricting, contraction. Wherever there is vasodilation, the channels present are ATP-dependent potassium channels, but wherever you've got wherever there is a constriction, you have oxygen-sensitive potassium channels. Oxygen-sensitive potassium channels. Right? Pulmonary vasoconstriction, you've got oxygen-sensitive potassium channels. Right? Now, let's see. ATP-dependent potassium channels and oxygen-sensitive. Now, what is happening over here in the case of high altitude pulmonary edema, where it produces a pulmonary vasoconstriction? Please understand, if it is a vasoconstriction, there is no question of edema. Have you understood? If it is ulta of what was happening in the cerebral circulation, there should be no edema here. But please understand, this is a patchy vasoconstriction which is produced. Patchy vasoconstriction money. Now the vasoconstriction, now let's see, this is a pulmonary blood vessel. The vasoconstriction is more severe in those areas where the hypoxia is more, vasoconstriction will be severe in those areas where the hypoxia is more. Mane kya? You know that ventilation is not uniform throughout the lungs. You know that PO2 levels will be different in different areas of the lungs. Right? So wherever hypoxia is more, right, where there is where the PO2 levels are low, the vasoconstriction is very severe in those areas. But what will happen to flow into other vessels, other branches where the vasoconstriction is not so severe? Now I have an increased flow into those areas. So there is a vasoconstriction in some areas, and there is an increased flow to those areas where vasoconstriction, where there is little or no vasoconstriction. Where there is little or no vasoconstriction. So increased flow to those areas where there is little or no vasoconstriction. So what happens now? In these areas, there is increase in PC, and these areas, there will be now a pulmonary edema. So the key word here is, please understand, hypoxia produces a cerebral vasodilation. Vasodilation, why does it produce a vasodilation? Because these areas have got, the cerebral vasculature has got ATP dependent potassium channels. But hypoxia produces a pulmonary vasoconstriction, but the catch is it produces a patchy vasoconstriction. And if there is a patchy vasoconstriction, now there is increased flow to those areas where there is little or no vasoconstriction. The hydrostatic pressure increases in these areas, and this produces a pulmonary edema. Okay? So this is your acute mountain sickness and the pathogenesis. High altitude pulmonary edema, high altitude cerebral edema. Next, let's see, there is also something known as chronic mountain sickness. Now, chronic mountain sickness will be seen in acclimatized individuals. This is seen in acclimatized individuals. Acclimatization is complete, and we still see a chronic mountain sickness. Still the patient goes into high altitude pulmonary edema, and he goes into high altitude pulmonary. So this is in a fully acclimatized individual. That means he is staying in high altitude for 4 to 6 weeks. Fully acclimatized. Right? This is now you see what is chronic mountain sickness. Chronic mountain sickness, like I said, in a fully acclimatized individual. Here, this chronic mountain sickness is also known as Monge's disease. Now let's see the pathogenesis of Monge's disease. See, number one, there is a huge increase in the RBC count. This now causes an increase in the viscosity. Right? Blood viscosity increases. There is increase in viscosity. This now increases the resistance to flow. Right? Second factor which results in the because of polycythemia, yes. Second factor which affects the which causes this problem is because of hypoxia. Because of the pulmonary vasoconstriction, it can give rise to pulmonary hypertension. So increase resistance to flow and the pulmonary hypertension. This results in a right heart failure. Right heart failure leads to a congestive cardiac failure. Both right and the left hearts become involved. And when the congestive cardiac failure develops, now you get the pulmonary edema. Clear? So question, high altitude pulmonary edema is seen only in acclimatized, only in unacclimatized, or both acclimatized and unacclimatized. So your answer becomes both. Right? This is your Monge's disease, chronic mountain sickness. Whenever there is the polycythemia because of high altitude, because of increased secretion of erythropoietin, there is increase in blood viscosity, increase in resistance to flow. Plus there is a pulmonary vasoconstriction which has resulted in a hypertension. Both of these lead to a right heart failure, congestive cardiac failure, and pulmonary edema. Mumbai, will the ventilation perfusion ratio remain the same or decrease in high altitude pulmonary edema? There will be. See, what's happening here is now there are areas you cannot really comment on the VQ ratio, isn't it? Because there is going to be there is decrease in perfusion because of the vasoconstriction, but again it's a patchy vasoconstriction, isn't it? And there definitely will be some effect on the ventilation perfusion ratio, but I'll have to look it up and see if there's actually been, is there any article or is there any research which has been done into VQ ratio at high altitude. We can look it up and see, right? Okay. Now, Manipal, I'll tell you when we reach cardiovascular system. So this is as far as your high altitude physiology is concerned. The next one is deep sea physiology. Now deep sea, the problem in deep sea physiology, high altitude, the problem was low barometric pressure. Deep sea is, Vishakhapatnam, your class is till 9 o'clock. Deep sea physiology, the problem is the high barometric pressure. Right? Yes, I'm coming to nitrogen narcosis, Jammu. This is in deep sea physiology. Now, what happens in deep sea? Let's see that. At sea level, the pressure is 1 atmosphere. So, Jammu, answer this now. For every 33 feet or 10 meters below the surface, the pressure increases by 1 atmosphere. Right? This is sea level 1 atmosphere. At 33 feet, the pressure is 1 atmosphere. So below the water, if you go 33 feet below the water, below the sea level, this is the pressure increases by 1 atmosphere. At a depth of 100 feet, yes, what is going to be the pressure? Raptures of depths, Jammu, bol. How much is going to be the pressure? Jammu, क्या बोल रहा है? 760? At a depth of 100 feet, the pressure will be 4 atmospheres. कैसे 4 atmospheres? 1 atmosphere at sea level, 1 atmosphere at 33 feet, 1 atmosphere at 66 feet, 1 atmosphere at 99 feet. So total मिला के हो जाएगा 4 atmosphere, right? So this is going to be 4, right? So for every 33 feet or 10 meters, right? 10 meters. Now somebody had asked me this has come in DNB this time also. What is the pressure at 100 meters? What is the pressure at 100 meters? Is it 10 atmospheres or is it 11 atmospheres? 11 atmospheres. Karamsad, good. Gulbarga, how did you come at a figure of 3.3? Okay, so at a depth of 100 meters, the pressure will be 11 atmospheres. Please understand, it is 33 feet or 10 meters. One and the same thing. For every 33 feet. Are you deliberately giving me these wrong answers, Rohtak, Gulbarga? Is it because you want me to wind up early? Yes. Power cut here. What? What is not working? Baran is not working. Okay. And neither is mine actually. What, Kolhapur, why were you hyperventilating that there is a carbon dioxide washout? Oh, there was power cut induced hypoxia. Okay. So थोड़ी देर और बैठो. Okay, थोड़ी देर और बैठो and let us just finish this. Yes? Okay, so I said for every 33 feet. Now listen to this. Listen, let's just finish this. Let's finish this and let's be good kids and let's try and be diligent kids and let's finish this. Right? Okay. Now Jammu is not really at high altitude, should not be hyperventilating, right? So, okay. Now, let's see. So let's just continue this. I'm not going to look at the chat box at all. Now this is for every 33 feet below the sea level, the pressure increases by 1 atmosphere. This 33 feet or 10 meters, one and the same thing, right? 33 feet or 10 meters below the earth, below the sea level, the pressure increases by 1 atmosphere. So like I said, at 100 feet, the pressure increases by 4 atmosphere, or at pressure is 4 atmosphere, and at 100 meters, the pressure would be 11 atmospheres. See, what is the problem here now is, whenever the pressure is more, there will be now an increased solubility of gases. There is going to be an increased solubility of gases. If the pressure increases, if the atmospheric pressure increases, there will be an increased solubility. It's like when you see a Pepsi bottle, isn't it? When the Pepsi bottle, when the dhakkan is on the Pepsi bottle, the carbon dioxide, that means carbon dioxide is under high pressure. So the carbon dioxide manages to be in the dissolved state. So now there is an increased solubility of gases. So here we have a diver which is at 120 or 150 feet, yes. We have given this diver 100% oxygen. Now 100% oxygen we've given, you remember we've done this question 4 times atmospheric pressure, and therefore the PO2 will be 3000 millimeters of mercury, dissolved O2 increases, and therefore when you give him 100% oxygen, now you're dissolved, there will be a huge increase in his arterial oxygen tension. And whenever there is an increase in the arterial oxygen tension, this will now result in an increase in free radicals. And the free radicals are O3 minus, H2O2, which now start damaging cell membranes. And this condition is known as oxygen toxicity. Oxygen toxicity. The free radicals are increased, O3 minus H2O2, and what are the symptoms of oxygen toxicity? This diver who is getting 100% oxygen now starts complaining of there is a substernal distress, there is a choking sensation, he starts coughing, and he finds it very difficult to breathe. So this is because of generation of free radicals, right? This gives rise to oxygen toxicity. Right? Now, to minimize the oxygen toxicity, to reduce the oxygen toxicity, what do? We give him an oxygen and an inert gas. And the commonly used gas is going to be oxygen-nitrogen mixture. Like oxygen-nitrogen mixture, right? No, I'll come to helium. First, we give him oxygen-nitrogen mixture. Now, what happens is, now nitrogen is, as it is, a highly soluble gas, right? So now, there is now an increased solubility. There is an increased solubility of nitrogen in the plasma, in the cell membranes, and in the myelin. There is an increased solubility of nitrogen in the plasma, in the cell membranes, and in the myelin. And this increased solubility of nitrogen in the cell membrane, in the plasma, in the myelin, now this alters ionic conductivity. And when it alters ionic conductivity, the patient goes into a condition which is known as nitrogen narcosis. Nitrogen narcosis. Please understand, this nitrogen narcosis is not due to any chemical reaction. Nitrogen narcosis is due to a physical, there is an increased solubility of the nitrogen in the myelin, which has altered ionic conductivity. And this nitrogen narcosis, the patient has symptoms similar to alcohol intoxication. And what are the symptoms of alcohol intoxication? You know them very well, right? He becomes disoriented, he starts behaving in an abnormal manner, starts laughing, starts crying, starts pulling his mask. This is nitrogen narcosis, and this is called raptures of depths. Raptures of depths is nitrogen narcosis, right? And this is, if you've read Tintin comics, you know this is especially that Red Sea wala comic. Uske andar Captain Haddock has this raptures of depths, and people think that's because of he's that is it a drunkard. They start thinking it's because of alcohol intoxication, but actually it is because of nitrogen narcosis. This is called raptures of depths. Now, to minimize nitrogen narcosis, we prefer helium. We prefer an oxygen-helium mixture is preferred. Oxygen-helium mixture is preferred, right? Why do I prefer oxygen-helium mixture instead of oxygen-nitrogen mixture? We prefer helium. Why helium? Now, helium is, number one, a less soluble gas. Number two, it is a less narcotic. It will produce less symptoms, less narcotic symptoms. And number three, it is a less dense gas. Less dense gas, so that means now resistance to flow will be less. There is less resistance to flow. It's a less dense gas and there will be less resistance to flow. So oxygen-helium mixture is preferred. Oxygen-helium mixture is preferred. There is less soluble, less narcotic, and a less dense gas. There is a decreased resistance to flow. Right? This is as far as your see. I don't know if you've ever heard of this. When you have a patient in status asthmaticus, you do not give this person 100% oxygen. You do not give him 100% oxygen, and the reason is obviously you do not give him 100% oxygen is because it will minimize his respiratory drive. It will start affecting his respiratory drive. It will decrease his respiratory drive. So what do? In a patient of status asthmaticus, we give him a oxygen-helium mixture. This is called Heliox. You give him a oxygen-helium mixture. Again, why do I prefer to give him oxygen-helium and not oxygen-nitrogen mixture? The reason is, like I said, helium is a less dense gas. So this patient who's already in status asthmaticus, if I give him a gas which is easier to breathe, it will help him. So it helps him breathe a less dense gas, that is helium. Right? So this is your, why do we prefer helium? Like I said, is because it is a less soluble gas, less narcotic, and less dense. Okay? This is your oxygen-helium mixture. Now this was all these problems, oxygen toxicity, nitrogen narcosis were occurring while he's underwater. Right? This was while he was underwater. This is your, see here, he's underwater, and the problems that he's encountered underwater were oxygen toxicity and nitrogen narcosis. This is because the solubility of the gas is the what? 5 of oxygen. Oh, percentage. Percentage can vary in Heliox, no? But normally when you give up status asthmaticus, you give him 50% oxygen, 50% helium, right? But of course it will vary. When you give it to diver, you get less than 10% of oxygen and 90% of helium, right? I'm coming to Ben's Dehradun. Now this is while the diver is underwater. So the problems he encounters are oxygen toxicity and nitrogen narcosis. Okay? To minimize nitrogen narcosis, we give him helium, oxygen-helium mixture. Now, when the same diver comes to the surface, now he undergoes what is known as decompression sickness. When he comes to the surface, he undergoes a decompression sickness. What is decompression sickness? See, remember I told you the example of a Pepsi bottle. Pepsi bottle with the dhakkan on is carbon dioxide is under high pressure. So Pepsi bottle with the dhakkan on is the diver underwater. with a cap on is diver under the water. High pressure, carbon dioxide is in the dissolved state. But moment you loosen the cap, moment the dhakkan is off, the diver, it's like a diver on the surface. What happens to the carbon dioxide? Carbon dioxide starts bubbling. And that's exactly what happens. Underwater, the gases were in a dissolve. There was increased solubility of gases. But when he comes onto the surface, now the same gases begin to bubble. There is formation of gas bubbles, gas emboli. There is formation of gas emboli. Right? And the most common symptom of decompression sickness is bends. What happens is 95% of the patients suffer from bends. Bends is very severe pain in the joints and the muscles. Very severe pain in the joints and muscles. This is bends. Why? Because these gas emboli go and block the blood vessels in the joints and the muscles. This gives rise to a condition known as bends. 95% of the patients are bends, B-E-N-D-S. Right? 95% of patients suffer from pain, very severe pain in the joints and muscles. That is called bends. Right? 5% of the patients can present with pulmonary symptoms and cerebral symptoms. Right? Can present even in coma. Pulmonary symptoms because embolus in the pulmonary vessels, or cerebral symptoms. This is 5% of the patients can present with even in coma and can even present coma and death. Right? So bends. Please understand, underwater is what he will suffer from: oxygen toxicity, nitrogen narcosis. When he comes to the surface, now it will be decompression sickness. Decompression sickness is also known as Caisson's disease. Caisson's disease. Right? This will be when he comes to the surface. It's like the bubbling of carbon dioxide, which you see when you open a Pepsi bottle. You release the pressure and the carbon dioxide starts bubbling. When the dhakkan is on, it is like a diver underwater. Right? So this is as far as your deep sea physiology is concerned. Caisson's disease. Now there was one question here. Let's see. No, Ludhiana, you must understand, any gas, whatever gas you give, if he comes to the surface very rapidly, if his ascent is very rapid, whatever gas was in the dissolved state in underwater will now start bubbling. Have you understood? Not necessary it has to be oxygen, nitrogen. It can occur with oxygen, helium also. See, that is why these divers have to come very gradually to the surface. And you remember the treatment of decompression sickness? You again put him into a hyperbaric chamber. We discussed an hyperbaric chamber. You increase the pressure, make the gases go into the dissolved state again. Then dhire release the pressure so that this problem is minimized. Yes, of course, nitrogen or helium ke beech mein, helium ke saath, there will be less problem of decompression sickness because helium is a less soluble gas as it is. Okay? So this is your... Now there was a question which was asked, and let me see if you can answer this. It says, the decompression sickness is seen in only divers, only pilots, या divers and pilots. Let me see. Decompression sickness is seen in only divers, only pilots, या divers and pilots. It's actually divers and pilots. Please understand. See, when will a pilot suffer from decompression sickness? Diver, we've understood. But when will a pilot suffer is when he flies his plane at very high altitudes. Planes are pressurized chambers. So these are pressurized. He's not traveling in unpressurized chambers. Planes are pressurized chambers. Yes. Supposing there is a suddenty, supposing there is a mishap at very high altitudes. There is sudden decompression at this high altitude. Now the same gases which are in a dissolved state will start bubbling. Please remember, because as you go high, as you fly high, let's say at a distance of about 11 kilometers from the Earth's surface, the outside temperature is -50 and the pressure is extremely low. Supposing there is a sudden decompression at this very high altitude, this will give rise to decompression sickness. So whatever gas is in the dissolved state will also start bubbling. This is decompression sickness. It will be seen in divers as well as pilots. Next part in environmental physiology is what is positive G and what is negative G. Positive G and negative G. See, skydivers, what are the heights that they are diving from? Yes, you could have a same happens in skydiving. See, skydivers are being exposed to low pressure, and what are the heights that they are jumping from? Yes. And yes, because these and plus they are very momentarily, only for a few seconds are they exposed to these low pressures, right? And please, they're not as high, right? Now, I don't understand there is no pressurized chamber in case of skydiving, but skydivings are moving without oxygen, so it is not at very high. It cannot be very high altitudes. Have you understood? It is not at very high altitudes. So it's definitely not what I was talking about. See, our commercial flights fly at 11 kilometers above the Earth's surface. The jet fighter pilots are flying even higher. Let me see your questions. It says, scene of pilots ascend rapidly, rate of ascent in divers to prevent decompression sickness. The plane they are also open. So okay, so this is your, basically you must understand that the only if planes are pressurized chambers, the jet planes for example are pressurized chambers, and if there is astronauts also can show decompression sickness. Yes, this is because they are moving around in pressurized chambers, so it's sudden decompression, right? I don't know, Belcom, what are you trying to ask me? These figures I will have to, okay, these Jammu, I'll look up these figures and tell you at what these are given. I'll find out these pressures and let you know in the next time. All right. Okay. Okay. So positive G and negative G. Now positive G is whenever, see, what is the G force which is acting on all of us? The positive G which is acting on all of us, this is a force of 1G. The G force which is acting on all of us is 1G. So positive G means when the G force increases. Again, we are not going to experience, we normal individuals are not going to experience a force of positive G or negative G. This is experienced by our fighter pilots. Now fighter pilots, when they are flying their planes, this is a jet plane, especially when they make these loops in the air. Yes. We've done this in physics of class 11, 12. They are going to be subjected to these forces of positive G and negative G, right? Significant amount of positive G and negative G forces. When they are coming down, it's a negative G. When they're going up, it's a positive G. We've done it in physics. Now, what is the effect of positive G? Positive G now, it causes a peripheral pooling of blood. It causes a peripheral pooling of blood. When there is a peripheral pooling of blood, it decreases the cerebral blood flow. It decreases cerebral blood flow. And this now causes a condition known as a blackout. It causes a condition known as blackout, right? There is a decrease in cerebral blood flow, causes a blackout. And how is this blackout prevented? Our pilots wear special types of suits. Those are called anti-G suits. Anti-G suits. What are these anti-G suits? They inflate around the lower limb veins. Yes. See, what is the effect of positive G? I said positive G force, we are not going to be subjected to positive G and negative G forces, so at least significant positive G and negative G forces. These are our fighter pilots. Fighter pilots, when they are flying the planes, in these, they do these acrobatics in the air. That is the time when they are subjected to these G forces. Yes. Now, G4, whenever there is positive G, when the G force becomes 4G and 5G and 6G, there is a peripheral pooling of blood which decreases cerebral blood flow and causes what is known as a blackout. And to prevent this, our jet fighter pilots, our Air Force fighter pilots, they wear these special suits which are known as anti-G suits. Anti-G suits, right? Now, what is negative G? Anti-G suits. Yes, they inflate around the lower limb waist. Anti-G suits which inflate around the lower limbs. Around the lower limbs. So they prevent the peripheral pooling of blood. That is called the anti-G suits. Yes, these fighter pilots are also trained. Whenever they are exposed to positive G forces, they are also trained. How can you decrease the peripheral pooling of blood? If they are trained to contract their abdominal muscles. Again, if they contract the abdominal muscles, there is less peripheral pooling of blood. So there is a less chance of a blackout. Besides wearing these G suits, which is called the anti-G suits. Okay? So this is the effect of positive G. Now, what is the effect of negative G? Let's see the effect of negative G. Now, negative G, this is when the G force becomes less than 1G. Now, in this case, there is an increased blood flow. There is an increased blood flow towards the head end of the body. Increased blood flow towards the head end of the body. Increased blood flow towards the head end of the body. When there is an increased flow towards the head end, now at the same time, that means there is going to be an increase in the cerebral arterial pressure, right? There is increase in the cerebral arterial pressure. But the cerebral blood vessels usually do not rupture. And why does this happen is because not only is this blood flow increased towards the head end in negative G, there is also an increased in flow of CSF towards the brain, towards the head end. And CSF has a protective effect on the cerebral blood vessels. So rupture of the cerebral blood vessels is uncommon. Rupture of cerebral blood vessels this is uncommon. Why? Because the CSF flow towards the head end also increases. So it prevents a rupture of the cerebral blood vessels. But at the same time, what happens to the scleral and the conjunctival vessels? There is congestion of the scleral and the conjunctival vessels, right? There is a congestion which is seen. Scleral and the conjunctival vessels, and this produces a condition known as a red out. Yes. So again, these questions have been asked. Blackout will be seen whenever there is a positive G force, and red out, please understand, though there is increase in cerebral arterial pressure, but rupture of the cerebral blood vessels is not common. It is not common. This is because of the congestion of the scleral and the conjunctival vessels. Yes, astronauts will also be, astronauts are subjected to anti-G forces, negative G forces. Yes. Now, Mysore, what is the again, how much of positive G that we can tolerate depends on individual to individual. You and I have, in Bangalore there is a simulator. Hindustan Aeronautics Limited had a simulator where they actually, jet fighter pilots, their screening is done. A person who's able to tolerate more G force is selected to become a fighter pilot. Generally about 5 to 6 G. Yes, but like I said, individual susceptibility. You and I will probably have a blackout much earlier, but our jet fighter pilots are trained, and they are also, even their selection is done on this. They're made to sit in a simulator, and they are subjected to high G forces, and then you can see at what G forces is there a blackout. So it is individual susceptibility. Now, negative G, see, the problem with negative G is not much. Like I told you, there is increase in cerebral arterial pressure, but there is because of increase in CSF flow towards the head end, our cerebral blood vessels are protected against rupture, right? It is the congestion of the scleral and the conjunctival vessels which gives rise to readout. Fast moving lift, yes, fast moving lift, but usually will not produce this kind of a blackout, because your lifts are again not going to increase the G force to such an extent that there is going to be a blackout. What is this? Consciousness maintained. Yeah, very much so. Your astronauts, you've seen this now. You've seen those pictures of people in space. They are very much conscious and they can communicate with you. There is no problem at all. They do all their experimental studies in space. Yes? Okay, Gorakhpur, please ma'am, what? Positive G. Okay, positive G force. What is positive G? I said positive G is in positive G, like I said, there is a peripheral pooling of blood which causes a decrease in cerebral blood flow and causes a blackout. It's pretty simple. This is Gorakhpur. This causes a decrease in cerebral blood flow and a blackout. So blackout is seen in positive G, negative G. So your answer should be positive G. In negative G, what you see is a readout. This is because of an increased blood flow towards the head end. Yes, and this is now producing what we call, because of increased flow towards the head end, it also causes congestion of skeletal blood vessels, hence the name readout. Haldwani, the prevention of readout is not such a, Dhule, the prevention of readout is not such a big problem, because it's just causing an increased congestion. It's the blackout which we are bothered about, isn't it? We are bothered about the blackout. This is because of the, so that is why they use anti-G suits. What? Okay, so this is your positive and negative G. Okay. So, all right. We've finished our environmental physiology, and now we can start our next chapter. Yes, let's see our next chapter. Chalo, okay. So, okay. So what I'm going to do is, chalo, let's sit and study for half an hour. Will that be all right? Right? Half an hour? Will that be okay? See, let's be good kids and let's, okay. And okay, so let's continue for half an hour. Yes. Let's take on our next chapter, which is renal physiology. Renal physiology. So let's see renal physiology. Okay. Okay, all right. What I will do is I'll wind up now, but then please next time when I'm next 2 classes, we are not going to wind up early like this. This is only going to affect our. We'll have to go fast, and that will affect us, right? So I'll wind up, and I'll see you next Saturday. Next Saturday at 2 o'clock. So promptly we'll be starting at 2 o'clock, right? And chalo, okay. अब बोल रहे हो कंटिन्यू, पहले बोलते हो नहीं, फिर बोलते हो कंटिन्यू. चलो, ओके फाइन. So it doesn't matter. We'll wind up now, and we will then. Yeah, enjoy your Sunday. Okay, whatever is left of it. Okay, thank you so much. Good night. Yeah, I'll just tell the presenter. He will show you the slides. First half an hour. First half an hour. Bareilly, what do you want? Which slides? बेटा पहला half an hour का slides है? पहला हाफ एन आवर का स्लाइड्स है. हाँ, ये रहा. फर्स्ट 1 आवर. फर्स्ट 1 आवर कैसे होगा इनका? फर्स्ट हाफ एन आवर का देखो. इनका फर्स्ट हाफ एन आवर मिस हुआ है बेटा. इनका दिखाओ स्लाइड्स. अभी उनका टाइम है. ये बता देंगे, बच्चे बता देंगे. अच्छा, ये साइड में करूँ. Where do you want it till Chennai and Bilaspur? Please see this. Please tell me. We've done nerve fiber classification. Then this was. ये कहाँ से आ गया? अच्छा, this is skeletal muscle के proteins. Yes. This is the axoplasmic transport, the skeletal muscle. Tell me Chennai and Bilaspur, till what time, till what do you need? Chennai, nerve fiber classification only. In Bilaspur? First half an hour. बेटा उनको first half an hour का दिखा दो. Recording है क्या अभी? Recording होगा? होगा? They'll show it to you. First half an hour का है? First half an hour का recording? First half an hour का recording है? दिखा दो वो बच्चे, उनको ना उनका मिस हो गया था उनका. बिलासपुर और बरेली. हाँ, स्टार्टिंग हाफ. Bareilly, I think you need your first half an hour. They are saying that you'll have to wait for half an hour because they need to sort of rewind. कल कितना बजे इनका क्लास? 8:30 Bareilly, tomorrow from 8:30 to 2 you have 8 to 2:30 you have a class. So after 2:30 he will show you the first half an hour. ठीक है? ठीक है? देख लेना आप. चलो, okay. Thank you.